Okay, we can start in a few seconds, so we can give time to attendees to join the webinar.
Mm-hmm.
Okay, I think we can start. So, welcome everybody to this session of this last day of the NeuroMatch conference 3.0. So, today I'm Alberto Antonietti from Blue Brain Project Geneva, and our backend host here, our guardian angel is Ali. Thank you very much for helping with the hosting of this session. Today, we will have three great talks that are very related one with each other. And one reason is that all our speakers come from the same institute. So they are studying kind of similar related subjects. So we will have a very coherent session here today. And I would like to invite to the stage our first speaker. She's Andrea Costa, a PhD student at the University Medical Center in Groningen, at the University of Groningen in the Netherlands. So please, Andrea, the floor is yours for your first international talk. So good luck with that.
Thank you very much for the nice presentation. So I will present now the study entitled The Effects of Cognitive and Mechanical Constraints on Modular Neuromuscular Control in Younger and Older Adults During Being Walking. This is a study with international collaboration of the University of São Paulo in Brazil and also the University of Illinois at Chicago. So it's broadly accepted that the central nervous system has to manage multiple degrees of freedom to produce a well-coordinated motion. In this way, it is very one of the recruitment of synergies is a good example of how the central nervous system can take advantages of the modular control. Here we are taking muscle synergies as group of coactivity muscles that can be flexible, recruited to convert movement goals into task-specific biomechanical outputs. The manipulation of cognitive and mechanical constraints can affect performance of walking and also spatial temporal parameters of gait as step speed, step length, or step width. The role of the arms during walking is very important because they have to counterbalance the leg movements. And while more challenging tasks as this in the video or as being walking, the legs can also ensure the stability by shifting the center of mass position. Regarding of cognitive constraints, we already know that older people has a more a less automatic and are more dependent on brain areas responsible for vestibular system, visual system, and also sensorio-motor system, while young persons has a more automatic gait, being dependent only on visual brain areas. However, we don't know if these constraints can also affect the neuromuscular control of gait, and if age can mediate these effects and how. In this way, our aim here is to examine the effects of cognitive tasks and changes in arm position during beam walking in modular control of younger and older adults. We expect that the more challenging conditions as higher cognitive load and restriction in arms will increase complexity with higher number of synergy and lower variability account for by the first synergy. Also increase the coactivity with more significantly active muscles recruited per synergy, and also increase the efficiency with the sum of the activation of these active muscles per synergy. On the other hand, the age, we expect that it will decrease complexity and efficiency of neuromuscular control, but not coactivity that should be greater in older adults. So we had two groups. The younger group was formed by 17 persons with 24 years average, while the older adults group was formed by 14 people with 69 years average. They performed two blocks of beam walking. So the beam has 6 centimeters wide, 2 centimeters high, and 4 meters long. And the first block was performed without the cognitive task, and the second block with the cognitive task. The cognitive task was sequential subtractions by 3, starting in a number between 300 and 900. Within each block, the arms were totally randomized between free arms, as we can see in the video in the left, and cross arms, as we can see in the video in the right. So they performed 40 trials in total, being 10 trials for each condition. We used non-negative matrix factorization to extract the muscle synergies of 13 muscles from the right leg and trunk. And then we analyzed the complexity, the coactivity, and efficiency of the muscle synergies. The complexity was assessed by the numbers of synergies recruited, and also by the variability account for by the first synergy. Coactivity was assessed by the number of significant active muscles per synergy. Here we are going to call as WMUS. And efficiency was assessed by the sum of the activation of these active muscles per synergy. Here we are going to call as W sum. We saw that mechanical restriction, so the restriction in the arms, increased the variability accounted for by the first synergy. But these results we could just see in older group, in the older adults group, suggesting that this group has less complexity while performing the beam walking with crossed arms. The cognitive load increased the WMUS. This is the number of significantly active muscles per synergy, but this result was seen just in the younger adults group, suggesting that this group has more coactivity in the presence of the cognitive task. The age showed less number of synergies for older adults and also higher values for variability accounted for by the first synergy. These two variables together imply in less complexity in older adults group. We also saw higher value of the W sum. This is the sum of the activation of muscles per synergy, implying that older adults has less efficiency in their neuromuscular control. So what can we discuss about it is that the cognitive task increased the coactivation in younger adults, what goes against what we already had in the literature, because the literature always find that increased the coactivation in older adults, especially during higher cognitive load, but we can suggest that our younger group could shift their neural control from feedback to feed forward to be able to guarantee safety during the motor task while they can focus on the cognitive task. Regarding the restriction in arms movement, it decreased the complexity in the older adults, which again goes to go against to the literature, as in literature we have more complexity in more difficult tasks. So if we have cross arms, it would be a more difficult task, and we expected to see more complexity. However, when we have the arms crossed, the central nervous system has to deal with less degrees of freedom and has to produce less biomechanical outputs. So this can explain how why the complexity is lesser in this condition. However, we have to pay attention to the high variability we had in both variables. And this can be one of the reasons that we just saw the differences in just one group and not in both groups in each variable. Regarding age, we have that age decrease the complexity and efficiency of neuromuscular control, and it agrees with the literature and with our previous study. And we suggest that older adults do that to preserve motor performance and safety during being walking. However, the number of synergy decreased in older adults, and in literature we usually see that it remains the same across all lifespan, just changing with pathology or fall history. So it disagrees with the literature. And about the efficiency, we agree with the literature, seeing that it can be degraded in individuals with sensorimotor impairments. This is the cases of older adults. And we always also saw that aging process can affect also factors needed to, for a good neuromuscular efficiency, as use of cortical resources or generation of neuronal activity in primary motor cortex. So in conclusion, we have that cognitive and mechanical constraints affect neuromuscular control in different ways based on age, increasing the coactivity in younger adults and decreasing complexity in older adults. And age per se also affect the modular control strategy, showing less complexity and less efficiency in older adults. And I want to thank you for attention. This is my email for contact. And I also thanks FAPES for the financial support. That's it.
Thank you very much, Andrea. Congratulations, very interesting talk. And we have one question from JJ. In your first slides of the results, did you test for the interaction between group and complexity? He points out that it's not right to say it's significant for one and not for the other. And he suggests to, he or she suggests to need to test for interaction and also put a reference that I can also copy in the chat. And he concludes saying that it's an excellent job for a first international talk. And I agree with that.
Thank you very much for the compliments. And yes, we tested for interaction as well, but we didn't find any interactions. Actually, we run the analysis with the age as covariant covariate, and this was the results we had, but no interaction was found.
Perfect, thank you. And I have a question myself. I didn't get the method that you used to extract synergy. So was it non-negative matrix factorization or something?
Yes.
Something else? Okay.
Non-negative matrix factorization. Yeah.
Okay, that's what I was expecting. Okay, thank you very much,
Thank you
Andrea.
very much.
And so I think we can continue with our second speakers today. She is Lisanna Bakker. She's a PhD student at the University of Groningen, again in the Netherlands. And her work is focused on neural correlates of balance learning in aging. Today she will discuss her first paper. Congratulations for the paper publication, where she investigates the task-specific and neural adaptation after a single balance training session in healthy young adults. So please, Lisanna, the floor is yours.
Thank you, Alberto, for your nice introduction. So my main focus of my PhD is focused on balance and learning to balance. And balance is something that declines with an increasing age. So we see that elderly, of course, fall more often. But luckily, there have been developed balance training programs. These usually take several weeks and consist of several balance training interventions. But they do show that they can improve balance performance, reduce the number of falls, and also reduce the number of injuries in younger adults. However, as I said, these usually take several weeks. But a study by Van Dien et al. Showed that participants were trained for 30 minutes to stand on one leg on an unstable board. And just these 30 minutes was already enough to lead to substantial balance skill acquisition. So maybe we wouldn't need such a long training period, but actually just a short training period, similar to the skill acquisition that we see in manual skill acquisition. You just need a short period to improve balance skills. So while Van Dien showed that it's possible to learn a skill that quick, we are not yet sure if you're also able to retain a skill that is a balance skill that is acquired so rapidly. We also want to know if it is transferred to other balance tasks, and we want to understand the underlying neural mechanisms. So therefore, the aim of this study was to determine the effects of a single balance training session on balance skill acquisition, retention, and transferability. And also we want to examine the neural mechanisms underlying these behavioral adaptations. So we expect that balance training or balance skill is best acquired when you actually train the specific balance skill, but we did want to control using an active control group to see if not just being active already leads to an improvement in balance performance. And we also use a non-active control group. Then we also expect that the improvements will transfer to the non-trained balance tasks, and we expect to see a correlation between the behavioral and the neural adaptations. How did we examine this? We included 36 healthy young adults in the study. The mean age of that was 21 years old, and they were randomly allocated to one of three groups. So on the top here, you see a balance training group. They would stand on an unstable board for 20 minutes. Then the cycling training group, they would be sitting on a stationary cycle, also for a duration of 20 minutes. And the non-active control group would just be seated during the intervention part of the experiment. The measurement, this consisted of 2 different days. So on the first day, there was a baseline test, an intervention, and then a post-test. And then after 7 days of rest, there was a second day of measurements, and the participants came back for a retention test. And during each of these test moments, these measurement moments, we assessed both the balance behavior as the neural excitability. I'll tell you a little bit about how we measure that in a bit. And we also collected during the first day questionnaires, for example, about physical activity and sleep quality. So balance performance was assessed using 3 different tasks. First, we assessed it on the wobble board task. This is the same task that the participants in the balance training group also trained at. So they would stand on this board and they were instructed to keep it as horizontal as they could. And we would extract the time that they were in balance. Next, we also looked at a beam walking task. So during this task, participants had to walk over 2 different beams with different widths. And we assessed how far they were able to walk on these beams before and after the intervention. And the third task was a static balance task. So they would be standing either on a stable surface or on an unstable foam surface. We also had other tasks. So for example, eyes open, eyes closed, and feet in like a normal stance or a narrow stance. So feet next to each other. And we examined the center of pressure velocity because they were standing on top of a force plate. So it allowed us to extract the center of pressure velocity. Then we also looked at the neuroexcitability. So we used 2 different techniques for this. On the left, you see intermuscular coherence. We mostly looked at the intermuscular coherence in the alpha and beta frequency bands because this gives you an idea about the input to the muscle pairs coming from the subcortical and the cortical regions. So we looked at both synergistic muscle pairs, as you can see here, soleus, lateral gastrocnemius, peroneus longus, and also antagonistic muscle pairs. So included the tibialis anterior, the soleus, and also in the bottom, the rectus femoris and the biceps femoris. The next thing we did was we also looked at, oh wait, I want to say one more thing about intramuscular coherence. We collected this data of intramuscular coherence during this static balance task. So while they were standing on different surfaces and with eyes open, eyes closed, etc. Then we examined the transcranial magnetic stimulation. So using this DMS, we looked at the motor evoked potential, the MEP, but we also looked at short and long intracortical inhibition and intracortical facilitation using different interstimulus intervals and different stimulus intensities. So this allows us to, this gives us information about the cortical, but also the subcortical input to the muscles. So what did we actually find? On the left here, you see the performance of a participant from the balance training group at baseline and at post-test. And what you see is a deviation of the board in the medial lateral and in the anterior posterior direction. And this orange circle right here is a target circle that was also displayed for the participants in front of them on a screen. So the participant was supposed to keep the board horizontal. So we would allow a deviation of 5 degrees from the horizontal as being in balance. And as you see, before the participants had a really tough time to keep their board in balance, but after the 20-minute training period, they really improved their performance very much, and they were able to keep it horizontal almost all of the time. And when we look at the group level, you can also see that, because here on the left, we have the baseline versus post-test differences, so the improvement after just 20 minutes. And here you see a balance group, the cycling group, and the control group. This line over here is displaying the mean of the group, and those dots are the individual different scores for every participant. So you see that on average, they improve almost 339% in the balance training group, which was significantly more than the other 2 groups did. And then when we looked at the retention, so this is the baseline versus the retention scores, we see that the balance training group still scores, is significantly improved more than the other 2 groups did. However, we did not find any significant change in the standing balance and in the beam walking task, so we did not find any transfer. And also we did not find any neural excitability, any changes in the neural excitability. So now I will discuss these findings with you. Let's start with the main find of the study was that we found a really big improvement in the task that was trained. It was 339% improvement compared to baseline on average. And this is larger than the session normalized improvements that we see in long-term studies. And in addition, we also see that in the study from Van Dien, we saw that a lot of the improvements that they found in their study already occurred in the first 16-second long trials, which shows, makes us suspect that similar to manual skill acquisition, there it is the rapid initial learning that dominates the balance improvements, also in longer term studies. So we expect that most of the improvement, even in long-term studies, already happen in the beginning, in the first few sessions. Therefore, future studies should examine the time course of these adaptations in more detail. So they shouldn't only examine the improvement after 1 week, for example, or after several weeks, but really look at what's happening, because maybe most of the change is already happening really quickly. And then we didn't find any transfer of, even though we found such a big improvement in the trained task. And this is actually in line with the study from 2015 that shows that there was no transfer even between very similar and challenging balanced tasks. And there's also this paper that looked at all the different intercorrelation between the tasks, between different balance tasks, and it was actually quite low. And when we looked at intercorrelation in our tasks, between our tasks, we actually saw only medium intercorrelation between the tasks. So here there is the distance walked on the balance beam, and here there is the time in balance on the balance board. And you see it's only correlated 0.4. And this implies that there are low mechanistic links between the tasks. So for example, in our task, they are training to counter the rotations of the board, but this is not very useful when you are standing on the floor. So therefore, in future studies, they should include a test task that is similar in spatial temporal structure to the trained task. And then lastly, the lack of neural adaptations. So we found a dissociation between the behavioral adaptation and the neural adaptation. This might be due to the time course. And possible that the behavior adaptation occur at a faster rate than the neural adaptations do. Another possibility is, I think that we assessed our neural adaptations during either standing task or the TMS, we assessed it during sitting and standing. However, we did not assess these adaptations while the participants were standing on the balance board. So, these studies show that they were only able to detect these neural adaptations when they assessed them during a postural perturbation or during a task that was very similar to the trained task. So we think that in order to really capture these neural adaptations, they appear to be really task specific, so this should be considered. And also the time course of the neural adaptations also has to be considered in future studies. So in conclusion, we found that just 20 minutes of balance board or wobble board training led to acquisition and retention of a new balance skill. However, we did not find any transfer to other balance tasks. And also we did not find an effect on neural excitability by examining, looking at the TMS measure and the intermuscular coherence. Therefore, in future studies, they should consider the time course and the task specificity of behavioral and neural adaptations. So if you want to examine the neural correlates of balance learning, it's important that you take these behavioral and neural adaptations, like that you assess them during the trained task and at multiple time points. Thank you all for your attention. Are there any questions?
Thank you very much, Lisanna, for your great talk. Very, very interesting. And while we wait for question from the audience, I have a question myself. So could you please re-explain me how the TMS protocol worked? So where did you stimulate with the TMS, and what did you record?
Yeah, so with the TMS, Let me go to that slide. Yeah, so we stimulate the motor cortex, but we stimulate the area of the tibialis anterior muscle. And so we collect the muscle activity and we stimulate that specific area. And then using different intensities and different interstimulus intervals. So if you just give a single stimulus, you will just get an MEP, a motor evoked potential, which looks a bit like this. But when you use SIKI, so here you can see there are two different pulses after with only really short interval between them, that actually causes inhibition, and you can see that that this peak is actually smaller than it would be if it was just a normal MEP. And that's also what you see here in the leaky, the long intercortical inhibition. So we can see the inhibitory and facilitatory mechanisms of the brain using this technique. Does that answer your question?
Yeah. I didn't get the details. Thank you very much. Oh, we have one question from JJ. Do you think that longer transfer could improve the transfer to other tasks, and that transfer only take place later in training, longer training?
So if longer training can improve the transfer?
The transfer, yes.
Okay. Yeah, so I'm not sure about, I think if you train longer, it will maybe lead to, for example, strengthening of the muscles, which could lead to a transfer on the longer term. But as I said, these tasks are quite different. So I'm not sure if the training of a balance board task could lead to an improvement on the balance beam.
Okay, perfect. Thank you very much, Lisanna, for your question and for answering for the talk and for answering the questions. And I think we should move on to our last speaker today. She is Margot Bootsma. She is a PhD student at the Department of Human Movement Science, again at the University Medical Center in Groningen. And so please, Margot, the floor is yours.
Yeah, thanks for the introduction. So let's see if this works. Yeah. So as we age, we experience a lot of unfavorable deteriorations in a variety of functions, such as the ability to perform simple motor tasks, such as putting your key in the keyhole. And next to this behavioral changes, we also experience some changes on the brain level. And on a structural level, we see the loss of both gray and white matter, causing the brain to shrink. And what you see over here is the average annual loss of gray matter per year in healthy elderly normal subjects. And you can see that this average loss goes up to about 4 to 5% per year in even healthy older adults. And on a functional level, if we ask younger participants to execute a motor task with their right hand, so a unilateral motor task, we mostly see activity in the opposite contralateral hemisphere. However, if we ask older participants to execute the same motor task, we see that this contralateral activity increases, but we also see increasing activity in the opposite ipsilateral hemisphere. So there's a bilateral overactivity in older adults while performing the same tasks as younger adults. And based on all these changes, you might expect that the ability of older adults to learn new motor tasks also deteriorates. And this is what long has been thought. And but that's also been shown by a lot of studies, such as this study showing quite a dramatic drop in the learning rate between the ages of 50 and 60 years. However, there are also now more and more studies who show, such as the study from our own lab, who show that although the motor performance is different between younger and older adults, so they have worse motor performance, the actual rate of learning is the same. So they are able to learn this task as equally as younger adults. And there are even now studies who showed a superior learning rate in older compared to younger adults. So all in all, there is a lot of inconsistency in the literature about the ability of older adults to learn new motor tasks. And that is why with the current study, we aim to look further into these effects of age on skill acquisition and retention, and also dive a bit into the underlying brain mechanisms. And over here, you can see our study design. So we recruited 72 participants, among which 36 younger and 36 older adults. And they visited our lab for 2 days in a row. And on the first day, they practiced a mirror star tracing task, which you can see over here, in which we asked the participants to trace the outline of the star as fast and accurate as possible, while they were only allowed to look at the star in their moving hand through the mirror. And while performing this task, they were instructed that accurate performance is to stay within this width of the wall of the star, which we call the bandwidth. Before, immediately after, and 24 hours after they practiced this task, we measured their motor performance as well as their brain activity, which we did with 64-channel EEG. So you see here the whole setup for one of our older participants. So if we ask both the younger and older participants to do this, this is what a representative star looks like on the very first attempt. So the blue one is the younger adult and the red one is the older adult. And well, you do see some performance differences already here. But then if we let both participants practice for a while, you see that for both the younger and the older adults, the star, well, looks a lot better. And this is actually also what we see reflected in our outcome measures. So we quantify motor performance both in terms of movement time. So that is the time needed to execute one trial of the star, and it's a measure of speed. And we have a measure of accuracy, which is the bandwidth error, and which represents the percentage of trials in the percentage of samples in a trial which participants were outside this width of the wall of the star. So for both outcome measures, lower scores means better performance. And what you can see here is that there was indeed differences in motor performance between older and younger adults. So older adults had worse motor performance, but their rate of learning, so the slope of improvement from the pre to the post test, was actually quite similar for speed, and the rate of learning was even a bit higher in older compared to younger adults for accuracy. Interestingly, then if participants came back 24 hours after they learned this task, we saw that, well, for the younger adults, they maintained the improvement in speed, so they were still faster at 24-hour retention than that they were before learning, but they deteriorated on accuracy. So their accuracy level at 24-hour retention was actually back to their baseline level. And for older adults, we saw the opposite pattern. So they retained part of their improvements in accuracy, but they deteriorated in speed again at 24-hour retention. And prioritization of accuracy over speed in older adults is something that has been reported before in the previous literature, and it's mostly explained as a learned strategy of older adults in order to maintain the precision of this task, and in this case also in order to obtain enough feedback to actually be able to improve on this task. So all in all, what this behavior results show is that older adults are still able to learn new motor tasks, but that the approach to the task depends on age, so that older versus younger adults adopt a different approach. If we next look at our EEG data, what you see over here is a topographical plot of the task-related beta EEG power. And EEG power in the beta band is known to be dominant at rest and gets suppressed as people actually executing a motor task. So therefore, it is thought that lower EEG lower power during task execution is related to higher neural activity. And in this topographical plot, cooler colors represent lower task-related power. So the more blue, the higher the activity in a sense. And what you can see at baseline is that, well, for older adults, the activity pattern was pretty centralized and equally spread across the two hemispheres. We saw a slightly more lateralized activity pattern to the contralateral hemisphere for younger adults. And then as participants practiced the task, we saw a decrease in task-related power. And as you can see here, this was mostly apparent for older adults. And then when participants came back 24 hours later, we saw that actually an increase to baseline levels again. And if we zoom a bit into this result more, and we look at the power in the motor cortex, because, well, that's the area of the brain which is, of course, related to motor performance, but also to motor learning. What you see here is, again, the task-related power. And just a reminder, lower task-related power is indicated of higher neural activity. And this is in the motor cortex, and we see that the decrease in task-related power, which I just mentioned from the pre to the post test, was most apparent in the older adults and also mostly in the contralateral hemisphere. And it's possible that this increased modulation of EEG power in older adults represents a compensatory mechanism to enable these older adults to learn this task to an equal level as the younger adults, despite the changes that are happening in their brains. And this compensatory view, well, we actually find more evidence for this if we look at the correlation between differences in movement time from the post to the retention test and the differences in contralateral beta task-related power from post to retention. So what you see in this graph is this correlation separately for the younger adults left and the older adults right. And on the x-axis, you see the change in movement time from post to retention. And just for visual purposes, I refer to the axis such that right to the 0 denotes an improvement in movement time from the post to the retention time point, and left to the 0 is a deterioration. And on the y-axis, you see this. You see the contralateral beta task-related power, also the change in this power from post to retention, and the positive numbers denote an increase in task-related power, and the negative numbers is a decrease in task-related power. And what we see with this correlation is that we found a correlation which was specific for the older adults. And there was a relation between the deterioration in movement time and the increase in task-related power. So it means that less deterioration of movement time for post to retention was actually related to a smaller increase in task-related power from post to retention. So that means that the older adults who were able to maintain at least part of the changes in task-related power we saw directly after learning, those were also the older adults who were able to maintain part of the improvements in movement time we saw directly after learning. So this suggests that this decrease in task-related power we see after learning in older adults is indeed compensatory to be able to learn this task. So to wrap it up, with this task, we saw that older adults are still able to learn new motor skills, actually to a comparable level as older adults. But we see that during learning, older adults seem to prioritize accuracy in this task, while younger adults seem to prioritize the speed. And we also saw that age affects the neural mechanisms of visual motor learning, and specifically, we see a higher decrease of power for older adults, which is possibly a compensatory mechanism to still improve in this motor task. So with this, I want to thank all my collaborators and co-authors, as well as the students who helped me collect all this data. And as a take-home message, I hope I showed with these results that although we do experience a lot of deterioration as we age, not all is lost. And I hope with the right motor skills and interventions, we can change the picture of aging from this video I showed in the beginning to the video of this old man still dancing and moving happily. So thanks for your attention, and I'm happy to answer all the questions. And also my email is here if you want to discuss anything further.
Thank you very much, Margot. Super nice talk, and very interesting how you did this study collecting both the behavioral and performance outcomes and the EEG data and trying to find a correlation between the two. So while I encourage the attendees to ask questions, I have one myself. So at the beginning, you showed that there are those conflicting results about contralateral or ipsilateral activation in young and older adults. Is that something that has been found on EEG studies, like the one that you did, or also in some fMRI studies or neuroimaging studies?
Yeah, this bilateral overactivation with aging is actually quite robust finding found with a lot of different techniques. So it's also been found a lot with fMRI studies next to the EEG studies.
Okay, thank you very much. Well, for now we don't have any questions from the attendees, but I encourage everyone to reach out to the speakers if they have additional questions, and also all these talks are recorded and will be available also on YouTube for future watching. So you can also receive mails and asking for details or for discussion even in the coming days or weeks. And okay, so I would like to thank you all the attendees and Margot, Andrea, and Lisanna for their three wonderful talks. And thank you very much for being here at Neuromatch 3.0. And thank you, Ali, our super backend host here in this room. And I will just say to enjoy this last day of the Neuromatch conference, and happy Halloween also. Thank you. Bye, everyone.
Okay, I think it's 10 o'clock or whatever time it is at your current location right now. So we'll go ahead and start the session. I'm Robert Rosenbaum. This is session 3009. I'll be your MC today. Just a reminder really quickly, you can ask questions in the Q&A and vote on questions that you want to hear answers to. And you can also use the chat feature So our first speaker is Percy Mystery from Vinod Menon's lab at Stanford University, and I'll let him take it from there.
Thanks, Robert. So I'm just going to share my presentation. I hope you all can see it. So I'm going to talk today about combining behavioral and neural data using computational modeling approaches. And the sort of background there is that there are, of course, multiple approaches that one can take to combining behavioral and neural data. I'm going to showcase 3 different approaches that we have used and the context in which they may or may not be useful. This is, of course, not an exhaustive set. And the key point I want to make is that using conventional sort of canonical computational and cognitive models might not be adequate. I make the case that we need to augment these models with things like process dissociation, adding systemic temporal variability to the computational models, or adding sequential effects, inferring latent states or cognitive strategies over and above what might be conventional models like drift diffusion model of evidence accumulation and so on, to get the richest set of inferences that one can make from joint behavioral and neural data. And so I'll just dive into the first approach. And this is typically a good approach to use if you have a very good sense of what a strong theoretical basis for what is the structural cognitive model to use. For example, it's a 2 alternative first choice task, and you have a drift diffusion model that can explain behavior very well. But there's greater uncertainty about how the neural data interacts with this task and this behavior. What we do is we augment a statistical model such that the neural data forms inputs into the parameters of the cognitive model. And all of this inference is done within a joint framework. So it's not a two-stage framework. And then we can, what is paramount in the inferences is that the cognitive process parameters should be able to use the structural cognitive model like a drift diffusion model to explain behavior. But if there is any variance in the neural data that helps explain the variability in the cognitive process parameters, this gets captured in the statistical part of the model. And that helps us to sort of detect functional associations between specific cognitive sub-processes and the neural data. So I'll just illustrate that with an example to make it clear. So this is a social perceptual task where children and adolescents had to listen to their own mother's voice, unfamiliar voices, and environmental sounds. The voices basically uttered very short, nonsensical words whilst their brains were being scanned. And after that, they also had to do voice identification tasks to discriminate between their mother's voice and unfamiliar female voices. And the idea was to look at what underlying brain network systems might modulate differences between recognition of mother's voice and unfamiliar voice in a period when their social skills are developing strongly. And so they might be more inclined to shift their attention and acuity towards unfamiliar voices in the hope of sort of expanding their social behavioral skill sets. And so what we did is we applied this kind of a drift diffusion model that explained their behavior in the voice identification task, because it was a 2 alternative first choice task, which is the graphical model on the right. But what the inputs to the parameters over there was this diagram A on the left, which extracted neural metrics on the brain connectivity. So we looked at the voice selective circuits, the salience, the default mode network, and the reward circuits in the brain. The connectivity is within these sort of modules, as well as the connectivity between these modules. And the idea was to see at a group level, how well could that variability in this connectivity explain the variability in the drift diffusion model parameters. And what we saw is that the key difference between when children and adolescents reacted to unfamiliar voices versus mother's voice was the involvement of the whole reward circuit. So we saw that the connectivity of the reward circuit with both the voice sensitive and DMN networks was much, much more stronger, which is this red box on the left and these orange lines, when the acuity to unfamiliar voices was higher, but the reward circuit was completely missing from the action when the acuity to mother's voices were higher. So that sort of implicates the reward circuit in this sort of social recognition of unfamiliar voices during a very, very key developmental stage in children and adolescents. So, and the key part was when we used, without this joint modeling approach, traditional approaches using just simple raw behavioral data and looking at task activation did not identify this reward circuit in such a strong manner. So that was a relatively simpler example. Then we go on to the second approach where we use neural features to constrain the cognitive process parameters. And the idea here is that we might have a sort of strong theoretical basis for the relationship between behavioral and neural data. So for example, the fact that hippocampal regions are involved in memory. What might be less clear is for a particular task, what cognitive sub-processes underline the behavior and how do they relate to the neural data. For example, we might have competing cognitive models or sub-processes, and the behavioral model is not able to discriminate between which has better explanatory power, or the cognitive process might be quite complex. It might have multiple strategies or states and have a lot of moving parts, and it's not clear which functional sub-process is related to what neural aspect. But what's different in this approach is that the direction of this arrow from the cognitive parameters to the neural data. So instead of using neural data as input, here we are constraining inferences about the parameters by using the neural data. And in a statistical sense, what this does is the influence of the neural data in constraining the cognitive parameters is much more stronger in this approach. And this makes sense because we've come from the premise that we have a much more stronger theoretical basis for the association between behavioral and neural data. And to showcase an example, we used a task where young children had to do simple arithmetic addition subtraction problems. And we looked at children who had mathematical learning difficulties, and we gave them 8 weeks of one-to-one math tutoring. And the idea was to see whether we could identify plasticity in brain networks in response to the tutoring. And we did find changes in the hippocampal and anterior insular regions, and we also found changes in accuracy with training. But what this did not identify is which cognitive aspects did the training actually influence such that it resulted in improved accuracy. For example, we know from prior literature that children might use a mix of different strategies, memory versus counting versus decomposition and so on. And that's a big area of research in numerical cognition. But we aren't able to isolate whether the change in accuracy was because children were using a different mix of strategies, whether they were applying strategies more effectively, or whether they were applying them in a more optimal sense, identifying which items were better suited to which strategies. And when we applied a simple canonical drift diffusion model to behavior and saw whether the parameters of that model could predict neural data, we got a weak signature there, but you can see that the confidence intervals on the prediction are very, very large. So it's not very meaningful. Plus that this model could not identify unique measurements or aspects of the cognitive process that respond to tutoring. So we developed a more nuanced model where we built in attentional control, different strategies, and I won't get into the dynamics of the model, but the idea is that there were a lot of moving parts in this model. Now there's performance monitoring and executive function and different strategies, and we allowed for a shift between strategies within a trial. And so what this model essentially does is gives a much more refined measurement system. So we have multi-dimensional measurement for each individual child in terms of their performance on that arithmetic task. And that's represented in this sort of graphical model on the left. And when we use the parameters of this nuanced model to predict predict the sort of changes in the brain plasticity, you can see you get a much stronger result. That's the graph on the right. And that's the sort of comparison of the two graphs. And moreover, using this model, we could also identify what particular aspect of the process. So we found that it's not really any mix of strategies that children were using or more optimal discrimination between strategies, but it's more that the efficiency of their memory process execution was significantly improved. And that was associated strongly with changes in the hippocampal modularity that we saw. So that's sort of the kind of improving the functional resolution of the changes in the brain plasticity that we see with tutoring that we could identify using this model. And for the third and last sort of approach, we had a joint neurocognitive model. So we eliminate the statistical part of the model because now we have strong structural models for both behavioral and neural data. And we applied this to a response inhibition paradigm. So this is a stop signal task where people are given a cue as to click left or right. But if they get a stop signal before they've pressed, then they have to inhibit their actions. And the stop signal comes at a certain delay after the cue. And a traditional brain analysis might look like something like this, where you have a hidden Markov model for state switching. And the idea is that this is a very attention-dependent task. So it's used to assess inhibition or impulsivity in children with ADHD, for example. And by looking at the changes in the covariance pattern of the brain regions across time, you can sort of infer different attentional states that children might have. But the challenges with that approach is it's difficult to ascribe a meaning to what a specific state really means, apart from the fact that you can identify that something is changing in the brain. And it's very difficult to compare states between individuals. So what we did is, and again, I won't go into the details of the mathematics of this model, but what we did is build a cognitive model that has meaningful states, a proactive control state, a reactive control state, and an attentional lapse state, such that the parameters of the model dependent on which state they were in, and the state that they were in dependent on their whole process of belief updating sequentially over trials. And so now we have meaningful attentional states. And just to give you an idea of the individual differences we can capture with this kind of a model is one individual who's tracking the delays in their stop signal time very, very acutely, whereas someone else who's sort of tracking in the right direction, but in a very, very loose manner. So the first individual has much more stronger proactive control than the second. And now we allow inferences about states jointly from both the behavioral model, the one on the right, and the brain covariance data on the left. So the neural data provides a constraint on the behavioral switching, so it reduces the influence of noisy behavior, for example. And the behavioral data provides contextual meaning to neural switching. So the states now have psychological sort of meaning and they're just not statistical states. And when we looked at kids across the spectrum with a different range of ADHD scores, and we looked at looked at the difference in the covariance matrix for low and high attentional states, and then the difference between low and high, children who scored low and high on various ADHD metrics. And what we saw is that the nuanced process dissociated model, which is the one on the right, you could actually discriminate very strongly between these groups, low and high ADHD score groups, compared to the traditional canonical approach where you see that everything is mostly flat. And so in conclusion, there are sort of multiple approaches we can use to jointly look at behavioral and neural data within a computational modeling context. Which approach is best depends on the specific research question. But an important point I wanted to make is that once we start augmenting the underlying models with a lot of context-specific features, temporal variability, or process differentiation, or latent cognitive states and strategies, the kind of inferences that we can make are much more richer, and we are able to discriminate between things that traditional models don't. So thank you, and I'll take any questions.
Could I ask something? Can you hear me?
Sure.
Maybe this is a very naive question. Could you use these methods to train, let's say, a recurrent neural network and constrain the activity of your neurons, so the network would still produce the behavior you want, but with a certain activity in your neurons?
So you're saying replace the cognitive models with neural networks?
Yeah.
So that's certainly plausible, and it's something that I've given very, very loose thought about, but I haven't even begun to look at how implementation might work in that sort of case. So the answer is yes. If it's definitely possible. I don't know if someone has specifically tried something like that. It's possible, but I haven't come across work where, I mean, of course, there are people who've looked at applying these neural networks and looking at the representations in these neural networks and matching them to neural data. But whether the neural networks can also produce the behavioral aspects of the data simultaneously, along with the neural aspects, is an interesting question. And it's plausible. I have not actively worked on anything like that, and there might be work out there, but I'm not actively aware of that.
Okay, thanks.
Great, thank you. So the only other question on the Q&A was about a preprint. So if you have a preprint for this work, maybe you could type it into chat during the next. Sorry, go ahead.
Thanks. So thanks, Jules. This is multiple projects. Each of these was a separate project. One of this is under review. The other couple of them, we are finalizing the manuscripts. So no, we don't actually have a preprint out right now, but hopefully very soon.
Okay, great. Thank you.
Thanks.
Okay, so perfect timing. The next speaker is Valentin Wired from INSERM and ENSP in Paris, and I'll give the stage to him.
Thanks, Robert. Hi, everyone. I'm happy to tell you about this new work in my group, studying how random noise in cognitive computations may actually promote cognitive resilience to adverse and uncertain conditions in humans and recurrent neural networks. So the work starts from the basic observations that many of the decisions that matter in life are uncertain. Whether you're a hunter-gatherer looking at footprints in the sand, looking for a prey to hunt, whether you're a stock trader about to buy volatile stocks, or a British citizen deciding whether to remain or leave the EU. And errors are constitutive features of human decisions made under uncertainty, often with serious consequences, getting charged and injured by an aggressive animal, crashing the economy, or ending up with a no-deal Brexit, right? While these three decisions are clearly very different, I'm going to argue that they rely on the same basic cognitive process, which I will call cognitive inference, which consists in integrating several pieces of uncertain or conflicting evidence to form accurate beliefs about one's environment. In the lab, the tasks we use to study cognitive inference are often deceivingly simple. In this canonical example, subjects are asked to identify the jar from which the marbles presented at the center of the screen are drawn. As you can see, because the jars contains marbles of the same colors but in different proportions, the task is essentially uncertain. In this case, the marbles are more likely to be drawn from jar A. Despite the apparent simplicity of this task, humans make a substantial fraction of suboptimal errors, as can be seen in red on this graph. As you can see, like the optimal Bayesian process, which is represented by the dots on this graph, subjects make more accurate decisions when they have more marbles to form a decision, but they remain essentially always below the optimal performance in this task. Over the last few years, we've decomposed these suboptimal errors into three main sources. Perceptual errors during sensory processing on the left, cognitive errors during inference in the middle, and choice errors due to exploratory decision policies. With Jan Drugovic a few years ago, in a perceptual classification task very similar to the marbles task that you've seen before, we found that a dominant fraction of human errors is due to cognitive errors in inference rather than to perceptual errors. And more recently, with Charles Finling and Vasilisa Skvortsova, two postdocs in my group, we have shown that reward-guided decisions in bandit tasks are subject to the same source of cognitive errors, errors which can explain a large fraction of decisions that are otherwise labeled as exploratory. In both studies, we asked whether these cognitive errors are due to biased and wrong inference, or rather to variable and imprecise inference. And by studying jointly the accuracy of the decisions and their internal consistency, we found that about 2/3 of cognitive errors are due to imprecise computations. In other words, what I will call computation noise in the rest of this talk. So this was true for both reward-guided decisions here and for perceptual decisions here, right? So our interim conclusion that computation noise seems to drive a large fraction of decision variability under uncertainty triggers at least two additional questions. The first one is the neural substrate of this noise. And the second is its possible cognitive function, which is why what I will talk about in the rest of the talk. So if computation noise can result in sizable decision errors and Boris Johnson as a prime minister, the question is, why is this noise still there, right? Why didn't evolution do its job and suppress it? So there are at least two possible non-exclusive answers to that question. The first one is that maybe the cost of noise suppression is too large, right? So noise appears in this case as a cognitive liability in the equation, right? Another possibility is that maybe computation noise may provide emergent cognitive benefits, which is what we've investigated. So we started this particular study from the well-known observation that current AI systems, despite their limits, so in this case, it's probably a deep convolutional network trained to perform image recognition, these AI systems show limited cognitive resilience to adverse conditions. In this case, this colored pixel noise added to the image in the center, right? I'm assuming that none of you has classified the image on the right as a gibbon, right? But rather as a panda. The current approach in the field consists in engineering human-like cognition in AI systems to make them more resilient to these kind of adverse situations. But one problem that we saw with Charles, my postdoc, with this is that this kind of great architect approach is likely to proceed by adding ad hoc sophistications to the AI systems to engineer new cognitive abilities. So Charles, postdoc in my group, had a different idea that we pursued. He proposed to add computation noise in recurrent neural networks trained to perform cognitive tasks to test whether and how noisy computations may surprisingly and counterintuitively increase the resilience of these simple AI systems to adverse conditions on which they were never trained, right? So we started from the very most basic, simple Hellman RNN network, okay, in which we added computation noise, meaning that the computations performed by the recurrent units in the networks were not exact anymore, but stochastic, and were then sampled from a normal distribution around their expected computations, right? And then we trained networks on canonical cognitive tasks. In this first example, we trained the network to associate symbols, in this example, the star, but there were 8 symbols that could be presented to the network with possible actions. In this case, the network has to choose between the red and the green target. And for each of the symbols, there is an action that will be more likely to be rewarded if it is chosen by the agent, right? So you see that the star, for example, you should choose the red target to be rewarded 70% of the time, okay? So we trained the network with these 8 symbols to learn their probabilistic association with reward at the end of the trial. And after a few thousand training steps, both exact and noisy recurrent neural networks converge to the optimal decision policy, which is to use a greedy policy to choose for each symbol the most likely action that is paired with reward, right? But then, of course, we didn't compare the networks in this very simple context, but then we asked them to perform a variant of this original task, where now the network has to decide based not only on one symbol in isolation, but on a sequence of symbols presented in succession, right? In this example, you see that then the network was faced with a sequence of these same symbols that he has been trained with, and each symbol is still associated with the same probabilistic association with reward as in the first training stage. But now the network, basically, to perform optimally, would have to perform cognitive inference, which is to integrate the probabilistic relationship associated with each symbol to form a posterior belief about the most likely rewarded target. Okay, and the idea was to measure cognitive resilience, was to train the networks on this original task A with isolated symbols, and then test the ability of these networks to solve this more complex task, which is more adverse because the networks in a single sequence will face different symbols that are likely to be associated with different rewarded targets. And what we found is that unlike the exact RNNs, who were not able to improve their performance with increasing number of symbols that were presented to them, the noisy RNNs were capable, just like human subjects, to increase their accuracy of their decisions as a function of the number of symbols that we presented them in a single sequence, right? Why is it the case? Is that the exact RNN converged on a very strong strategy, a very strong primacy strategy, where they decided on the basis of the first symbol they were presented with. By contrast, so here, this is the result of a logistic regression, where I plot you the contribution of each symbol presented in a sequence on the x-axis as a function of their position, okay, from the first to the last symbol. And you can see that by contrast, the noisy RNNs were capable of integrating the different symbols presented in the sequence, even though they were never trained with any such sequence, right? Human subjects that were trained also in task A, so with isolated symbols, were able without any reward to perform task A prime, exactly like the noisy RNNs, right? And like humans, which were able to combine the different symbols that they were presented with as a function of their reliability, the noisy RNNs were also able to show this hallmark of Bayesian inference, which is to combine the different pieces of evidence as a function of their respective reliabilities, something that, of course, the exact RNNs were not able to do without explicit training on that specific task. And more surprisingly, we found that the noise in the noisy RNNs was instrumental to perform the task, because the noise was representing the reliability of the separate symbols. So on this x-axis, I'm just sorting the symbols as the function of their predicted rewarded target. And the most unreliable symbols in the center were associated with more variable activity patterns in the networks than the more reliable symbols. we didn't restrict our analysis to these weather prediction tasks, but we moved also to bandit tasks to find the exact same conclusion. So we trained these exact and noisy RNNs on very simple two-armed bandits with fixed reward schedules. In this case, in this particular game, action 2 is paired with a probability of reward of 0.95, okay? And so if you train the networks to learn to perform in these tasks, where in each new game, the best symbol may change, you see that both types of symbols are capable of reaching almost optimal performance after a few trials. But now what we did is that we tested the same RNNs in a situation where in the middle of a single game, we would flip the reward schedule. So now the good shape would become the bad shape. And so the RNN would have to change its decision. And we found that only noisy RNNs, exactly like in the previous example, were able to adapt to these reversals without any training. This happened because in the noisy RNNs, as soon as a reversal happened and the network faced bad or negative outcomes, noise increased in the network and destabilized the strong attractor states in these networks to allow the networks to reverse its behavior after a few more trials. We then tested something important, which is that all of these findings about noisy networks say that moderate amounts of computation noise will increase the performance of these networks in these adverse tasks, right? So what we did is that we tested a large group of subjects in the same task and look whether across the different subjects that feature different amounts of computation noise, subjects with moderate amount of computation noise will also perform better than subjects with low amounts of noise or subjects with more noise, right? And we found exactly that inverted U-shaped relationship in the weather prediction task, but we found the exact same relationship in this very different two-armed bandit task, okay? So the conclusion of this work is that it seems that moderate level of computation noise promote cognitive resilience to adverse conditions without any training nor ad hoc sophistication in these types of networks. So I'm just going to end by thanking my funding sources, and of course by thanking Charles for his work. And thank you all for listening.
Thanks. Yeah, really nice talk. We have a few questions. Jules has the most upvoted question. Jules, do you want to ask it yourself or do you want me to read it? If you'd like it to ask it yourself, you can. Okay, I'll read it. I'm not sure about if Jules can even unmute their microphone. So Jules asked, can you or how would you discriminate computational noise from the mismatch between the actual neural processes and the mathematical function used to model it?
Yep. So at least if we start from the human data, we try to do exactly that. So of course, that's exactly the question that I asked about whether this variability or these cognitive errors we see are due to wrong inference, which is exactly what you described, which is that we have the wrong model of what people use as a model to solve the task, or whether it is truly due to imprecise computation, so to what we call noise, right? And what we did to do that, to study that, is that of course it's impossible to study all possible kinds of biases and all possible ways in which people can be wrong. So what we used is that we studied jointly the accuracy of decisions and their consistency. And we did that in these two papers to try to get to an idea of what is the bias variance trade-off of these errors. And in both cases, we found that variance is dominant over the bias term. So we do think that a large fraction of what we see is not due to the fact that people use systematically wrong computations, but truly to noisy computations. And in the RNNs, the noise we added to the network is clearly random. There is no structure to this noise. We just added, we sampled the result of computations from normal distributions. So in this case, we created noise ourselves, so we know it is noise. So we know that the benefits we show are due to noise and not to a bias term.
Great, thank you. I think we only have like 30 seconds left, so I don't think that's enough for another question, but the questions will be, you can answer them via text if you want.
Oh, yes, okay.
The Q&A session.
Thanks.
Okay, great. So moving on then, the next speaker is Manuel Molano Mazon. I'm having trouble reading my own handwriting, sorry. From EDBUPS in Barcelona, and I'll hand it over. You're muted.
Okay. Yeah. Can you hear me now?
Good. Yeah.
I don't know what happened. Thanks. Yeah, thank you, Robert. Thanks everyone for being there. So today I will be presenting the work we've been doing. We are pre-training recurrent neural networks on ecologically relevant tasks. I will explain in a second what this means. In order to get networks that behave more like rats do in a two alternative first choice task. So we are also interested in these suboptimal behaviors that we found in rats when they do this simple task. The task is, as I say, is fairly simple. Rats are listening to a stimulus, and they have to decide whether to go to the left or to the right. And this stimulus is noisy, so they need to make an effort. But still, they have another source of information because we have organized trials in a way that trials are correlated. We have, let me just take the pointer. We have repetitive blocks in which the probability of repeating the previous site is larger than 0.5, as you can see in this example. So you can see the type of sequences we obtain. And we have alternating blocks in which the opposite is true. The alternations are more likely. So the question here is how rats are able to combine the current stimulus information and the previous trial history, or their expectations about what is the most likely reward or side. So as Hermosa Mendizabal et al., this is a paper from this year, show rats were able or are able to use this trial history. They bias their decisions as a function of the context. So you can see here the psychometric curve in which we plot the probability of repeating as a function of the evidence for that repetition. And in the context, in the repeating context, they are repeating more often than in the alternating context. This shift here is a way we have to quantify the bias, how much rats are using this trial history. So, so rats are able to use the trial history, but they are only using it after a correct trial. After an error trial, they don't use it. They choose to ignore this trial history, which we believe is a very interesting strategy. We call it reset strategy. And as in the previous talk, we believe it's interesting because it's not optimal. The optimal strategy in this task would be after an error to switch the bias. So this is a cartoon, but this is just to show you how an optimal agent should behave. If you are repeating in a repeating block, you make a mistake, you should alternate and keep repeating on the other side. But rats don't do that. And we have found that this is pretty robust. We tested this strategy in several variants of the same task. So we build this reset index that is based on a generalized linear model that I will be happy to discuss, but it is fairly simple. The idea is that we do 1 minus the contribution of the trial history after an error divided by the contribution of the trial history after a correct. So you may imagine that in this situation, this index will be high because this contribution, the numerator will be slow, will be small. And this is what we found. We have different variants of the same task. We have different repeating probabilities. We have tasks in which the duration of the blocks is shorter. So this is what we call high volatility experiment. And we even tested the rats without a stimulus. They in a task that they can only use the trial history, and still we obtain very high levels of reset. So rats use this reset strategy. I'm just showing here an optimal agent that does the switch strategy, and you can see the reset index that we would obtain for that kind of agent. So this is what we have so far. We trained rats for around 20 to 30,000 trials, and we obtained, so these rats find a strategy that is fairly good. So they are shifting their bias after a correct trial, depending on the context. But after an error, they are not able to fully leverage the statistics of the trial-to-trial correlations. So the main question is the question we made is how networks would do this task. Would they develop this reset bias? We train LSTM networks with reinforcement learning, And here I'm showing you the performance of these networks across training above that of a perfect integrator. A perfect integrator is an agent that only uses the stimulus. And you can see that most of the networks find or reach this zero value in which they are only using the stimulus. And this is the type of psychometric curves that we obtained. There is no shift whatsoever. When at some point these networks are able to find a better strategy that uses the trial history, and you can see that their performance increases. And at the end of the training, you can see the shift after correct trials. This is dashed lines, but there is also a shift after error trials. So this is the switch strategy. You can see that colors are inverted after errors. So basically we have that networks are trained for much longer, and they are able to do the task, and they are able to do the task much better than rats do. And reaching this point, we wonder what is happening. We train rats for even longer, for even 80,000 trials, but they never show an indication that they were able to find a better strategy. So our hypothesis then was that maybe the brain of the rat is not able to fully leverage these statistics in the two alternative first choice tasks, because it has been shaped by evolution, by development, to behave in tasks that are in environments that are more complex, and it has some priors and some inductive biases that don't allow the rat to fully leverage the statistics on the two alternative first choice tasks. So what we did was to pre-train our networks in what we call the N alternative, well, it's called N alternative first choice task. So we increase the number of choices, but we maintain this feature in which some transitions are more likely than others. And the question is, so we pre-train, then we test the pre-trained networks on the task, and the question is whether we would recover the reset. Of course, there are many dimensions in which we could increase the complexity of the task. Why did we choose to increase the number of choices? I will explain briefly with this slide. The main idea is that we wanted to break the symmetry between correct trials and error trials in terms of the information they provide. In a two alternative first choice task, when you're doing a sequence and you do a mistake, you are able to infer directly that if it was not right, it was left, and then you can apply your trial history expectations. However, when you increase the number of choices, this is no longer possible. Imagine you have this type of sequence that is much more likely. You do 1, 2, 3, you do 4, but it's an incorrect, and you cannot know what was the correct choice. So you are not able to apply your trial history, and so you only can look at the stimulus to make a decision. And this is what we believe the brain of the rat is designed to do. So what we did was to train our networks on 3 different contexts: the repeating context, clockwise and anti-clockwise. You see the type of sequences that are more likely. Networks are able to learn the task with a performance above of a perfect integrator. These traces are the average for networks trained with different number of choices. So this will be for the higher number of choices that we have tested. And you can see that all of them go above 0, which means that they are using the trial history. And here I'm showing an example of one network that has been trained with maximum number of choices of 16. And you can see that after correct, sorry, I forgot to add the legend. After correct, the dashed lines, we have a bias. After error, the bias is mainly negligible. And when we plot this reset index that I was showing for the experiments as a function of the number of choices, we see that the reset index increases up to levels that similar to the ones we obtain in the experiments. So this is my last slide. The main idea is that we believe the brain of the rat has been shaped by evolution to behave in complex scenarios in which the number of choices is higher than 2, and thus it has some bias, some priors or inductive biases that don't allow the rat to fully leverage the statistics of the two alternative first choice tasks. And we show that by pre-training the network on a more ecologically relevant task, we are able to have networks that are behaving more like rats do in the task of interest. So with this, I would like to finish by thanking you all and thanking my collaborators. I will be happy to answer questions.
Sorry, forgot to unmute. Thank you very much for the talk. Very nice. There aren't currently any questions in the Q&A. If you'd like to ask a question.
Yes, I have one.
Yeah, absolutely.
Yes, so great talk. I was wondering, whether there was another possible difference between the RNNs and the rats, which is that, there was this recent preprint by Ashwood and Pillow on bioRxiv saying that these animals, these rodents, they have discrete strategies that they alternate between during this kind of task, these kind of idiosyncratic things where for a few trials, they will always respond right and things like that. So could it also be another difference that you could explore then?
Yes. So I would say that this is totally possible, and it's not is compatible with our proposal. Our proposal would be that these two strategies these two discrete strategies, this Markov process, arise because evolution has shaped the brain of the rat to behave differently after an error and after a correct, because normally in real life errors are non-informative as with respect to the structure of the environment. But yeah, of course, this would be another way of modeling this behavior. Yeah, thanks.
Thanks.
I have a question. Have you looked at what an optimal strategy might look like if you didn't know the number of contexts or whatever, if there was some distribution on the number of contexts? It could be that something that the rat's doing might be actually closer to optimal in that regard.
Yeah, so this is this is an important point, because it is true that one difficulty we have is that it's difficult to train networks when you change continuously the number of the context, because they don't realize they have to pay attention to the context. So we train with 3 different contexts, and they learn that they can identify the context by looking at the repetitions and alternations. In my opinion, rats it is difficult for me to imagine that the brain of the rat can adapt to thousands of different contexts. I guess they can adapt to transition probabilities in one room or in one cage, and to other transition probabilities in another cage or in different experiments, but to thousands of contexts, it seems difficult. So I think this is actually maybe closer to, but it is of course difficult to infer how many contexts can rats adapt to and how many choices they are designed to work with. So this is something that we will explore in the future. Thank you.
Cool, thanks. I guess one more question, since we have time anyway. Does this we don't have that much time, but are there any widely used sort of behavioral protocols that experimental protocols that you think this sort of might call into question? I don't know that much about the literature on which behavioral protocols are used, but
you mean that whether these results are putting into question
I guess the interpretation of rodents' behavior and other protocols that might have 1 or 2 or more different sort of states or contexts?
Yeah, so our suggestion, our main suggestion is that, and we are not the first ones is that networks should be pre-trained before testing them on so it's more from that point of view. It is true that maybe one should be careful when you train your animals or subjects in a 2 alternative first choice task. Maybe you get behaviors that are not optimal, and well, as Valentin show, it could be because they are designed to do more complex things, and this is what we have seen as well. So maybe deciding that this is suboptimal because yeah, because subjects are not very intelligent, it's not the best solution.
Yeah. Okay. Yeah, makes sense. Okay. So thank you very much to all of the speakers. Really nice talks all together, and we're out of time now, but hope to see you guys in some of the other rooms soon. Thanks.
Thanks, Robert. Bye-bye.
Thank you.
Bye.
To the task, like, are those summary statistics still coming along for the ride just because you encoded the scene?
So I did like 2 years ago, I did an experiment where I looked at the this shape encoding by using two different features, so orientation and color at the same time, meaning that you have lines that have a particular orientation, and then you find the target either based on color only or on orientation only. So meaning that the question for us was whether you still learn the distribution of the irrelevant feature that's not defining the target, but it's kind of there. And you do lose the shape encoding. You still get some information about the average color or the average orientation, so that kind of comes along just by being presented to it. But we do see that this nice resembling of the shape, so these RT functions that really look like the distribution. So I don't know whether I can show this again. Like here, so this resembling of the shape is becoming relatively messy. So it seems like you're losing maybe the shape information, but you're still aware that the distractors were green, although you were searching for the target based on being tilted to the left or something. So it's kind of reducing it. So the fact that the target and distractors are really, the feature is important to find it, kind of helps the encoding process of the shape of the shape one.
Awesome.
But mean and variance seem to come along more in task, like when it's task irrelevant.
Right. Very cool. Well, we're almost at time where we will be involuntarily sucked out of this Zoom room. And I just wanted to thank our panelists because you guys did awesome and your research is so super cool. And I encourage all of our audience members to find these people on Twitter and look at their work and live good lives. I don't know.
Thank you.
And also we are streaming with Zoom as well as YouTube. The participants number you see here, that might be not the case. In YouTube, we might have more participants. Maria, I lost your video. I lost Maria now. Okay, now we are waiting for our speakers to join us. So how can I pronounce your name?
Sudhakar.
Sudhakar.
Yeah, that's fine.
Okay, thank you.
Okay. We are online. Okay, it is nice to see you, Maria. Welcome back. Don't worry about the timing, because currently although the time started and we have to finish at 9:45. It is, I mean, if the first speaker doesn't come, the time is going for his slot. It's not going to affect you. For our attendees, we are currently waiting for our first speaker. He's having some problems to connect. As soon as he connects, we will start. But if he doesn't connect, we will start in 10 minutes with Sudhakar's talk. Okay, our first speaker is with us now.
Can you hear me?
Hi, Arami.
Yes. Hi.
Finally, you made it. You have very little time. That's why maybe you can start. I mean, would you like to share your screen?
Yeah, I share. It's okay?
Yes, yes, please share your screen. There is this green button at the bottom, at the down.
Yeah.
Click it and share your screen and start your talk whenever you are ready, because you have very limited time now.
Yeah, I know. Share. I shared. So it's okay?
No, I don't see it.
New share. I shared, but doesn't work.
Probably you choose the wrong window. You should select the presentation or whatever you are going to use to present your data. Yeah, now it is
my
here.
Presentation. Yeah.
Okay.
Okay.
All right. Stage is yours.
Okay.
Please speak a little bit loudly.
Loudly, yeah.
Yep.
Dynamic brain and neuroplasticity. Involvement of homo and heterosynaptic plasticity. We know that plasticity refers to the ability of organisms or cells to adjust their phenotype in response to alterations in their environment. In neuroplasticity or brain plasticity, the brain synapses are modified by environmental thinking, emotion, behavioral, and neural alterations throughout the brain development. Two kinds of plasticity, homosynaptic plasticity and heterosynaptic plasticity are induced in synapses. Homosynaptic plasticity is induced in synapses that were directly activated in a neuron during the development that needs the presynaptic activation. It is input specific or associative, has the associativity because it associates the presynaptic and postsynaptic neuron activity. On the other hand, heterosynaptic plasticity can be produced at synapses that were not active during the induction. Any synapse at the cell can be subject to this kind of plasticity. But totally, both of them are required for normal action and balance the synapses. Let me to explain you one of our studies on layer 6 of the visual cortex and one type of a specified type of the cells, corticogenic plate neurons, CG cells. CG cells can receive two important input from LGN, lateral geniculate nucleus, and also from upper layer, especially layer 2 and 3. This layer can affect both of them, thalamocortical and corticothalamic lobes, and which may get the information that ultimately ascends to the higher cortical layers. In this study, we use we get retrograde labeling in the CG cells. For this aim, we use alexaflor and we inject, this is LGN of the thalamus, and this retrograde layer 6, you see. Here is visual cortex, layer 1, 2, 3, 4, 5, and finally 6. Under fluorescence microscope, you can see the cells. These are CG cells that retrogradely are red, and these green ones are GABAergic neurons. So we can completely recognize them. I mean, CG cells and GABAergic cells. In this study, we used, as I told, we had two input from LGN that can reach here from white matter, and from upper layer, especially layer 2, 3. If we use TBS to upper layer, so we can induce homo-LTP in this synapse. On the other hand, in other synapse, hetero-LTP will be induced. Then we search about their mechanisms. This homo-LTP from upper layer was mGluR 5 dependence and was postsynaptic. It was due to postsynaptic mechanism. And this hetero-LTP was presynaptic and dependent on endocannabinoids from induction of homo-LTP in other cells. On the other hand, if we use TBS to white matter, we could induce LTP in this synapse that was NMDA receptor dependent, and in other pathway in the upper layer, we could induce hetero-LTP. So this first one.
Arami, you are going over a little bit of your time. Could you please, Arami, could you please, wrap it up?
Yes. This is a top down and this is bottom up. So in this study, we could compare these two flow, bottom up and top down flow, and so we can explain the gain control function of CG cells in layer 6. That is regulated by reciprocal line-term plasticity of synapses converging from the upward and downward pathways. Thank you.
Thank you. And please stop your sharing now. In the same down panel, you will see now stop sharing.
Stop share. Yeah, stop share.
Okay, thank you. Now, unfortunately, we don't have any more time for questions to Arami, but if we have more time at some point, we can have some questions. But if you have any questions to Arami, ask them through chat and question and answer panel. Now we will move to our next speaker, Sudhakar Tumala Velagapudi, and he is working as a faculty at VR Siddhartha Engineering College, Andhra Pradesh, India. Previously worked as a postdoc at the University Medical Center in Germany on big data brain MRI processing along with Professor Niels K. Fogg. Today, he will talk about a machine learning framework for fully automatic registration for the big data in MRI, which the link will be shared in chat shortly. His talk is titled Machine Learning Framework for Fully Automatic Quality Control of Registration in Big Data Brain MRI. Stage is yours, Sudhakar.
Yeah, so I will share my screen.
Yep.
Can you see my screen?
I can see you, not your presentation.
Can you see it?
No, this is your name. I don't see your presentation.
I don't know, it's starting and stopping again.
Sudhakar, maybe you can talk about it. Oh, we started to see it.
You can see it?
I mean, I can see screen broadcast. You are sharing screen. I see your screen now. Yes, we see it now. Perfect. Now we can see your slide.
Okay. So I'll just talk about this work which we implemented recently at University Medical Center Göttingen regarding big data brain MRI. So so in recent years, I mean, there is availability of largely available data sets, and due to advances in high performance computing facilities, now it is feasible to conduct brain studies on big data. So this work is primarily focused on development of hippocampus profiling for epilepsy subjects. So this basically involves a lot of pre-processing steps and also involves quality control. So when we talk about this big data, so it is the quality control that is challenging since we have more number of images to deal with, and manual quality control is not feasible, and we need some sort of automatic quality control. So the quality control could start from checking the quality of images before we start pre-processing, and then the quality control comes at the stage of pre-processing, where one important or essential steps involve rigid and affine transformation of the images in a subject space to a template. So that is the step where we focused on developing some kind of machine learning techniques so that we automate the process of quality control of these registrations rather than manually checking. So if I show you, these are, for example, the big data sets currently available, or some of these data sets are currently available openly, so that you can plan your studies accordingly if you want. So the main idea here is to develop a hippocampus profile. So that could be used as a diagnostic tool to compare the hippocampus of patients. It could be epilepsy or Alzheimer's or some other disease. So the basic pre-processing pipeline involves different steps, and for this we have to use different kinds of neuroimaging softwares. So we used a Python-based interface, which is NePipe. So the advantage of NePipe here is that it allows interaction between different neuroimaging software tools such as ANTS, FSL, SPM, AFNI, and FreeSurfer. So this basically enables you to switch between different tools whenever you feel that certain processing step may be better in using certain software, instead of doing a whole process using just one software, right? So I will just briefly discuss about a few pre-processing steps. So once the images are acquired, they have to undergo this reorientation so that they come to the standard reorientation. And then the second step involves cropping, so removing the unwanted portions using the robust FOE tool in FSL. And then the most important step is the bias field correction. So this could be implemented using two methods, which is N3 in FreeSurfer or N4 in ANTS. So N4 is a bit advanced version of N3, so that that gives you more low field intensity removed version. So here the bias field is the inhomogeneity field due to the variance of the magnetic field strength along the core. So you can see that in the middle image, and once it is corrected, you can see the low varying intensities are removed. And then once it is batch corrected, then the subject space images has to be registered to the template. So there are two kinds of registrations here. One is the rigid registration, and the second one is the affine registration. So the second step, P2 to fly to corresponding T1, we can ignore further now. So this is the affine registration, where it involves 12 degrees of freedom. So that means scaling, translation, shear, and rotation. So now the current study focused on these two types, how we check the quality of rigid registration of T1 to MNI and the quality of affine registration of T1 to MNI. So for this, the three cost functions are employed: the normalized correlation coefficient, normalized mutual information, and a correlation ratio. And these were computed locally. So since we used a supervised learning, we need some sort of misaligned images. So the center one is the correctly aligned images. So this is an example shown for rigid registration. So middle one is the correctly aligned image to the template using 6 degrees of freedom, and the top one and bottom one are generated misaligned images, so that supervised learning is possible. So this is how the local costs are computed. So the first equation demonstrates how we computed the cost functions locally by defining a volume of interest. So Vs is the size of your volume of interest. We took a volume of interest of 3 by 3 by 3, and St there represents the stride and how we slide the volume of interest over the images. And M is the moving image, T is the template, and ABC there represent the range of your volume of interest. And cost is your N normalized correlation coefficient, or normalized mutual information, or correlation ratio. So correlation ratio here is the square root of the equation 4. And W there is some sort of weight because these local cost functions are computed within the brain region. And to avoid the effect of, or to avoid the say of these costs around the brain region, because at the brain region, when you define the volume of interest, not all voxels have the intensity values. They may be mostly zero intensities. So we don't want to say that the correlation or mutual information or correlation ratio is high just because it computed only on a few voxels. So this weighting factor is introduced there. And the Cn there is the number of local cost computations, which are basically averaged. And these Vs and St, the voxel size and the stride are the free parameters. So for this study, we fixed them to 3 and 3. So for cross-validation and testing, we used a data set from ABIDE, which is Autism Brain Imaging Data Exchange. So we considered 215 T1 weighted images. So out of that, 170 images were used for cross-validation, and the remaining 45 were used for testing. So in the brackets, you can see 87 and 23. They are the number belongs to the patients. So the stratified 5-fold cross-validation was implemented. And in the misaligned case, there are 5 images generated for each correctly aligned image. So you can see that the misaligned images are 5 times higher than the correctly aligned images. And then once we have the cost values computed for each correctly aligned and misaligned images, we implemented several supervised classifiers, starting from linear discriminant analysis, support vector classifier, Gaussian naive and KNN, random forest and AdaBoost. For KNN, we used a K value of 15. For random forest, we used 100 trees, and for AdaBoost, we fixed 100 stumps. So you can, since the data is misbalanced.
This is your last 2 minutes?
Yeah. So since the data is misbalanced, we used the F1 score. You can see that the F1 scores reaching almost 0.99 for testing. So the cross-validation scores are reported after doing 100 repetitions of stratified k-fold cross-validation. So these are the corresponding ROC graphs obtained on the test set. I can see that they are already almost reaching 0.99. in both rigid and affine case. And since these are developed only on one kind of data, this has to be validated on other cohorts, and also this could be extended for other MRI modalities such as T2-weighted and FLAIR. And to avoid the computation of local cost functions, and deep learning methods could be developed so that the features are learned automatically for this purpose. So the Python pipeline for this framework is available there in the GitHub. And also, if you want to have a look at the preprint, that is also available in the BioXU server. So thank you.
Thank you, Sutapa. This looks very useful, especially for the future works. And there is some questions for you in the chat, but unfortunately we don't have time to read it out on the video. Please just go to the chat panel and answer it. And now we will move to our final speakers, Maria Osetrova. And Maria is from Russia. She is, I'm going to come. She's a graduate student in the Center for Neurobiology and Brain Restorations at Skoltech in Moscow, Russia. She is doing her thesis work in the laboratory of Professor Philip Katovich. Their lab is focused on metabolic and structural organizations of human brain from the evolutionary point of view, as well as in the context of mental illnesses. And the project today Maria will talk about is devoted to the analysis of the lipid compositions of the healthy adult human brain. And her talk is titled Comprehensive Map of Human Brain Lipidome. And stage is yours, Maria.
Thank you for this kind introduction. I think all the main points you have already said. Yes, I'm from Russia. I work in laboratory of Professor Philip Khaitovich in Center for Neurobiology and Brain Restoration at Skolkovo Institute of Science and Technology. And indeed, our lab focuses on analysis of molecular and biochemical features of the human brain in scope of evolution, as well as mental illnesses. And today's talk is devoted to a more basic basic science project, the lipidomic map of healthy adult human brain, which will be like the starting point for further analysis. And first of all, why are we interested in lipids in the human brain? There are some reasons for that. First of all, a huge variety of lipid species exists, and a significant part of human genome is devoted to lipid metabolism, and brain itself has the second highest concentration of lipids in the human body. And due to variety of biochemical structure, lipids can connect differently in brain. They serve as energy substrates, they maintain cellular structure machinery, and they also use the signaling molecules, which is especially interesting for the brain. And also, lipids are understudied yet, so we have a chance to be the first ones to find something new and awesome. So here's our experiment design. We dissected 75 anatomically distinct brain regions from brains of 4 healthy adult individuals. We extracted lipids and did liquid chromatography coupled with mass spectrometry because it's the main method used for analysis of lipids. And in parallel, we did gene expression analysis for a subset of 35 brain regions from the same donors in the same regions. And we were able to annotate about 400 different lipid species from 20 lipid classes. And the result number 0 is that we have lipids that behave differently in the brain. And when we cluster brain region based on the lipid composition and based on the gene expression data, we have similar but yet quite different picture. And if for mRNA data, we see that the main factor is the spatial proximity of brain regions. So we have the neocortex clustered here, cerebral gray matter here, and some subcortical areas here. For lipids, the picture is a bit different. There is still a signal from white matter, but also some cortical regions are closer to subcortical ones. And here we investigated directly this correlation to special instances. And indeed, it was very similar for mRNA, but a little bit different for lipids. It means that there are some regions that can be closely located by maybe very different in the lipid composition. And we looked for another factor which can explain the lipid differences. And spoiler alert, this factor turned out to be the myelin content of the brain. Because when we plot the overall lipid content of the brain from our data, we see that it's very similar to the myelin map measured using structural MRI data. So we used myelin as a factor to classify lipids based on their behavior. And indeed, we see that the myelin correlated lipids are the most abundant group, but also there are lipids that have that are highly anti-correlated with myelin. Also some lipids with like flat profile across all regions, which we call housekeeping lipids, and also a small cluster of lipids we can't explain by myelin signal, but which have a specific regional profile. So we wanted to see whether we can dive deeper into these differences, and we wanted to analyze relation to specific lipid species to cell types. So we used the expression data to select gene expression markers for 6 cell types, and we assigned each lipid species to a specific cell type based on correlation scores. And we saw that indeed some cell types were specific to one lipid cluster. For example, oligodendrocytes were specific to this myelin plus group, which is expected because myelin is formed by oligodendrocytes. And on the other hand, we see that there are lipid classes that are specific to a particular cell type, which is interesting. For example, the same oligodendrocytes are a cluster of lipids assigned to oligodendrocytes are enriched in sulfuric acid ceramides as well as deacylglycerides. And we validated this result using a cell sorting experiment for mice brain with fluorescent neurons, and we saw significant difference for neuronal versus non-neuronal markers in the sorted versus unsorted fraction. And finally, we wanted to see whether lipids can be related to brain functioning, and we took the functional connectivity data from open source. And there we had a little problem because the functional connectivity is a matrix and we have vectors. So we wanted to compare them in 2 ways. We transferred a matrix to a vector. So we want the first principal component from the functional connectivity matrix and correlated it to each lipid profile separately. And we saw that this unexplained cluster of lipids was significantly higher correlated with functional connectivity of the brain, which is interesting. And we saw that there are specific lipids in this group. For example, polyunsaturated fatty acids, which are known to be important for brain functioning. And on the other hand, we transferred our vectors to matrix, creating like a biochemical connectivity of brain regions, taking either lipids or expression separately or combining the two. We did 1,000 iterations for randomization, and we saw that interestingly, MRI and lipid data is correlated to functional connectivity better than each data set separately. So these are the conclusions from our work. So I think I won't read them because I said all of them already. And I want to thank all my colleagues, especially my scientific supervisor, Professor Filip Hajdović. And here are some contacts if you want to collaborate or you want to suggest something to our work. And of course, let's discuss. If you have any questions, I will be glad to answer them. Thank you.
This is interesting, Maria. It's just like you are looking at different aspects of the brain. Could you please elaborate a little bit? With understanding lipid, what kind of things we can do? It's just like these lipid instructions or the map in the brain. What kind of information they provide for us to understand how brain, can we use it in a way that brains work, how brain functions? Do you see what I'm trying to say?
Yeah, as I said in the beginning, this particular project is a bit like, you know, basic science. So we, for example, we have the expression map of the brain, we have like protein atlas, and we don't know that much about lipids yet. But there is again a huge variety of lipids, and we want to first like see how it should be in normal condition, and then we can do comparative analysis. For example, our lab, we are planning to do the schizophrenia lipidomic brain map, which is now work in progress. We also we are now collecting samples from major depressive disorder. So when we know how this lipid landscape of the brain is in normal condition, we can see what changed, and then like taking out these lipids that are significantly different in pathological conditions, we can see what is broken, for example, which metabolic pathways are damaged in a specific condition. And also, of course, in terms of evolution.
Is it dynamic? I mean, as I said, I don't know. I don't know the literature that much, but it's just like for the lipid structure, as you mentioned, it is very early stage of understanding this, as far as I see from your talk. But the lipid structure, for example, is there any changes when we are doing something? Do you see what I mean? It's just like when I'm sleeping or when I'm awake, is there a change on it? It's not, right?
No, not in this scale, but of course, lipid composition of the body itself and the brain also is influenced by the diet. For example, in our lab, we have a very interesting project devoted to parallel analysis of brain lipid composition and breast milk lipid composition for a variety of species, and we see this correlation between the lipid composition of milk, and we see how lipids are very rapidly changing during the very early stage of development, and how these lipids from the milk come to the brain, and how human, for example, breast milk is very different from other species. So of course we think that that's what makes us humans so smart.
So you are saying that at developmental stage, it is a very important point, and it might give us some understanding how the brain is just like reshaped or developed if we understand the lipid structure well.
Exactly.
Sorry?
No, I'm listening to you. Sorry.
Yeah, it is an interesting topic. If you have any questions from our panelists to Maria, please unmute yourself and ask your questions. And it is the same if you have any questions for Maria. She's already putting her emails and Twitter on the screen, as you see now. Feel free to contact her. And she also mentioned in her email that they're open to collaborations and to understand lipid structures in different aspects.
Can I ask one thing?
Yes, please go for it.
You said you computed the myelin maps by taking the ratio of T1 weighted to T2 weighted, right?
Yes.
So how do you do the registration? Did you do T2 to T1 or T1 to T2?
I didn't do it myself. I took the data from previous research.
I see.
And I just, yes, I did. I as far as I know, this T1 to T2 ratio is not perfect for evaluation of myelin content. Like it works as zero or first approximation for us. And we see interesting results now, so probably we'll do like direct experiments to measure myelin with methods that are designed specifically to measure myelin. And
okay. Oh, you just took the already created myelin maps.
Yes, it was an averaged map from the Human Connectome project, And of course, those were not exactly the same brains, because we get the brains like dissected into pieces, not the whole brain. I think in further project, especially when it comes to some pathological conditions, it would be interesting to directly measure the myelin maps of the exact brain we are measuring and do this parallel analysis. I think that should show some interesting results, because, for example, for schizophrenia, we see that the major changes are in the cluster of lipids that are associated with myelin. So, for example, for schizophrenia, we should see some signal on the structural MRI as well.
Yeah, okay, thank you.
That's interesting. Yeah, it can be used for different diseases, maybe. Yeah, if there is no more questions, we are coming towards the end of our session. We had some technical problems to connect to Arami, but thank you very much, everyone, especially to our speakers, Arami, Sudhakar, and Maria, for your beautiful talks and discussions. And many thanks to our audience for joining us today. I should also thanks to Ali for giving us the technical help at backend. I hope you enjoyed this session and will enjoy the rest of the NeuroMatch conference and follow.
There we go.
We have a couple of minutes to start. We are waiting for our other attendees to join us. I think it is time for us to start. Hi, everyone. Thank you for joining us in this session. Unfortunately, our second speaker, Morteza Moazzami, is not feeling well. That's why he won't be able to join us. We have two amazing speakers for you in this session. And this is an interactive session. Each panelist will have a short talk and plenty of time for discussion. Please post your questions in the question and answer panel. This talk format is discussion based, so ask anything you like, anything, any direction, details, whatever you want to explore more. And if you like to ask your question on screen, please put on screen in your question, and we will invite you as a panelist into our video, and you will have time to discuss it with our speakers. Now we will start with our first speaker, Jake Strode. He's a postdoc working with Mate Langiel and Mark Stokes. They are using computational approach, mainly dynamic systems, control theory, and machine learning methods. And they combine with various data sets of monkey prefrontal cortex recordings to understand the dynamics of prefrontal cortex during working memory. In this talk, he will present a normative argument as to why they typically observe strongly sequential activity during stimulus representation together with stable delay activity in working memory task. His talk is titled Just-in-Time Computations by Non-Normal Attractor Dynamics in the Prefrontal Cortex. Stage is yours, Jake.
Thank you very much for the lovely introduction. Okay, so a key motivation for this work is that experimentalists historically have typically observed one of two regimes in prefrontal cortex during working memory, either sequential, i.e. sort of time-varying activities, or stable or persistent dynamics. And then aligned with these observations, theoreticians have come along and tried to capture these dynamics, again, with really two distinct kinds of models. One are sort of feed-forward non-normal systems, which capture sequential dynamics, or attractor systems, which typically capture stable dynamics. However, more recent studies show that actually prefrontal dynamics seem to be quite a rich combination of both sequential and stable dynamics. So in this study, we want to kind of understand what's the relationship between these two regimes and are they really so distinct? So the first thing we did is to look at some data of monkeys performing a classic memory-guided saccade task. And all these recordings are from lateral prefrontal cortex. And one of the main pieces of analysis that we did is to perform cross-temporal decoding. So what we did is we trained a decoder at one time point in the task to try and decode what the memory cue was, and then test how well does that decoder generalize to any other point in the task. And that's what I'm showing in the bottom middle here. And so what we observe is quite a stable pattern of neural activity during the delay period. So this means that a decoder is generalizing between any pair of time points well during the delay period, right? But actually we get quite sequential activity during the cue presentation period, right? So a decoder trained during the stimulus period doesn't generalize well to the delay period and vice versa. And additionally, decoders trained during the delay period don't generalize well to the go time. So we see quite distinct dynamics, both a combination of both sequential and stable dynamics in this task. And we found that the primary reason for the distinct dynamics in the different task epochs is that neural activity is strongly orthogonal in the different epochs. And I can go through more details in the discussion later if we want to. So then we want to look at some classical models and see do any classical models capture these dynamics? Okay. So all these models the dynamics of these models are described with these quite typical dynamical equations. And the first model we look at is a linear integrator put forward by John Murray from a few years ago. So when we perform cross-temporal decoding on this model, we find that straight after cue onset, decoders actually generalize to any other time point quite well. And that's because the cue inputs drive neural activity straight away into these stable delay directions, okay? And so that's quite distinct from the data, right? We don't observe this strongly sequential activity during the stimulus period. The next model we looked at is a linear feed-forward network, which is a model of sequential activity. And as expected, we see that decoders only generalize to nearby time points, right? We get this diagonal pattern of decodability, and we observe no stable delay activity, okay? Which again is distinct from the data. And the final class of models we looked at are nonlinear attractor models, so both bump attractor models and discrete attractor models, which I'm sketching here. And what we find is quite a similar profile to the integrated model, except importantly, because the model is nonlinear, you can actually now escape from your attractor state, which you can't with the integrated model. And so these models all have distinct cross-temporal decoding profiles, but additionally, not one of them in isolation captures what we're seeing in the data, right? And so next we want to look at really what's going on in the data. Can we come up with new models that capture what we're seeing in the data? So here's a sort of sketch of what we're trying to capture, right? So during the cue period, neural activity is driven in this sort of vertical cue direction here. And then only after cue offset does activity really seem to sort of rotate or move into a stable delay direction. And then neural activity kind of stays within some attractor state during the delay period, okay? So here's the model we came up with. It's a non-normal model, so it's a model with a strongly feed-forward connection. And it's just two neurons and it's completely linear, so it's hopefully quite a simple model. Neuron 1 is an integrator, so it just has completely persistent activity, okay? And then neuron 2 just connects to neuron 1 in a feed-forward way. And here I'm sketching the flow field of the model. So I'm sketching neuron 2's activity against neuron 1's activity. And I've labeled neuron 1 as the slow mode, right? It's an integrator. So whenever you apply an input to neuron 1 and you release the system, you just stay there for all time, right? But what we find is that in such a system, in this non-normal system, if you apply an input in what we identify as the most amplifying mode using control theory, which is basically an input that strongly aligns with neuron 2, you actually generate huge amplification along the slow mode, right? Much greater than if you started your system off along the slow mode, okay? And again, we see that this input is strongly orthogonal to your stable delay direction, much like what we're seeing in the data. And this is really different from what you'd expect in a normal system, i.e. a symmetric system, okay, which many classical models are based on. In a symmetric system, we find that both your most amplifying mode and your most stable mode perfectly align, they're identical. And so in such a system, if you want to end up in the stable mode, your only option is to start there, right? So you're not gonna get orthogonality from stimulus to delay period. But in a non-normal system, you can utilize these feed-forward pathways. And in fact, in a non-normal system, your most optimal input should be aligned with the most amplifying direction, which will be strongly orthogonal to the stable direction. We tested this not in just these toy handcrafted models, but in random integrating networks, up to 1,000 units. And we see that actually, if you align your input with these most amplifying directions, you start off strongly orthogonal to the delay direction, and you get this huge amplification into the delay direction. So you can robustly encode your stimulus quite well in these models, but your input starts off strongly orthogonal to the delay directions. Okay. Then we want to ask ourselves, do we find elements of these non-normal or these feed-forward dynamics in the data itself? So we fitted linear dynamical systems to the actual recorded neural firing rates. And in a cross-validated way, we see how well do these models capture the data. And so a linear model actually captures the data quite well for both monkeys. But if we constrain the model to be highly non-normal, so an upper triangular network, for example, the model actually fits the data similarly well to an unconstrained model. And additionally, if we constrain the model to be symmetric, i.e. a normal model, which doesn't have any feed-forward pathways, we get a considerably poorer fit, and it's actually poorer than just a random model. So that provides quite direct evidence that actually the data itself is governed by a non-normal system. I mean, here's a sort of cartoon sketch of what's going on. So in a classical model, your input drives you straight away along these stable, persistent delay directions. But actually in the data, and what's expected, what's optimal actually in a non-normal system is actually your inputs drive you in an orthogonal direction, which makes use of these amplifying feed-forward pathways. And only then after stimulus offset does your activity then really increase along these stable directions. Okay. And then in the final part, I'm going to just talk about training nonlinear networks on the task. Okay. And so we have these vanilla ReLU nonlinear networks. And what we found is if we apply our training objective just before the go time, which means we want to separate neural activity corresponding to different Q conditions just before the go Q comes, right? Then we obtain stable delay activity and sequential activity from stimulus to delay, just like in the data. So out of the box, you really make use of these non-normal feed-forward pathways, and you don't have to like enforce it into the system itself. And so these dynamics are quite similar to what we observe in the data. And we contrasted that with other cost functions. So if we apply the cost function, what we call a classical way at the go time, so it's literally what the actual motor response should be, we get quite sequential dynamics, much like classical feed-forward models. And if we apply the cost during the full delay period, like a kind of full working memory cost, if you like. So as soon as the stimulus goes off, I'm representing the stimulus in a stable way. We get classical attractor models. Okay, so your Q-related dynamics are very similar to your delay dynamics. And then a final bonus is we looked at data from a task with a distractor. So it's a classical memory guided saccade task, but there's an additional delay period where a distractor comes on. And what we found, to cut a long story short, is that our just-in-time networks capture this data set quite well when we perform cross-temporal decoding both in the full neural activity space and in a low-dimensional subspace. And actually, the delay trained networks also perform the task, right? But they are completely resilient to this distractor. They don't change the neural activity at all, which is not what we see in the data. And a classically trained network, again, just generates sequential activity, which is unlike what we see in the data. And so that's the end. Here's a brief summary. So our observations were we have stable delay activity in PFC, which is orthogonal to the stimulus-related activity. Classical models typically don't capture this. And our main explanation is that in a non-normal system, which again, I should say is guaranteed with probability 1, your circuit will be non-normal. There will be feed-forward connections in it. Actually, your optimal inputs should be aligned with the most amplifying directions, which naturally themselves are orthogonal to the stable delay directions. And the computational objective PFC seems to be performing, which gives rise to these dynamics, is just to have just to separate Q conditions just by the time of the go Q. And then you get these naturally sequential dynamics and stable dynamics. And our bonus is things seem to generalize to this task with distractors. And so that's it. Thank you very much for your time, and I'm happy to take questions.
Thank you, Jake, for this nice and short talk. We have a question. If you have any questions for Jake, please use question and answer panel and the chat to ask your question. We have questions from Matthias Henning asking for how do you regularize to get non-normal models in the linear nets? Just enforce triangle weights.
Exactly. We just enforce the network to be triangular. There's actually two variants. One is strictly upper triangular, and the other is we also train the diagonal, so we learn the time constants. Both actually give basically the same result. So that's exactly what we do. In these nonlinear networks, we have no constraints at all, right? So they just naturally, the networks are naturally non-normal. And so they're sufficiently non-normal that we observe also sequential dynamics there. But yeah, when I fit linear models to the data, I enforce them to be upper triangular.
Okay, thank you. Another question is from Bin, asking for what exactly do you mean just-in-time network, just-in-time objective function?
So yeah, so I didn't have anywhere near enough time to try and explain it. But basically, we have a classical cost function for this task, which is a cross entropy loss, which is just used in machine learning models all the time. And the difference between these different models here is the time at which we apply that cost function. So in a just-in-time network, we apply the cost just before the go queue comes, which kind of aligns with roughly, I think, how many people think about prefrontal cortex, that you want to represent the stimulus and have that information ready to be read out just when the go queue comes. So you want the motor regions to be able to read out what the stimulus was in prefrontal cortex just when that go queue comes. And so the just-in-time cost function, for example, for a fixed delay task is we just apply it just before the go queue comes every time at the same time. And then a random delay task, we apply it just before the estimated earliest go queue and then up until the go queue on that particular trial. So I hope that sort of roughly answers that question.
So in prefrontal cortex, you know, we have this pre-choice activity as well, what we see very clearly. But I'm just wondering, in your model, can you capture that as well?
What do you mean by that? So pre-choice, so you mean just before the go queue?
Yes, before the go queue.
But what type of dynamics do you mean? That's like ramping activity or something like this, you mean? Or?
Expectation, getting ready, this kind of, because prefrontal cortex carries these early activities. Because currently, as far as I see in your model, you're always looking after 0. I'm just wondering, because it is a recurrent neural network, if it is just in time, it should be fine to look before and after, right? I don't know. I'm just like, what do you speculate?
I mean, nothing's really going on in our model before the queue comes on. It's just sort of random dynamics. There's noise in the system, so it's just randomly moving around a spontaneous state. But just before the go queue comes, I mean, we have analyzed the single units in the model, and we see both units that are completely persistent, units that are sort of more active during the early part of the task, and units that are more active just before the go queue comes. So we see these quite heterogeneous dynamics, which are also observed in data. But yeah, that's kind of all I can say really about that, I think. Yeah, I hope that kind of slightly answers the comment.
And one of our attendees is asking if this is published data.
Right. So we're writing up as we speak. Hopefully we can put something online this year, although maybe that's a bit too hopeful. I've got a short recorded talk online, which you can also see, which is basically this poster again. But yeah, if you're really interested, then of course email me and we can chat. I've chatted with a few groups so far about this.
And this video will be available in YouTube probably next week, as soon as our technical people will handle it. And you can reach out to these talks all over again next week. If you have more questions for Jake, feel free to ask. I think Tal has a question.
Yeah.
Yep.
Lovely talk. I was wondering also, in line with what we asked, how do you incorporate things like temporal attention? Like if you know that the target is going to appear in an X time, and you have like a ramp up of activity that predicts it, or something that is going to deviate from the stable activity. How can you incorporate this into this model?
I mean, so basically, I guess my general answer to these questions is that, all of these that have been observed tasks and how neurons change their activity throughout the task, these kind of things seem to emerge often out of the box in these kinds of networks when you train them on the task. And so these networks, the ones I'm showing here, I'm literally just training them to perform the task. And then whatever dynamics we get, they're just out of the box, they just happen. So I'm not enforcing anything in the system to do any kind of preparation activity. It's just literally trying to perform the task. Now, for the fixed delay variant of the task, there's an important side note to your question, which is that in the fixed delay task, the network knows when the queue is coming. And because these are recurrent networks, there is noise, but still they can predict really well when that go queue is going to come, much better than an animal, right? And so one could argue that when you train networks on a fixed delay task, that's probably pretty different from how an animal would perform a fixed delay task. The animal is probably estimating roughly when the go queue is going to come. It doesn't have a clock in its mind that it perfectly knows. So that's maybe a difference between But for the random delay task, I think that's pretty reasonable that the networks are kind of performing them and using similar mechanisms to what the brain might be using. But yeah, for a fixed delay task, you might have to add something else in to make it appear as though it's, because it doesn't need to use the go queue at all in a fixed delay task. Basically, the network can just completely neglect the go queue and just use time. So yeah, sometimes you might have to add something in there to force it to use the go queue.
Thank you.
So for example, like on a match to sample task, it has to use the second item to perform the task. Whereas in the memory guided Saccard task, as soon as the stimulus comes on, it knows what to do straight away, right? This is like a classical comment by, Earl Miller, for example, says that the memory guided Saccard task isn't a very good example of working memory because it's already motor preparation, right? So yeah, that's a kind of side comment as well.
There are no like catch trials where you don't get the go queue.
Right, no.
Okay. Thank you.
Our previous attendees is asking for the link that you mentioned for your previous short talk.
Yeah, I will get that. I'll put it in the chat.
Now we will wait for couple while Jake is getting the link for you. We will wait a couple of minutes for Tal, because our second speaker is not with us. We will wait until the 11:30 for London time for Tal to start her talk. Jake, you need to do all panelists as well as the attendees.
My apologies. I will do that. Oh, yes.
Yeah, if you have any questions for Jake, please feel free to ask. We have we have 10 minutes basically. Any questions?
Yeah, feel free. I can put up the slides again, go into any other details. I think a typical question that people also ask is that, why haven't we kind of studied this too much before? And I think there's many different data sets, and I think all the data sets are kind of different and have some subtle differences and some quite strong differences. And it seems in our data set, things are a lot more sequential from stimulus to delay than some other data sets. So that may be some reasons why other models haven't tried to capture those dynamics. Yeah, that's another comment I should say.
I mean, because you are focusing on the prefrontal areas, they are, yeah. I mean, these animals are monkeys. Monkeys are well-trained animals, just like they trained, I don't know, maybe 6 months or so. That's why when they are doing this task, they know it really, really well. Yeah, I would expect that there is this getting ready activity is really dominating, that, especially in time. If we are talking about in time, getting ready, answering, or getting outcome, getting ready for outcome, expectation for outcome, this kind of things. If I don't know, I would expect I don't know how to interpret, implement this into the model. It's just like hard, but it would really might be interesting to.
But I mean, for example, a sort of answer to that is that if these monkeys really are, you might think as ready as they possibly can be for this task, you would think there'd be no sequential dynamics from stimulus to delay. Why wait around for your activity to then get into the stable regime? Why, as soon as the cue comes on, why aren't you going straight into the stable regime, right? So, and so that's kind of why we're answering that, is that actually the best thing to do is to not go straight in there. If you want really robust, stable delay activity, you can actually use these orthogonal directions, which give you even greater amplification. And so even when the monkey is super ready, it's still using these orthogonal directions in the activity, and it doesn't go straight into that stable, working memory subspace straight away.
Yeah, we have a couple more questions for us for filling in this time from Matthias and Tim. And Matthias is saying that one is, if you have looked at the PCs that have task-related activity Do you find the relevant signal in the first view? Asking as in motor cortex, a lot seems hidden in low variance, at least in mouse. We found at least in mouse.
Yeah, let me share slides and go back. So yeah, so at the bottom here, we're doing one version of a sort of PCA plot. I think I've got some in the backup slides, but maybe I won't bring them up. And so this is basically, we apply PCA, we identify the top 10 PCs in the neural activity in one task epoch, and then project neural activity from any other task epoch through those PCs and just see how much variance do we capture. So the diagonal is literally what's the % variance captured by the top 10 PCs in each epoch. So for example, the Q epoch is lowest dimensional, right? And the spontaneous period is highest dimensional. But we see that the off diagonals are all really, really low. So basically things are really orthogonal within the different task epochs. The only discrepancy is from Q to Go, because actually the Go signal is all stimuli being presented at once. So that's probably why there's some overlap. And yes, so we do see the relevant signal in the first few PCs during the stimulus period. During the delay period, things are a lot more noisy. And so you probably need quite a few more PCs to capture the signal in the delay period. And they say, yeah, there's hidden in lower variance. Yeah, I think in the delay period, there's definitely the signal is hidden in many, many more PCs. And the stimulus period is really fun. The top 2 PCs really capture a lot of what's going on. But yeah, the delay period may be more like what Matthias is saying, suggesting then. And then I see another question from Tim Vogels, my old boss.
We have another questions for you from Tim Vogels. He's asking, how does this relate to your previous work? Or better, where are the explicit differences? Can you rebuild socks to make them do what you want these nets to do?
So hi Tim. Maybe he could put his video on at some point as well. But yeah, these, you can think of these as so a sock is a stability optimized circuit. And basically, these are a good model of motor cortex. So in motor cortex, the circuit is really non-normal. There's loads of feed-forward pathways. You get this huge amplification of activity when you perform a movement, and then activity decays back to baseline when you're done with the movement. And that makes sense. But in prefrontal cortex, it's kind of like, well, why would you want all these non-normal amplifying dynamics? But the difference between prefrontal cortex and motor cortex is in motor cortex, everything goes back to 0. But in these models, everything has to go to a stable mode. You have to maintain the stimulus during the delay epoch. And so the model is actually really similar to these stability optimized circuits, that the models are non-normal and they make use of these non-normal pathways, and we use these amplifying directions, but activity falls onto a persistent integrating mode during the delay period, which is really different from these stability optimized circuits or models of motor cortex, where you actually want things to go back to baseline, because you want to forget about the movement once you've done it. You're just a movement generating machine, right? But here you're a memory machine, so you're trying to maintain what the stimulus was. And yeah, I think was the second part of this question, can you build socks? Yeah, I mean, what I'm showing here are random networks of different sizes. They're not socks, they're just completely random, but they're random enough that you get these feed-forward pathways, and the system can make use of these feed-forward pathways, and you get this large amplification onto the stable modes. And so you don't need something as extreme as a sock. In fact, just a random network is non-normal enough to capture what we're seeing in the data. Yeah.
Okay, this looks good. I think we can now move on to our next speaker. Thank you very much, Jake. It is a nice model, and let's see how it will fit with the other neural data and everything. And as I mentioned, our second speaker are canceled. That's why we will move to our third speaker, Talseidel Malkinson. And she is a clinical psychologist and cognitive neuroscientist doing her postdoc at the Paris Brain Institute with Paolo Bartolomeo. She studies neurotypical and pathological visual attention in the human brain using a variety of neuroimaging methods and psychophysics. She completed her PhD in neurobiology and neuropsychology at the Hebrew University of Jerusalem under the supervisions of Ehud Zohari, working on the representations of visual motion in the brain. Currently, she's working on mastering the art of doing science while in the confinement with kids. Now, her talk title is Intracerebral Recording Study of the Spatiotemporal Dynamics of Human Exogenous Attention. Stage is yours, Tal.
Thank you very much. So I'll share my screen with you first, and then we'll just start. So I'm going to basically speak a little bit about my project, this current project, and not go into too much details, but hopefully during the discussion we could go into as much detail as you want. So let's start. So what is exogenous attention? Exogenous attention is the automatic stimulus-driven orienting of attention towards salient stimuli. We used one of the classic ways to measure exogenous attention, the Posner cueing task with peripheral cues. Here, participants have to detect as fast as possible a target, here the X, that is preceded by a non-informative peripheral cue. The cue appears in a congruent location or in an incongruent location, and has to that appear in equal probabilities. And the impact of the cue depends on the stimulus onset asynchrony, the SOA, the delay between the onset of the cue and the target. So here's the typical RT pattern, shown also by our patients. The same stimuli with a slightly different delay lead to opposite behavioral patterns. In short SOA, there is RT facilitation. Congruent RT is faster than incongruent RT. Whereas in the long SOA, we see the opposite. Incongruent RT is faster. This phenomenon is called inhibition of return, and that is the center of this talk. We wanted to know what is the neural basis of this behavioral pattern, and we hypothesized that the responsible mechanism must be sensitive to the timing, the spatial location, and to task relevance. We also wanted to dissect the processing stage at which inhibition of return occurs from visual to response processing. We used intracerebral EEG that provides a high-resolution window into human neural activity. We recorded from electrodes implanted in epileptic patients while they were performing the Posner task. Pulling all our data into a single pseudo patient, we had 1,403 usable contacts from 28 patients. We extracted target-locked high-frequency broadband power known to be associated with action potentials. And we did it in each of our 8 conditions. So we had long and short SOA, congruent and incongruent, target contralateral or ipsilateral to the contact. We first wanted to find where in the brain the activity follows our experimental manipulations. In order to reveal the central patterns of neural activity in this big data set, we developed a custom k-means clustering approach, allowing us to find clusters of contacts with similar temporal activity, taking into account all our conditions simultaneously. We found 3 clusters with activity patterns that changed along our experimental conditions. The first one, here in yellow, showed fast contralateral responses. The red and green cluster showed bilateral responses, but the red one showed spatial modulation, where contra responses were stronger than ipsi, while the green one showed stimulus type sensitivity, where target was stronger than cue. Importantly, at odds with the yellow cluster, the red and green clusters showed an integrated response for cue and target in the short SOA in the top panel, but segregated responses in the long SOA, the bottom panel, demonstrating its sensitivity to cue target timing. And you can also see that the 3 clusters form the temporal gradient of activity. Next, we wanted to see where are the clusters located. The yellow visual cluster was located in the occipital regions and also around the frontal eye field. The red and green clusters are located in posterior temporal cortex and frontoparietal networks and divided between them, the TPJ between them into a caudal and a rostral partition. Thus, the 3 clusters form together also a posterior to anterior spatial gradient. The red cluster was more lateralized to the right hemisphere, like some attention networks, but the green cluster was left lateralized, perhaps in relation to the right-hand responses in the task. We then wanted to see if these clusters show inhibition of return-associated activity, and if so, is that too lateralized? So all our clusters showed interhemispheric differences, but only the red cluster showed inhibition of return-associated activity that was also lateralized to the right hemisphere. Specifically, the peak time and the decay of the response to the target were faster in incongruent trials, similarly to the RT pattern. Next, we wanted to see if we can directly link neural activity to response time. Looking at differences in target locked activity at different RTs can reveal response-associated modulation. We pulled neural activity across all conditions in each cluster and divided the trials into RT beams, from the fastest RT in pink to the slowest in yellow, to see in which clusters we see response modulations. So the decay of the activity in the red cluster was modulated by response time, as was the rise of the activity in the green cluster. Visual cluster activity showed late RT modulation, reflecting perhaps a top-down effect. A complementary approach would be to examine response-locked activity. Now, the RT denotes the onset time of the target, and hence dividing trials into RT beams can reveal target-associated modulation. We repeated the clustering procedure, this time on response-locked activity, and found 4 clusters forming also a spatiotemporal gradient. We mapped the response-locked clusters onto our previous target-locked clusters and examined the activity across RT beams. In the most posterior cluster, we could observe target-associated modulation of activity. So to sum up, using a novel clustering algorithm, we identified a posterior to anterior spatial gradient, demonstrating also a gradient of activity from visual-related to response-related processing. The neural basis of inhibition of return could be in the nexus between perception and action, in the caudal TPJ prefrontal cluster here in red, which covers a subpart of known attentional networks, showing also right lateralization and inhibition of return-related activity. That's it. Thank you. And I'm ready for some Q&A.
Thank you, Tal, for the nice talk and the beautiful results. And if you have any questions for Tal, please use question and answer panel or chat to ask your questions. And I mean, could you please elaborate a little bit more about your cluster system? Because it's
sure.
Very interesting. It's just like to see.
Yeah, just really briefly. Yeah, so I can, let me show you. So I'll share my screen again. And let's so first of all, we took only the responsive electrodes. We had a separate statistical test that identified out of these 1,400 contacts, only those that responded reliably in one of our conditions compared to baseline. And then we entered all these conditions and all this activity across the 8 conditions into a k-means, and we tried to minimize the distances between the centroids of the activity across all these 8 conditions simultaneously. And we tested, of course, as with k-means, solutions with different numbers of clusters. And actually the one that was the best solution was a solution with 6 clusters. And the 3 clusters that I showed you are are the 3 that showed the activity difference across all our experimental conditions from these 6 clusters. So I can show you the activity of the 6 clusters if you want, if we have time for that. So these are our 6 clusters. So this was this yellow. You see my mouse?
Yeah, I can see them. They are.
Okay, so this is the 3 clusters that I was talking about. This occipital visual fast responses, contralateral responses, this red cluster and this green cluster that divide the TPJ, the temporoparietal junction between them, into a more posterior section and more anterior in the supramarginal gyrus. Now, these two other clusters show late inhibition and late activation, more close to the time of the response. But they showed like the same activity all across our conditions, so they weren't the clusters, the networks of interest for us in this task.
Okay.
In this
so
analysis.
There are many units in the non-responsive clusters as well.
Yeah, definitely. Mm-hmm.
That's interesting. Yeah, that looks really nice. I was just expecting some, you know, as we see in the task, when the task is more executive functioning, we see more activities in the anterior areas as well as the medial areas. I was trying to see if we can see any differences between the lateralized versus medial areas, but
in the prefrontal, you want to say, you mean?
In the medial versus lateral areas, you know, because
Okay.
medial area.
Yeah.
But it seems like, yeah, it's not
the gradient is mostly posterior to anterior. You can see between this red cluster and the green cluster, you can see some differences in the frontal lobe mainly. I think maybe on this axis of medial and lateral. I have to characterize it better. Yeah.
Yeah, that looks beautiful. If you have more questions for Tal, you can contact her through her email, and I think in the chat panel we shared her. Yeah.
My Twitter.
We can share your Twitter exactly. And if there's no more questions, I'm going to end our sessions. I would like to thank Jack and Tal for the beautiful talks, and many thanks for our audience for joining us today. I should also thank to Ali, who is out giving us technical help at the back end. Although you don't see her, this is possible. This session is possible with her helps. And I hope you enjoyed this session and will enjoy the rest of the NeuroMatch conference.
Hey everyone. Been told that we have a little bit of time as people come in to the webinar. So I just want to use this opportunity to tell everyone in the US to make a plan to vote. If you've already got a plan, why don't you go ahead and message one of your colleagues and make sure they have a plan. And if you're not in the US or you can't vote in the US elections, why don't you go ahead and give one of your US colleagues or friends, send them a message and just make sure they have a plan because it's super important. Can't stress that enough. So we've got a really interesting set of talks here that I'm looking forward to hearing. I need to get my notes on what the order is. So the first talk that we're going to have is from Vitor Piera. And he is going to give a talk on the characterization and analysis of visual processing in humans using fMRI data and machine learning techniques. So this should be a really interesting talk. So Vitor, you have the floor.
May I share the screen?
Yeah, please.
Oh, nice. Ready to start? So my name is Vitor, and I'll present my work with collaboration with Julius Zanon, Frederico Gadelha, and Randy McIntosh from Universidade Federal de Minas Gerais and University of Toronto. An introduction first. The visual system is one of the most important human sensory systems, both in practical importance and in dedicated area in cortex. Its diversity in terms of area is big, and there is still lacks in knowledge about its function. The aim from this work is to generate insights about how image feature relates to brain activity by analyzing a public experiment dataset. I'll show some details about the dataset I used, VIN1. The dataset was collected by Kendrick Key and Thomas Nacellaris in UC Berkeley. The authors published their findings in a Nature paper in 2008, and made it public in CRCNS. The fMRI data was collected in the posterior portion of the subject's brain, covering occipital cortex and parts of parietal and temporal cortex. A set of 8,750 images were displayed through a virtual reality glasses, while activations through the brain was recorded through fMRI scanner. And so these are samples of the images. There are many kinds of images: humans, animals, landscapes, textures, buildings, and so on. Now I'll discuss some of the results and the analysis we made on the data set. We first try to answer this question: are there cortical regions that respond similarly to the images? And we gave answer to this question using a non-supervised approach. With the k-means algorithm, we divided the dataset into 10 clusters. In the figure, we see this division in a null-dimensional space obtained with t-SNE for visualization. And so we have the cluster division in this space. We then plotted these clusters in a anatomical mask, and we see in all the cluster a sagittal symmetry. This observation is according to the experiment design that showed the same image to both eyes of the subjects. And in most cases, the clusters are restricted to certain areas, certain regions of the brain. For example, this one that is restricted mostly on V1. This other one that may be related to the ventral stream of informations, and so on. There are many others that don't have this anatomical coherence, and we hypothesize that these ones are related to distributed processing in the visual cortex. And then we try to answer these other questions. Are there groups of images that cause similar activity in the brain? And we use the same approach from the previous question to answer this one. And here we see the clustering from the images in the dataset. We used 5 centroids this time. And here we see a picture that organizes the the data. Sorry. This figure, we sorted the axis according to the cluster voxels horizontally and the clusters of images vertically. And we see patterns in the relation between some groups of images and the activation all over the brain. So in this group, in this image group 1, we see a high activation in all the areas, and in this group 2, we see activations negatively in all the areas. We'll explore this further later. And how those sorry, the next question we try to answer is how does image complexity relates to cortical activation. Then we use the border density as a complexity metric for the images, and we apply the Kenny algorithm for border detection and compute the border density, summing the border pixels, these ones lighter pixels here in these black images, over the total pixels in the image. And so we see that this image, which has more borders, more white light pixels in the images, will be considered more complex than this one of the polar bear here, that has less borders. We then made some comparisons in between the images and we saw that the pattern group, we classified the images in categories of animal, nature, person, transport, people, building, and we saw that the pattern category, I'll show some examples later, is the one which has most of the, it has the border, the more higher mean border density of all the groups. And we also see that the same category has the higher proportion in the voxel groups. This one in the group 1. Here are some examples of this pattern category, which has many borders and many details. So these are very complex images according to this metric. And we made a comparison between two image groups. The one which has the pattern category, this one in red, and another that has not many pattern category and has a negative activation all over the images. And we saw a little but statistically significant mean difference on the border density. And we can, from this, say that the image complexity is related to an increased activity level on all over the visual cortex for this subject. Extending the analysis, we try to answer which region is most responsible for this difference in activation. And we saw that only the cluster 3, group 3, has a statistically significant correlation between the activation and border density. The group is illustrated here, this group 3, and the interesting thing about it is that this group comprises a big amount of V1 area, about 30% of its volume, and V1 area is an area responsible for for the lowest levels of visual processing in the brain. It's responsible for extracting borders, corners, or orientations, and that's all according to the metric used here. And now I'll discuss some of results and show some propose for next future work. So first, we should see how other image feature metrics relate to activation. Using other feature metric may show relations present in other clusters. This cluster here is related to the ventral pathway. It goes from V1, V2, V4, and goes down there to the inferior temporal cortex, and features related to object recognition may be interesting to analyze in this cluster because we may find interesting patterns. And the dorsal pathway, this one here, we have some groups that may be related to this one, using features related to spatial localization would lead us to interesting patterns in these clusters. And other future work is to find to model the relation between image and brain activation using a convolutional neural net. An encoding model which takes as input the image and tries to predict the neural activations on each voxel would mimic the brain, and we could make some comparisons between these two, the model and the real brain. And this would give insights both for the brain function and the model specificities. And that's all I have to say. And here's some of reference.
All right, thank you very much, Victor. That was a really excellent talk. It's great that you're able to kind of recapitulate some of the known properties of V1, like this elevated activation to edges and oriented lines. I think that's really says a good indicator that your method is picking up on some really biologically relevant features. So I like that. So we have time for questions. We have time for questions if anyone wants to post them in the Q&A. I'm going to ask a quick question to start us off. What do you make of the fact that the pattern of activation that you see is so diffuse, whereas we know that the visual system is oriented, at least in its most early components, into really discrete processing modules. So have you thought about that at all, or do you have an explanation for that?
I didn't understand well the question. May you?
Just that there's so when you looked at your voxel-wise clustering, you had mentioned that it was really diffuse, and that you weren't seeing discrete, like kind of similar modules of activation, or like voxels that express similar activations being kind of grouped. And I was just wondering if you wanted to comment more on that particular observation.
Oh, nice. So we found many clusters that aren't so anatomically cohesive, but there are some that are very coherent. Mm-hmm. There are no cluster that constrict some visual region, but there's some clusters that may be related to some visual pathways or pathways of visual processing, such as ventral processing and ventral pathway and dorsal pathway. We see that cluster 9 has a nice relation to the ventral visual pathway, but we have that clusters that don't have a constricted anatomical location. We think that these clusters are related to diffuse processing in this area, both for feedbacks from between areas and maybe the flow of information between areas, like a distributed processing this way. I don't know if I answer your question well, but
no, that was a great answer actually. And I did find that the kind of as you were looking, some of your clusters or some of your networks that you were picking up did have really well-defined clusters. So it was interesting that you got something of a mix. So now we're going to have to move on to our final talk, but I want to thank Victor again for this super interesting presentation. Our next talk is going to be from Garikots Lerma Usabega from, I apologize if I didn't get your name correctly. You can correct me when you begin your presentation. So I'm sorry about that. But his presentation is titled "The Gradient of Population Receptive Field Stimulus Dependence in Ventral Visual Cortex." So you can take it away.
Thank you. So Gary is enough. I was lucky enough that my name works well in English.
Yeah, all right, Gary, it's going to take it away.
Yeah. Okay, so good morning, or wherever you are. So today I'm going to show one result that we've got, and this result is based on a work and in my previous group, which is Rosemary Lee and Michal Ben-Shahar and Brian Wandel. They published it in 2017, and it is going to be a very simple presentation, and I'm very interested in hearing feedback and questions, because we are now thinking on how to explain these results. So basically, we are going to show the stimulus dependence in the ventral visual cortex, so population receptive field stimulus dependence in the ventral visual cortex. So my group, well, now it is known from many years that there is a region in ventral occipitotemporal cortex that is more responsive to words, right? That
Gary, I just want to make sure.
Yes.
I just want to make sure. Are you supposed to be sharing your screen at the moment?
I'm not. Oh.
Oh no, you're not
Sorry.
sharing your screen. Yeah, I just want to make sure.
Oh shit. Sorry.
No problem.
No, I was in full screen. Is it working now?
Yes, it's working now. You're good.
Okay. Well, the first slide was just the title. So, and I was just showing this and this plot now. So, do you see your faces? No.
We can see. We depending on how people have their screens configured, we can see our own faces. Yeah.
Okay, so basically what I'm showing here is an inflated brain surfaces, left hemisphere. This is the ventral part, so this is the anterior part, this is the posterior part, so this would be V1, and this is the ventrocipitotemporal cortex, right? And it has been known from a while ago that there is a region there that usually is being called visual orphan area that is more responsible to words than other categories of visual stimuli. And in our case, we call that the BOTRC or BOT reading circuitry. And now it's not moving. Okay. And just in case, I want to explain as well what a population receptive field is, which is the technique we use in our study. And the population receptive field would be of a voxel, and voxel in the visual cortex is the portion of the visual field in which the stimuli, so this would be a ring that is expanding, that the stimuli is evoking a response in these voxels. And the visual cortex, we know that is retinotopically organized. So we know that that the organization of the visual cortex have a direct topographical relation with the visual field. And these population receptive fields we can characterize with the location of the center, which can be Cartesian, like X and Y, or we can use angle and eccentricity to show where the center of the PRF is. And then we've got the size as well, because usually the more foveal voxels, the size of the population receptive fields are smaller. And when we go more to the periphery, the population receptive field size is usually bigger. So in 2017, I was talking about this other work, and they showed that the eccentricity, so the location of the centers, is stimulus dependent. So in this region, in the BOTRC. So, and it's a really big difference. So between 2 and 4 degrees, both ventrally and dorsally. This is area around an IPS, IPS 0, 1, around this area. We see that the words are much more foveal, so that the center is usually located around less than 5 degrees, and the checkerboards and usually 6, 7 degrees. So it's a big difference, and this happens as well in the field of view. So basically, the field of view of a region of interest, and in this case, our region of interest would be the BOTRC, the visual orphan area. So it defines the portion of the visual field that evokes a response in any of the voxels in that array. So it's basically we are aggregating the PRFs of all the voxels in our ROI, right? And we see how the field of view available for words, it's much more foveal than the one for checkerboard. So basically, this result and this result are the same, looking at the differently. And oops, sorry, I went back. Yeah. So to understand this stimulus dependence, we run this new project. And we want to show the main result we have, and to be able to discuss with you. And what we did is we analyzed the areas that are likely the inputs to the BOTRC. So the signals here, so this is a little bit simplistic, but we assume that, and as the previous talk was saying, first from the eyes, we go to B1, B2, B3, HB4, BO1, and then only then it will arrive to the BOTRC. And for that we used 20 subjects, only English, that they were from the previous experiment, and a new set of subjects that they were watching both Hebrew and English. And we were using the moving bar paradigm as we see here. And inside the bars, we have our checkerboards or words, which is this case. And these are English words. We were placing Hebrew words as well. And as a control, we place false fonts as well. So we are usually the bars have been used typical retinotopy experiments, but here we use this evolution on the paradigm. And let's see if it moves. Yeah, and the main result we observe is the one on the title, that there is this gradient when we move from B1 all the way to BRTC. We see that B1, B2, B3, we already start seeing a shift all the way to the BOTRC. So I'm going to explain a little bit what's on the plots here. So in the x-axis, we've got the eccentricity for words. Eccentricity, how far from the center is the center of the PRF. From the fovea is the center of the PRF. And the y-axis, we've got the eccentricity for the checkerboards. And if there wouldn't be this stimulus dependence, all of the points would be in top of the identity line. And we added this 5-degree bar in order to control for the noise. So these are noisy measurements, and if it's inside the bar, we would say, okay, it's basically the same. But we see how starting in B1, which is always imperceptible, this gradient, this stimulus dependence starts growing and being more significant. In HB4, it's basically clear that all the checkerboards have more eccentricity, and B1, BO1 as well. And in the BOTRC, basically there are no word PRF centers that are wider or more than 5 degrees. And basically they disappear from, we see nothing here, and basically all the checkerboards are in organized vertically, right? That's our main result, and what I would like to discuss with you later on. So just to validate the method, we showed false fonts, which false fonts, you know, they look exactly the same as words. So visually, they are the same as words, and they have the same statistical regularities as words, but they don't carry any linguistic, no phonological, no meaning, no semantics, no any linguistic information. And we see that when we use false fonts, B1, B2, B3, B4, BO1, all of them look very symmetric to us. In the case of BOTRC, we see that maybe there is some preference for the false front, so more eccentricity for the false front, but it's not statistically significant. And we did the same with the Hebrew and English words. So these are Hebrew speakers that they were watching these two different scripts, and basically the results fall in the identity line as well. So, well, first of all, we are trying to understand why this might be, why might be happening that this this shift is occurring, and why the population receptive field when we are watching a word is different to the checkerboard. And then another thing that we are working on is that basically this measurement is giving us the field of view available when we are watching words is different to the field of view available when we are watching checkerboards or other type of visual stimuli. And we think that this could be useful for characterizing the individual reading abilities. So that's one of the directions we are working. And yeah, I think this is basically it. So thank you for your attention.
All right, thank you very much, Gary. That was an excellent talk. Really a lot to think about. We have time for only a few quick questions. So if you've got a question, please post it in the Q&A. I'm gonna ask a question about, well, actually, I'll go first to the question from one of our panelists. Jesper asked about the reliability of the two different stimulus classes with the PRF reliability. So how does the R² value compare between words and checkerboards?
So that's a good question. And I had the slide prepared here. Not the slide at Robin, because this is something we'll be working on. So can you see my screen now, or are you seeing still the presentation?
We still see. Thank you for your attention. But yes, we see your screen.
Okay. No, then I'm going to stop sharing, and now you just don't see anything, right?
We still see your screen. I'm gonna have to. So if you can answer really quickly, we have time for one more question. Otherwise, this might be
okay. Yeah, I wanted to show the plot, but now I'm stuck in Zoom wall. Okay, share screen. It's going to be very quick because he will see it. So now you see the thank you for your attention again, right?
Yeah, we see your screen.
Okay, so this is the R² and in the different places, and we see that the variance explained for when we are watching words or checkerboards, it's basically the same. Yeah. So I think one picture speaks more than 1,000 words, so that's why I wanted to show this.
Certainly that is a convincing plot.
Yeah.
Okay. So we have a question from Hans Optebeek. Could the bias towards foveal fields be related to spatial frequency distance differences in the relevant stimuli?
Yeah, that's a good question, and that's one of the follow-ups we are doing. And one of the things we are watching is that this difference of eccentricity, as we are watching exactly the same voxel, and it's radial. So it's not that we've got 2 degrees in this direction for words, and then we got 5 in the other direction. It's the same voxel. It organizes in the same, and now we are trying to test all of these tests he's saying. So controlling for the width of the bars, size of the stimuli inside of the width of the bar, in order to be able to, so for example, we didn't put it here, but we see a difference when we are using small words or big words. So I don't know if this is answering his question.
Okay, I think that'll do. I'm gonna ask you a yes or no, a one-word answer question. So you've shown us data that suggests that you're getting different PRF values for the stimuli of choice. Does that mean that we need to map for any visual area? Should we only be considering its preferred stimulus, or do you think that there's transferable information?
I don't know. We are worried about the biopia.
Do you think words are a special case?
And I don't know. That's what. And why should be different, right? Because all of these areas have been connected hierarchically, and why some it's giving us a different. We don't know. That's the question. So it's a good question.
Yeah. All right, great.
Thank
So we need to move swiftly along, but I want to thank you again. That was a great talk. Really interesting data. So our last talk of this session is going to be from Jesper Honidis. You can again correct me on your name. I apologize for that. And his presentation is titled "Decoding Color from Scalp Measured Electroencephalography."
Yeah, thanks a lot for the introduction and for putting this whole conference together and for the invitation to speak. So today I'm going to talk about how we can decode color using EEG. And to make it a bit more clear what we're going to be looking at, we're not going to be looking at just decoding green versus red, for example. But in this study, we'd like to investigate whether we can decode a whole range of different colors. So decode color more parametrically using multi-class classifiers. And in the current experiment, we're also going to be looking at multi-item displays. So we're going to be trying to decode multiple features at the same time from a visual display. And importantly, we'd like to look at visual color as opposed to kind of verbal labeling of colors. So we're mostly going to be focusing on the posterior sensory electrodes in our analysis. So before we go into the methods and the results, I would just like to point out that these results have been written up in a preprint, which is available on bioRxiv. We also put out the data on Open Science Framework. So if you would like to have a look at the data, you're more than welcome to. And also put together all the decoding functions in this Python toolbox, which you can download as well and use on your analysis if you choose to do so. So without further ado, I'd like to introduce the task. So the task that we're going to be looking at is a fairly simple one. So here we presented participants with 2 Gabor ratings. Each had an independent color and independent orientation. And what we basically tried to do is to decode all these 4 different features, so the 2 colors and the 2 orientations, in the period after the visual presentation. And at the end of the trial, participants recalled one of the features, either one of the colors or one of the orientations. And these data has been taken from a recently published task, which you can read up if you would like to do so. So we tested 30 participants and 960 trials per participants. Participants were presented with 48 distinct colors, which for our analysis, we binned into 12 different bins to increase the amount of trials per stimulus category. And we used linear discriminant analysis in our decoding. So in conventional decoding, what people usually tend to do is you take the sensors of interest. So in this case, we look at posterior sensors. So you take the 17 posterior sensors from one time point, and you try to train and test your classifier, and you get some evidence measure. And you repeat this process for every single time point until you come at the end of your trial. What we did here is something slightly differently. So we specified a window, and we concatenated all the data from that window into a single vector. So in this case, we'd have a window of 5 time points, resulting in 85 time points. And we slided this window over our data until we've reached the end of our time window. This kind of decreases the noise in the data and allows us to pick up on the difference in temporal dynamics between the stimulus categories. And we also applied a baseline correction within the window. So we demeaned our data within the window to avoid baselining issues. And for the upcoming results, yeah, as I mentioned before, we looked at the 17 posterior sensors, and we used a time window of 200 milliseconds. So that basically means that we used 20 time points to do this sliding window analysis. And well, what can we expect in our results? Well, it could be the case that after the visual presentation of the stimulus, our classifier can perfectly predict the presented color. So if we put the target color at the center, it will look something like this top figure. Could also be the case that the classifier is usually correct, but sometimes mixes up the correct stimulus class and the adjacent stimulus categories. And of course, it can also be the case that the classifier has no idea what's going on and just is at chance. So I wouldn't be giving this presentation if the latter was the case. So we actually see reliable tuning of the data towards the stimulus category after the stimulus has been presented, both for color as well as for orientation. We observe these results. And what we can do, we have this 2D tuning curve now, but to better statistically evaluate these results, we can convolve this 2D tuning curve with a cosine functions to obtain this more one-dimensional measure. And yeah, this stresses again that we can reliably decode visual color roughly 100 milliseconds after it has been presented, up until almost a second later. And these results are very similar for orientation as well. So we see very similar pattern for these two visual features. Now you can of course think that, yeah, I mean, it's nice that this classifier can pick up on these visual features, but classifiers pick up on all kinds of crazy features. So to get into this in more detail, we ran a searchlight analysis over the entire EEG topography to see where the left and right stimuli are most reliably decoded. And we actually observed that in the contralateral posterior sites, we observed most evidence for these different stimuli, which we would expect based on anatomical organization of the visual system. And we can contrast left versus right to get this contrast even more clear. So this kind of suggests that the posterior visual sensors most strongly drive this effect. And you can also think that this is nice, but maybe this is just driven by one or two colors that can be decoded really reliably. So in order to make this point even more clear, you have to demonstrate that it's not just one or two colors, but we can decode a whole range of these colors. And that is also what we show. We show that all colors can be reliably decoded in these analyses, as well as orientations, although for orientations you have this more cardinal bias that is present in the decoding analysis. And you can take this a step further and you can see how the neural correlates of different colors, how similar they are. So you can create the dissimilarity matrix for the neural signatures of all these different colors. And you can then also apply multidimensional scaling on this dissimilarity matrix. And what we then observe is that there is this fairly strong circular organization of these colors in these posterior electrodes, as well as for orientation, although for orientation it looks a bit skewed. Now, one of the final results is that even though we defined our colors from a single slice in C-lab space, it can of course be the case that after rendering our colors, that there might be some offsets generated in different features. For example, luminance is a strong one, so it could be the case that there are differences in luminance between the colors that actually drive our results. And so we measured our colors on the screen. We observed that for chromaticity, it's still very circular, but we did observe some very minute differences in the different luminances of the colors. So we wanted to test whether these differences in luminance actually explained our results. So we put together these dissimilarity matrices. So for example, the top left dissimilarity matrix kind of indicates the dark colors show that these colors have very similar luminance, and the lighter colors show they have very more dissimilar luminance. So we want to see how much this pattern is present in the data, and we compared this with kind of the measured colors. So the similarity of the measured colors here expressed as Euclidean distance between the different colors, and we compared this with the model that we used in our previous analysis. And we actually found that we could reliably decode the luminance of the different colors, but with much lower confidence than the actual color properties. And yeah, just finally, we also put together this tool where we subsample the amount of participants and the amount of trials per participant to as an aid for anyone who would like to do something similar in the future, to see how much trials or participants they would need, which you can further check out in our preprint. So just to summarize, we show that color decoding reflects the sensory qualities located in posterior electrodes, and it's possible to decode features from multi-item displays. And color decoding has a comparable magnitude to decoding of visual, stimulus orientation, and critically orientation, spatial frequency, motion. They all have spatial components, so they're more confounded by alpha power, eye movements, and visual color would have less issues with these confounds. So it might, in some cases, be a more clear measure that research can use in the future. So I'd just like to thank my supervisors, Mark Stokes, Freek van Ede, and Kia Nobre for their supervision, and all my colleagues in the labs and funders. Thank you very much.
All right, thank you, Jasper. That was a great presentation. I want to get right to the questions because we only have a few minutes here. We've got one comment that just popped up in the chat from Bevel Conway. The MDS plot for color shows a striking bias with end spectral colors, red and blue, forming the poles. Since the end spectrum colors will be consistently differently impacted by chromatic aberration, it raises the possibility that decoding is driven, at least in part, by individual differences in luminosity settings rather than hue. He does say excellent talk, by the way, which I definitely agree was an excellent talk. But do you want to give any more details about luminosity and hue?
So yeah, that's definitely a good point. And we tried to get at the issue in the final analysis where we show the contributions of luminance and different chromaticities. And of course, in this MDS plot, you can also see that indeed there is some kind of bias with different parts of the color space more clustering together. Although, and this has also been described in previous studies, but yeah, we do show that it's not, I think we try to show that it's not completely driven by these separations, so that it is more a parametric parametric distribution, which can also be seen in individual colors and in the fact that it's actually somewhat circular and that they're not even more clustered together. So it is a concern, but it's probably not.
Yeah, it'd be interesting to try to do some type of variance partitioning to see the extent to which you can explain different amounts of variance between color and hue. It'd be a different analysis approach, I guess, than the one you chose, but it would be interesting to see. So we've got a question from Paul Zer. Do you expect a similar limit in the amount of targets that can be decoded? For example, in orientation decoding, what do you think the maximum amount of color or orientated targets held in memory you would be able to decode? So how many targets do you think it's possible?
Yeah. So at the moment, it will be like 4 distributed across 2 objects. And yeah, I think once you push this a bit further, I think it might damage the classifier fairly substantially, because it probably exceeds the limits of working memory. So in some cases, you might encode one color and other cases the other. And then if you train and test your classifier, you might also include training data that does not reliably represent the colors and orientation. So I think 4 features is already pushing it a little bit. I would expect that it does significantly drop off if you include more items, but that would be an interesting future path, I think.
Cool. And then Miguel Santos says, what are the neural mechanisms underlying the color decoding? And why did you choose posterior electrodes?
We chose posterior electrodes because we're mostly interested in the visual aspects, so less so than just a verbal labeling of red, green, blue. And
we are just on time.
Oh, great.
Great. I just started the live stream. We can go live whenever you're ready.
Okay. So the first speaker is missing?
Yes. Mm-hmm.
Okay.
We might be able to let them in from attendees, but if not, I guess Samuel might want to go first.
Okay, sure. Let's.
Okay, so I'll start. I'll go off screen. I'll be in the chat if you need be. Okay, then have fun. Take care.
Thank you.
Okay. Okay.
Oh, here's the first speaker.
Oh, so our first speaker just joined. Great.
Oh, hello. Sorry about that.
No worries. We'll just start in a moment. All right. So just a couple of house rules. So let's try to be on time as far as possible. And around 13 minutes into your talk, I'll wave this red pencil if you can all see it. And this is just to tell you that there's about 2 minutes left, so you can wind up. And we'll have a bit of time for a couple of questions. Is that okay with you? Okay. Okay, good. Right. Okay, so let's then start the session. Welcome all to yet another exciting session of NeuroMatch. So we'll have a series of 3 talks in this session, and we'll begin with a talk on the neural basis of population codes by Blake Bordelon, who's a PhD student in applied math in Cengiz Pehlevan's group at Harvard University. So Blake, the floor is yours, and let's hear about your exciting work.
Right. Hello, everyone. All right, thank you for the introduction, Srikant. So let me just pull this up. So yeah, so today I'm basically going to talk about a notion of inductive bias. So this is kind of a learning theory concept, and I want to think about inductive bias in neural population codes. So how do neural representations influence how learning happens, how learning of downstream tasks happens. So what is inductive bias? So inductive bias is sort of a notion from machine learning or from actually just philosophy really about how you have to basically incorporate priors about the world that guide predictions when the data is insufficient to sort of generalize a rule from a finite number of observations. So for example, I know it's early, but imagine I give you the following sequence of numbers and I ask you like, what number do you think comes next? So maybe if you want, I know it's early, but if you want to, you can maybe guess what number comes next. or what numbers you think comes next. I'll give you maybe like 20 or 30 seconds. So let me see. Are there any guesses? I actually can't see the chat. So one thing you could think, like kind of the natural thing, what you might guess is, okay, each time the numbers are just doubling. So maybe this sequence is just blowing up exponentially. So the next number should be 16 and then 32 and so on. But that's not the only sequence. You could say going from 2 to 4, I add 2. Going from 4 to 8, I added 4. The next number I could add 6, and then I could add 8 and so on. That'll actually give you a quadratic polynomial. So the next number would be 14, then 22, so on. And then, also there's nothing to say that after this 8, I don't just have this like infinite sequence of 8s following the first one. So the whole idea of this is that there's, from this finite number of observations, I can't really generalize a rule. This is the problem of induction. And the choice of what sequence you would guess to make your prediction, it's driven by inductive bias in this case, since the data doesn't uniquely specify the solution. Okay, now how does this, so we're all neuroscientists, we're interested in the brain. What does this have to do with neuroscience? The brain has to basically learn and extract patterns, based on experience with the world. However, a lot of these problems are ill-posed, and how do neural representations, sort of innate or learned neural representations, influence how the brain learns new tasks when data is scarce? So, another way of saying this, using the language that I was just introducing, what are the inductive bias of neural population codes? Okay, so how could you extract this? So basically in this talk, I'm going to focus on a data set that was generated in this paper by these authors. And it's basically a collection of mouse V1 responses to static gratings oriented at different angles. And so what we want is we basically want to record a bunch of angle response observations, find all these tuning curves. So this collection of tuning curves are the average responses of all the neurons to these different orientations. This is going to characterize the population code. And from this, we're going to ask, how well would the code perform when it learns new, learns new tasks, maybe an orientation discrimination task or something. And from this, we're basically going to say, that the tasks that the code can learn easily, those are the things that the code has an inductive bias for. And by easily, I mean from a small number of examples. Okay, so here's a simple sort of example of showing what I'm imagining. So remember I said we had static gratings oriented at different angles. An easy task may involve discriminating between all the angles on the right and all the angles on the left. So you can think about this as maybe a low frequency orientation discrimination task. A harder task may involve, you want to maybe produce an output +1 for all these green regions over here and produce maybe output -1 for all these other ones. And so these Ys might be like some behavioral variable that you want the animal to produce, or some prediction that a neuron makes downstream from the population that you're recording from. And the question is basically I sample a bunch of points on these rings with a bunch of the corresponding labels. And I want to know how many of such observations are required to attain good generalization performance. So naively, you'd say, okay, this looks easier and this looks harder. And so actually, you can take the recordings and just train like linear regression models on the recordings, and you get basically these dots here. And as you'd expect, the low frequency tasks, it's easier to learn as the sample size P increases than the hard one. And what's interesting is in this talk, we're basically going to discuss a theory which generates these dashed lines that agrees with these experimental predictions. So the theory basically takes in information about the population code, just all the tuning curve information about the task you're trying to learn, and then predicts like how hard it is, basically, what your expected generalization error would be. And that actually, we're going to talk about how that is a notion of inductive bias for population codes. And this is just a simple example. But so in the model, so I'm imagining a fixed population code. So the responses of the neurons are maybe R1, R2, R3, and so on. And I'm learning a collection of readout weights W so that I can approximate this function Y. This is some target function. But the whole idea is that I've only given you a sample of P examples. So you don't have the full distribution, but you've seen P examples. And the question is, if you have a good inductive bias, you're able to basically, when you optimize this training error, this error on this p examples, you actually would hope that you're matching y on the whole distribution. So the real quantity of interest, generalization error, measures error on the entire stimulus distribution. So all the possible stimuli that you could have seen, p of x, and so the problem is how does the generalization error in like this quantity, this population loss, how does this depend on the code r of x and on the number of samples that I've seen when I did this sort of linear regression? And codes that, so in this talk, codes that generalize well for small numbers of examples, so codes that allow like learning from few examples or few shot learning or whatever, they have good inductive bias for those sets of tasks. Okay, and so what's actually nice about this setting is you can make a concrete mathematical connection to something that's been studied in machine learning. And this is called basically kernel regression or kernel methods. And so you can show basically that the learned function that you're going to end up with when you do this procedure of learning the weights, it's only going to depend on this object that's called the inner product kernel, or the in neuroscience you may be more familiar with this as a similarity matrix. But what it is it basically says take the population response to input 1, maybe like the response to a picture of a giraffe, and then take the response to a picture of a zebra, and then just take a dot product of those responses. And that gives me some notion of how similarly the population encodes those two different stimuli. Okay, and so why does the solution only depend on this inner product structure of the code? It's actually because suppose I minimize this loss function on that I mentioned, this training loss on this finite data set of p examples. And suppose I use the delta rule, for instance. So if you studied like reinforcement learning, for instance, this is pretty common or whatever. So what's interesting is that if I initialize w at 0, this is always just a number. So w is always basically going to be in the span of these neural responses, these neural response vectors for these p points, right? So w I could always write as some linear combination of these r of xi's, these responses to the different points in my data set. And then if you go back and say, okay, well, my function was w dotted in like dot product with r, you realize that, okay, the solution is just always going to be a linear combination of this function k evaluated at points in the data set and on a test point. So this is called the representative theorem if you've studied this in machine learning. And so, outside of the context of neuroscience, we, me, Abdul Janitar, and Cengiz Pehlivan have sort of studied this independently, kind of from a machine learning perspective. So the whole idea of these papers was basically to analyze what is this sort of average case performance of kernel regression as a function of the number of samples. And can we extract like what the inductive bias of this learning algorithm is? And so the kinds of things that this theory can predict. So for instance, imagine I take MNIST, and then which is like a data set of handwritten digits. And then I basically want to predict if I train on a subset of all the training points in the data set and then tested, what would be my expected sort of generalization error. So that generates this sort of solid line here. The experimental kernel regression solutions agree pretty well with it. And then you can also, I'm not going to get into all of this, but there are more complicated sort of things that you can realize. When the data is noisy, you can have these like multiple descents and everything. The learning curves can be non-monotonic. But if you're interested in this stuff, please check out these papers. But anyways, from a neuroscience perspective, the theory that I was just alluding to, it tells you that the relevant quantities that you're interested in, they can be extracted basically from the SVD of the neural responses. So imagine I have a matrix that's number of neurons by number of total stimuli, okay? And I do SVD on these responses. So this matrix would basically look like a collection of tuning curves over here. When I do SVD, the sort of left singular vectors are going to correspond to what I think in neuroscience we typically think of as, we think of as the principal components of the covariance matrix. So each of these vectors is orthogonal to the other, but U1 basically points in the direction of maximal variance, U2 in the direction of maximal variance in the subspace that's orthogonal to U1, and then so on and so on. So the cartoon picture is I have maybe a lot of neurons, but I have these other vectors that are potentially telling me that the data may lie in a lower dimensional subspace. And And lambda 1 tells me about how much variation there is along each of these directions. Lambda 2 tells me how much variation there is along the second direction, and so on. So lambda is, as I was just mentioning, it's basically the variance along each of these directions. And then phi, so we don't study phi as often, but phi are going to be these are the kernel eigenfunctions. These are the kernel eigenvectors. And these are the objects that are actually relevant to the theory that I'm mentioning. So the principal components are like maybe tell you about the structure of the population code. They tell you about the structure of like the geometry of responses. The eigenvalues tell you about, you know, how much variance so eigenvalues also tell you about the structure because they tell you about like the shape of this sort of elliptical approximation of the neural response cloud. And then these eigenvalues also in the theory of inductive bias, they're going to tell you about what eigenfunction, they're going to give you an ordering on which of these eigenfunctions are easiest to learn. So I'll explain that really quickly. So the theory is a bit complicated, but what's nice is that these eigenfunctions, okay. So basically, so let me just fast forward. So let's go back to this task really quickly that we were discussing. So in this example, all you have to ask is how does this easy task and how does this hard task decompose in the eigenbasis of the kernel? And then you could ask you could see basically that the easy task is easier to learn because there's a greater proportion of variance in the top kernel principal components. You can apply this to MNIST. Yeah, okay, so I just want to thank everyone. So, and then maybe I can take questions if there's more.
Thanks, Blake, for the interesting talk. I'm sorry, but in the interest of time, we have to move on.
Okay.
There's potentially time for one very quick question. Are there any questions for Blake? No? Well, I guess I could ask you one very quick question. So what are the implications of this study? So what are your main conclusions?
Yeah, so let me I think the main thing is actually
well, I guess it would just be easier if you sum up in a couple of sentences.
Yeah, so basically it's that you can there's a function space interpretation of what population codes are encoding, and this actually just doing singular value decomposition gives you an ordering over which kinds of tasks are easy. So the kind of eigenfunctions that you would get if you did SVD with largest eigenvalues are easiest to approximate. And so tasks that have good alignment with these these basically large eigenvalues, eigenfunctions are easy to learn. So this basically gives you a notion of inductive bias and sample efficient learning.
Okay. All right. So thanks a lot. Okay. So my Tissot watch tells me we are a minute late, so it's not all that bad. Anyway, thanks again, Blake, for the great talk. It's time to move on to the next speaker of the session. So the next speaker is Sam Failer, who's a senior research associate in the famous Cortex lab at UCL, headed by Ken Harris and Matteo. And we'll probably listen to some more exciting stuff in the visual cortex from Sam. So Sam, the floor is yours.
Great, thank you. Let me just share my screen.
We can't hear you very well.
Oh.
You sound like you're inside a well.
Can you hear me better now?
There's a bit of an echo.
Is this okay?
Maybe you could move a little closer to your laptop or to your mic or something.
Is that better?
Maybe just a bit.
Okay, let me change the, let me just try something really quick.
Sorry. Yeah, I think this sounds a bit better. So let's just move on.
Okay.
Yeah. Okay, great.
Great. Thanks for introducing me. Today I'll be talking about some findings we had in primary visual cortex following the learning of orientation comparison task. So essentially what we find is that with the learning of the task, we find that the task stimuli and are orthogonalized in visual cortex. So I'll go through those findings now. So just as I'm sure this is maybe a little basic for the crowd currently viewing, but we can summarize activity in the visual cortex as a population code vector. And I think Blake kind of touched on this a bit in his talk. In primary visual cortex, we can see how population encodes change with change in the visual stimulus. So in this case, we have a tree has specific visual features that excites a subset of cells, and that the firing rate of those neurons can be represented as a population encode vector in some n-dimensional space. And what's nice about this idea is that this allows the brain to easily distinguish objects with very different visual features. So in this case, we have a tree and a goldfish. Very different subsets of cells would be active for these different stimuli. And so in this n-dimensional space, these population vectors would be very orthogonal to each other. So the similarity, as Blake mentioned, quantified as this dot product between the two vectors would be close to 0 or orthogonal. What it also allows for is a generalization of visual stimuli. So in this case, we have two trees that have very similar visual features. So the population code will be slightly overlapping. And so the population code vectors will be more, the angle between those vectors will be narrower, and the dot product will be closer to 1. So what we wanted to ask was, how can learning affect these population code vectors in visual cortex? Can you take two stimuli that are in some ways, a lot of ways, very similar to each other, and through the process of learning, orthogonalize those vectors so that the brain is better at differentiating these two stimuli? Likely because in terms of a task, the behavioral consequence of these stimuli could be very different. So it makes sense for the brain to differentiate the meaning of these stimuli in the context of a task. So the questions we want to ask are, does learning increase orthogonality of population codes for task stimuli? And if it does, are these effects due to changes in bottom-up connectivity? Is it purely like a Hebbian-like process that changes the thalamocortical inputs, or is there some other kind of top-down neuromodulatory processes involved in this effect? So to answer this question, we trained mice on an orientation comparison task. So essentially, this task involved a mouse sitting on a stage with its 4 paws on a wheel, and it would turn the wheel to move the stimuli left or right. And what was shown to the mouse were 3 different orientations, one on each side screen, and the mouse had to move the stimulus closest to 45 degrees to the center screen. So in this case, 45 is correct. In this case, 68 degrees would be correct. And if you distill this task down to its very basics, essentially you have 2 types of stimuli. You have the informative stimuli, which is 45 and 90, which is shown, one or the other or both are shown in every trial. And then you have a distractor stimulus, which is 68, which really doesn't tell the mouse, give it any additional information about how it should make a choice on that task. So to assess the impacts of learning in V1 after mice learned this task, we did 2-photon calcium imaging in layer 2-3 across several layers, and recorded from between 3,000 and 7,000 neurons per experiment. And the experimental protocol is very simple. We basically just imaged mice in the naive condition before they had done any trials in the task, and showed full field drifting gratings to measure orientation responses. We trained the mice on the task, and then once they reached a proficient level of performance, which in our case was over 70% correct, we did full field drifting gratings after a training session, and looked at changes in orientation tuning. So these are some polar histograms of responses to different orientations in both naive and proficient mice. And so this is an example of a cell with a mean. So let me say one thing is that we quantify orientation preference in 2 ways. One way was looking at just the maximum, the modal, basically the mode of this polar histogram, which is like the maximal response. We also took a complex vector of every response and took the average of that to give us a one vector, whose angle was the preferred orientation of that cell, and the magnitude was its level of selectivity. So here we see in the naive condition, these cells, the mode and the mean of the responses is very similar. What we find in the proficient condition is a really odd thing where lots of these cells have bimodal tuning curves where the mode and the mean are actually not aligned. And we see these kind of dips in the tuning curves at the informative task orientation, so 45 and 90, even for neurons that prefer the task orientation, so in this case, 45. So this is really odd. We wanted to look at this in more detail. So first we looked at changes in the proportion of neurons preferring the informative orientations in the task as sorted by their modal preferred orientation, so the maximum response. And what we found was that there's actually a decrease in the number of neurons that prefer the informative task orientations, 45 and 90. And when we look at these neurons as a function of their selectivity level, we find that this is primarily its reduction in the number of cells is due to a reduction in these cells with low selectivity for the informative orientations, suggesting that maybe that what we're seeing is this kind of global, like population level suppression of activity to these informative orientations. And so lastly, we did to really quantify these effects on single cell tuning curves. We average the cells by their preferred orientation and their selectivity level. So these are average tuning curves of neurons, color coded by the preferred orientation based on the mean of the responses, response vectors. And we sort each column is a different level of selectivity. So we go from low selectivity to very high selectivity. We have a nice peaky, narrow tuning curves. And what we see in the proficient, which is consistent with what we saw in the single cell tuning curves, is that we see this large suppression to these informative orientations in these cells that have low selectivity, regardless of their orientation preference. We also see a asymmetry in tuning curves for neurons that prefer orientations near the informative orientations. So this is really kind of confusing. At a single cell level, things look kind of messy, but when we start looking at, we decided to look next at the population level, and things become a bit more clear here, is that generally what we see is essentially is a sparsing of the responses to these informative orientations. So this is we call these bullseye plots, where we plot cells based on their mean response. The color is their response to the 45 and 90. So if they have a max, basically only respond to the 45, they're green. If they only respond to the 90, they're magenta. And if they respond to both, there's some a gray or a black if they have a maximal response to both. And their position on this polar plot, or this bullseye plot, is their orientation preference, and their distance from the origin is their selectivity level. And so what you can see is that just by eye is that there's a large sparsing of the responses in the proficient mice for these informative orientations, and that you have a reduction in number of cells with large responses to these informative orientations, particularly for these neurons that have low selectivity. If you move out to the rim of the plots, you see there's not really much of a difference in the level of response to these task orientations. And we quantified this just using kurtosis measure. So this is a common measure of population sparseness. We took the mean responses to these task orientations for all the orientations we show actually in the passive condition. And we see that the largest increase in sparseness occurs for these informative orientations in the task. So as I kind of hinted about in the beginning, though, one thing is that these population codes, when you have less co-activity, what you expect to see is an orthogonalization of these population codes. So in the naive condition, you have lots of cells that are active and lots of cells that respond to both stimuli. So you expect that their population code vectors to be less orthogonal, like very close to each other. And what we expect to see in the proficient condition is that we should see this orthogonalization of these population codes as a consequence of the sparsing of the task, these informative task stimuli, responses to these informative task stimuli. And so to quantify this, we averaged trial responses to each orientation shown in the drifting grading experiments and on one half of the trials, and we took the mean response on the second half. And we looked at the cosine similarity, which essentially is the just dot product between those population code vectors. And we find that there's a decrease in cosine similarity between the task orientations and these non-task orientations. In particular, we see the largest amount of decrease in cosine similarity between the informative task orientations, so 45 and 90, supporting the idea that we're seeing this orthogonalization of the cortical population code for these informative task orientations. And again, this is just data from every mouse. So we see that there's a significant difference in the level, a decrease in the cosine similarity when we compare the 45 and 90 versus the orthogonal orientations, which we were using as a control after training. So it seems to be, again, this seems to be really specific to these, or at least we see the biggest effect for these informative task orientations. So lastly, what we want to do is we want to try to come up with a mechanistic model for this sparsing effect we see as a function after this learning. And what we settled on was this modulation of FI curves. So the idea is that with learning of the task, you get more FI curve modulation specifically for these informative stimuli. And so essentially cells that fall in this kind of middle region in this FI curve are those with low selectivity levels. And we can very easily model this with a simple piecewise linear function, which can very successfully transform the naive responses to proficient responses. And the convexity of this function basically is related to the level of sparsity you see with the response to these stimuli. So we're able to fit a stimulus-specific function for every, as a function with training, and it fits all the neurons in the population, regardless of their preference or selectivity level. And I'll just end right now. This is my last slide. We find that if you use this function to transform, we're able to transform the naive data quite successfully using these piecewise linear functions, where we can sparsify them by just taking the responses and transforming them. And we see that we can recapitulate a lot of these effects on the tuning curves that we see in the proficient conditions. So we see the suppression in the the informative orientations, and we see asymmetric effects in the tuning curves of the cells with orientation preference near the task orientations. So to summarize, we see these learning and orientation comparison tests as these multifarious effects on tuning curves. It's well explained by a sparsing of the population response, and results in this orthogonalization of these informative task orientations. So I just want to thank my advisors, Matteo and Kenneth, and everyone else in the Cortex Lab. And I just have time maybe for one question. Thank you.
Thanks a lot, Sam, for the cool talk. We have time for maybe one question, one quick question. Anyone? That illuminating question? No? Well, I have a quick question. So maybe I missed this, but did you target any specific cell type populations in your 2-photon experiments?
Yeah, sorry, I should have mentioned this is purely excitatory cells. So these are cells expressing GM6S and etc. Pyramidal cells only.
Okay. Well, the reason I ask is there's some previous literature from Rigan Kassouf's lab that tends to suggest that there's some kind of a role in dendritic architecture in Orient's response selectivity. So I was just wondering how some of your orthogonalization results and findings could change if you were to record from distinct populations of interneurons which have different dendritic architectures.
Yeah, I think that's something we're kind of looking into, because we have we're developing new methods in the lab where we can record and we can identify inhibitory neurons after our recording. So I think that would be the next step, is to try to figure out what the kind of focus in on the biological mechanism behind the sparsing, see what subclasses are actually playing a role in this effect. Yeah. But I think based on our modeling, we're pretty convinced that's not a modification in these bottom-up cortical projections. It seems to be something that's happening in a more dynamic way. And actually, I can show a slide real quick. We find that this sparsing is dynamic. So it varies on a trial-by-trial basis, which supports the idea that this is something that's happening through modulatory effects, not change in the tuning curves of these neurons.
Right. Okay, thanks a lot. I think it's time to move on to the final talk of the session. Sorry if we couldn't take more questions. I guess you're always free to write to Sam or Blake with your specific questions, but we just have to move on in the interest of time. So our final speaker of the session is Shanshan Kin. So we move back from London to Boston now, again to Cengiz Pehlevan's lab. So Shanshan is a postdoc in Cengiz lab, and he'll talk to us about a network model of noisy representation learning. So Shanshan, the floor is yours.
Thank you for the introduction. Can you see my screen and the cursor? Cool. Hi, everyone. Thank you for being here. Today, I'm going to share with you some of our recent work on the dynamics of trypsin receptor fields during noisy representation learning. So in the conventional view that our long-term memory or learned behaviors are associated with stable internal neuronal representations or neural assembly. However, this idea has been challenged by recent long-term recordings. For example, where a mouse is exploring the same familiar environment for many days, and then the same one neurons in the hippocampus was recorded, and then you can see the place fields of the C1 neurons continue to change at a time scale of weeks. Similarly, in these teammates visual sensory motor task, where mice were trained to make an association between this visual cue at the first stem of the T-stem, teammates, and the left or right turn. After learning, mice can perform this task very well for many days. However, the brain region, posterior parietal cortex, which is essential for this task, and then the researchers measured the population activity in the posterior parietal cortex neurons, and they found that neural activity continues reconfigurate and at a time scale of 1 or 2 weeks. So they call this phenomena as reputational drift. Similar phenomena has been observed in other brain regions like motor cortex, and even sensory cortices like olfactory cortex and the visual cortex. So this experiment raises several important questions. First, you may wonder what's the underlying mechanism behind this reputational drift. In this talk, I will show that drift is largely due to the noise during this learning process, especially the noisy synaptic update or turnover. Second, what is the underlying dynamics of the drift, right? Is it purely stochastic or not? So in this talk, I will show that the drift actually has some structure. It preserves the pairwise reputational similarity. Another question which is important, where I want to be able to cover, is that how could animals to achieve robust behavior despite these drifting representations? We will focus on a class of systems where neurons have localized receptor field. So in the first slide, I mentioned the place cells in C1 region hippocampus and also the posterior parietal cortex, where neurons have position tuning. These are just 2 examples. In fact, localized receptor field is very ubiquitous in among other brain regions. For example, in neurons at some auditory cortex, they have localized receptor field. Basically, they are tuned to the 2D space defined by this inter-oral time difference and intra-oral time difference to form this auditory map to guide the animal to locate the sound source. The localized receptor field can be applied even to more abstract space. For example, in this research, they found that neurons in hippocampus may develop this localized receptor field at this space defined by these different cognitive variables or behavior variables. Shown here are 4 example neurons. They have the localized receptor field along this manifold, on this manifold. Then we will use a minimum model to learn this localized receptor field and then explore the drifting dynamics of this localized receptive field. Previous studies have shown that a class of network called Hibin anti-Hibin network, illustrated in this figure, they can learn this localized receptor field. So the idea is that this is just one layer network. It receives forward input from the previous layer and also a natural inhibitory interaction due to competition. After learning, this network can learn this localized receptor field. The representation is just determined by this neural dynamics, where you can think about this UI at the member potential of this neuron. It receives forward input and this natural inhibitory input, and also we include this bias term. Now the firing rate, which is yi, can be regarded as a rectified linear function of this member potential with some threshold and a normalization factor. So this learning process is purely unsupervised. Basically, you update this forward matrix by this classical Hibin learning rule, where we call this updated rule for natural matrix M as anti-Hibin because there is a minus sign in this neural dynamics. So as I mentioned in the previous slide, that our hypothesis that this drifting dynamics is probably largely due to this noise during this learning process. So we were adding this noise in this learning rule to see how exactly this learned localized receptor field evolve with time. More specifically, we were using the Hibin anti-Hibin network to model the place cells in hippocampus and also the neurons in posterior parietal cortex. First, let's look at these place cells. So how do we learn this place field, right? One idea is that C1 regions in hippocampus receives input from grid cells in entorhinal cortex, and also they form these mutual competitions. So this just resembles our Hibin and Hibin networks. So how do we model this system? Well, basically we're model a 2D environment. So each position in this environment has two coordinates, which can be represented by the a large population gray cells. But these gray cells have different spacings, orientations, and also offset. So our input is for any position in this space, just a vector of the gray cells. And we'll use Hibin network to learn the localized receptor field. Indeed, after training, you can see the neurons develop this place field like tuning curves, although some of them are silent. If you put the receptor field in the original space, you can see all these neurons have receptor field that tire the 2D space. Next, I will show you a video where you can see 10 example neurons, how their receptor field evolve with time when we add in noise to add the update. So you can see these are the position of peak of the place field. You can see some neurons are very stable, for example, for number 10, where others are more variable. They jump around, disappear, reemerge, maybe at different positions. So this jumping behavior or drifting behavior have some consequences. For example, if you look at the repetitions of positions at the upper layer, so basically for any given position, I can see what's the repetition at the place cell level. So it's a vector, we call it population vector. And then due to this drift behavior, you can see the population vector also evolve with time. So we can quantify this by calculate this pairwise Pearson correlation of population vector compared with 10.0. What you can see is that this correlation vector coefficient of the Pearson correlation gradually decays to 0. So, but what's interesting here is that at any given time, even though the neurons disappear or reemerge, you can see there are fixed fraction of neurons that have active place field. What's more interesting here is that even though the population vector of position representation continue to evolve, the pairwise representational similarity, just defined as a dot product of the two vectors, you can see the similarity matrix at two distinct time points are essentially the same. That means population vector drift with time, while similarity matrix is stable across time. We can also look at the evolve of individual place field, right? So as shown in this video, this place field may disappear and reemerge. So this is a typical curve showing the peak amplitude of one place cells. You can see they have active period and silent period. Once this neuron have active place field, it tend to have a relatively stable amplitude. So we are interested in the time intervals during the simulation that these neurons are active. We're also interested in what the relative shape of this peak position of their receptive fields. So in the simulation, we find that this fraction of active time period is strongly correlated with the average peak at values. Similarly, we found that the relative shape of the position of a receptive field is anti-correlated with the peak amplitude. That means neurons that have strong receptive field tend to be more stable because it has a relative smaller shape of receptive field. So similarly, we can use this Hibin and Hibin network to model this posterior parietal cortex neurons. So this is the task, sensory cue guided binary choice, as I mentioned earlier. So after learning, we see that these 400 output neurons develop this localized receptive field. So this horizontal axis is the position along this T maze, and each of them have like a localized receptive field. When you order their peak position or sort them, you can see half of them have this tuning that can tire this position along T maze for the left turn task. Similarly, the rest of them will have localized receptor field that tire the teammates for the right turn task. So in the experiment, as I mentioned in the first slides, the population activity continued to change over time. Similarly, in our simulation, we found that neuron populations continue to evolve with time. Although at any given time, you can find maybe slightly different subset of neurons to, by reorder them, they can still represent this task as similar in the experiment. But yeah, again, we find that the representational similarity is preserved. And we also find this shift of receptive fields. So at similar experiments, that larger shift is less frequent than the smaller shift. This is similar in our model. With that, I would like to wrap up. So I showed that this Hebbian and Hebbian network can learn a localized receptor field with noise. It can recapitulate many of the drifting behavior observed in experiments. We found that even though population vector or representation continue to evolve with time, but the representation similarity is preserved. We also found that neurons with stronger receptor field tend to be more stable. With that, I would like to thank my supervisor, Jingis, and also all the other members in Pelivan's group and the funding source. And I'm happy to take any questions if you have.
Thanks a lot, Shanshan, for the interesting talk. I think we have time for one question. Any questions? No questions? Well, I just had a quick question. So in your plasticity implementation, so did you consider having specific rules for excitatory and inhibitory populations, or do they all pretty much follow the same rule?
Yeah, so here we didn't distinguish this sort of inhibitory neuron or excitatory neuron, right? So because this is a very simple network where you have only one layer of neurons. So because this W can be, either positive or negative, so we didn't distinguish them. So yeah.
Okay.
But in principle, you can add a interneurons. So basically, you can implement by adding a large group of interneurons. So now you receive input from the principal cells and feedback to inhibit these principal cells. So that you will get something similar. Yeah.
Okay. More questions? There's one more question. So in
there is a one in the Q&A.
Yes.
So the question is, any insight on why the neurons with stronger receptor field persist? Yeah, so as I mentioned here, right? So the reason that the learning rule is essentially competitions. So basically, neurons compete for the input that from the similar input regions. So if one happens to have stronger receptor fields, even though when you modify this width matrix slightly, so maybe it's beyond 0, right? Because we are using rectified linear functions. So you can see those that receive more input, that have stronger amplitudes, will not be affected when this noise to push your width matrix cross 0 or slightly less than 0. So that is one of the explanations of why neurons have stronger receptor field tend to be more stable.
Okay, so I think it's really time to wrap up, or we'll be kicked out from the Zoom room. So thanks again to all the speakers. This is not an easy format to present and discuss, so your efforts are particularly appreciated. So let's also take a moment to thank Arash, who provided all the great background support. So Arash, could you please turn on your camera and show yourself?
Hi, everyone. Thank you for your participation. It was my pleasure to listen to your talks and see your interactions. It was great talk. Thank you for everything. And with that, we would like to end the meeting for the next one. Thank you. Have a good one.
Thanks a lot and stay safe. Thanks. Bye.
We have 3 minutes. Okay, good.
So do you guys have to let people in then, or?
Yes.
We just start webinar at time, right? Then everybody will be able to join. By the way, I'm starting the live stream. So this is the link. Now we are on YouTube.
Oh, we are already online? Okay.
Right.
But we will do the introduction once the seminar starts, right?
Mm-hmm. And possibly slowly until attendees fully join, because it takes them like 30 seconds to set up their audio and settings.
Right. Okay. No problem. So then why do we start live streaming before?
Just to double check nothing's going on, because it takes some time, and if it goes wrong, we will miss from the live stream.
I see.
Like if
So
it breaks, then
can you sing for us then? Maybe this is your chance. You know, on YouTube, you could like become a viral sensation. or do like a TikTok dance.
Yeah, right. Actually, today it's we're celebrating Halloween, right? So we're supposed to be dressed up, and I see you have is that a background with a
it's my green screen.
Yeah, the Grim Reaper.
I was too shy for that.
So we have 30 seconds, but if you're ready, we could start, and I'll go off screen. So I'm starting now. Have fun. Take care.
Okay.
Okay. We are live. People are starting to come in. So, welcome everyone. It is a great honor to introduce to you today Erin Schumann. Erin is the director of the Max Planck Institute for Brain Research. He got her bachelor's degree in psychology in 1985 at the University of Southern California. That's my own school as well. A PhD in neuroscience from Princeton University, and then she did her postdoctoral research in the lab of Daniel Madison at Stanford University. While at the same time, soon after, she was recruited by the California Institute of Technology, where she started as an assistant professor and made it all the way up to full professor. At the same time, she was appointed at the Howard Hughes Medical Institute as an investigator. In 2009, she returned back to Europe as a director of Max Planck Institute for Brain Research in Frankfurt, in Germany, where she holds her current position. Her research interests focus on understanding the properties of mRNAs and proteins, for example, the transcriptomes and proteomes distributed throughout the neuron. She has examined how stability in neuronal processes can be brought about by local cell biological processes like protein synthesis, allowing synapses to respond rapidly and appropriately to changing stimuli. She has made several groundbreaking discoveries in this field. For example, her lab was the first to demonstrate the functional role of local translation in neurons. She also made a lot of technical contributions to the field, such as the development of the bone cut and fan cut techniques, which enable the labeling, purification, identification, and visualization of newly synthesized proteins in neurons and other cells. Last but not least, Erin is very well known for her efforts to promote the professional advancement of women in the field of neuroscience, and we are grateful to her for that. She has made it a condition of her recruitment to Max Planck that a new childcare facility be built on campus. And she also spearheaded an initiative outlining changes in the recruitment strategies of the Max Planck Institute, aiming at doubling the percentage of female directors by the year 2020 by 20%. For those and all of her science contributions, Erin was recently awarded by the Society for Neuroscience the Mika Selpeter Lifetime Achievement Award for her teaching, mentoring, and advocacy efforts. And also in 2020, she won the ALPA FKNE Diversity Prize for her contributions to advancing gender equality in brain research. So I'm especially happy to have Erin here together with us. So today she will tell us about exciting new research here that is not published. So Erin, the stage is all yours. Welcome.
Thank you very much, Jota. And thank you to all of you for tuning in. The story I'm going to tell today is really kind of a stealth project in the lab that represents a longstanding interest that we haven't really spent as much time as we could have on, which has to do with how animals respond to changes in their environment. And the operatives in this stealth endeavor, the main driver of this project is a senior graduate student, Lucas Anesser. And he was helped by a master's student, Ivan Alcantara, a lot in the very beginning of the project. And then when Ivan left to go start his PhD program in the US, Anya Gemmer, also a graduate student in the lab, and Christina Merkis, a master's student in the lab, joined the project. And throughout the project, we've enjoyed the collaboration with Sujin Ryu, who's a faculty member at Exeter. So what I'm going to tell you about today is some work that we've done analyzing how zebrafish respond to social isolation. So many animals are social beings, and there are advantages conferred to being social, having to do with avoiding predators, gaining access to resources like mates or food. And zebrafish are social beings like many other fish. And in addition, for many social beings, there are, it's very clear that there are downsides to not being social. For example, social isolation can cause long-lasting changes in the behaviors of animals. And the brain systems that respond to social isolation are not very well understood. So this is what we strove to understand using the zebrafish as a simple system. So the basic question we asked is, are there genetic modules that are associated with changes in the social environment? And to do this, we started with a very simple experiment, which was to compare the brain transcriptome of animals, zebrafish larvae, that were raised in isolation, and compared that to zebrafish larvae that were raised with conspecifics, so raised in an isolated environment or raised in a social environment. And practically speaking, the experiment looked like this. We took zebrafish more or less from birth, and then from birth until the time where we extracted the RNA, they were raised in either an isolated or a social condition. And we had experiments, groups of experiments where animals were isolated from birth until 5 days, birth until 8 days, until 14 days, or until 21 days. And then immediately at the end of this experiment, we extracted the brain, we isolated RNA, and then we did RNA sequencing. To try and the goal of this was obviously to try and understand whether there were any changes in gene expression in the brains of these animals as a result of being isolated. So when we looked at these various time points, we identified a variety of differentially expressed genes. And what I'm showing here is actually the sum of genes that were either up or down regulated at these different time points. So we had some 300 and something unique genes that were regulated. And this was at the beginning of Lucas's PhD thesis. And so obviously there are lots of ways to go. And one way would be to look for gene regulatory networks, to look for pathways. But what Lucas I think the next slide will show that he was actually quite lucky, because when he superimposed the genes that were regulated across all of these time points, the scenario that seemed very complicated, and probably still is quite complicated, became a lot simpler, because there were only 4 genes that were consistently regulated across all those time points. And these genes, these 4 genes, were actually down-regulated in the isolated fish relative to their social siblings, socially weird siblings. And when we took a look at this list, the first 3 genes here are actually immediate early genes. So these are genes that are activity-dependent genes that are turned up when neurons or animals experience activity. FOS, for example, is one that is used quite a lot to tag active neurons in behavioral scenarios. So these 3 genes, the fact that they were down-regulated in isolation made sense, because in fact, one might imagine that one of the features of being isolated is a reduction in activity. So we focused our energy on this last gene here called PTH2, which is a gene that is parathyroid hormone 2. So PTH2 was originally identified actually in a large screen. There is a receptor for PTH2, which is the PTH2 receptor. And this receptor is expressed in the periphery and also in the brain. But in the brain, the natural ligand for the receptor, which would have been PTH, is very, very low. And so Ted Usden, who is at the NIH, set out to try and find the ligand for the PTH2 receptor that was expressed in the brain. And for those of you who feel rather weary in doing any kind of microdissection, the Usden team purified and sequenced this ligand, which turned out to be PTH2, from bovine, from cow hypothalamus. And their starting material was 50 pounds of bovine hypothalamus. And so from that starting material, they were able to identify a ligand or peptide that they called at the time tuberoinfundibular peptide of 39 residues. So it's 39 amino acids long. It's expressed in vertebrates, so it's not expressed in invertebrates. And in rodent brain, the PTH2, as it is now known, cell bodies are in 3 main areas: in the periventricular gray, in the posterior intralaminar complex of the thalamus, or the PIL, and in the medial paralemniscal nucleus. And the projections to these areas are actually reciprocal. So they come from hypothalamic areas, from limbic areas, including the amygdala, from the superior colliculus, as well as the inferior colliculus, from the dorsal horn, the spinal cord, and some primary sensory areas. So we focused our energies now, and I'll describe for you the experiments we did to explore PTH2's reactions to the social environment. So the next experiment that we did was to validate the next generation sequencing experiments with qPCR. And so we did that again by looking at the effects of isolation for various age durations and comparing the level of PTH2 we observed in socially reared animals to animals that were isolated. And what we could see is that the qPCR validation looked very much like the next generation sequencing data, that in all the age groups that we examined, by qPCR, we also detected a reduction in PTH2 levels. Now, we and others have considered whether the reduction in PTH2 is a direct consequence of the isolation or might be secondary perhaps to stress that's imposed by the effects of isolation. And so to address this, we actually explicitly looked at several markers of the stress axis, and that's shown here in these, for these 4 genes here. And we compared, using the same data set, the next generation sequencing data set, the levels of these genes in the isolates shown in pale purple and in the social animals. And we could see for all of the genes that we looked at in the next generation sequencing data set, there was no apparent regulation of these stress genes as a result of isolation, suggesting to us that the PTH2 effect that we see is not just a secondary consequence of stress. Moreover, and maybe interesting for some of the people interested in social behaviors, we looked explicitly also at another peptide that's of course been very much implicated in social behaviors, oxytocin. And we also saw that the effect of isolation on oxytocin, there was no effect on the transcript. And another neuropeptide we looked at as a control is arginine vasopressin. And again, all of these data confirmed to us that the effect that we were looking at appeared to be specific, at least for the genes that we've looked at so far for PTH2. And we validated some of these stress-related transcripts also with qPCR. So these data really suggest that PTH2 is directly regulated and that it's not amongst thousands of genes that are regulated, but is a direct target. Now, one of the things that we're interested in is whether the effect of social isolation was long-lasting or whether the animal could recover if it were reintroduced to a social environment. So the way we did this experiment was to take isolated animals and put them into a social context and then ask if the PTH2 levels would recover as a result of that new social experience. So here you can see I'm plotting the PTH2 levels on the y-axis and the hours of social interaction that the animals experienced following isolation. So the first experiment we did was to examine following 12 hours of social introduction. And we could see already at 12 hours, the range of the purple bar here represents the range of data that we would see in socially reared animals. And the purple bar down here represents the level of PTH2 that we would see in isolates. So we could see after 12 hours already that there was significant rescue. So we dialed back looking to see if we could see rescue of PTH2 with even shorter durations of social interactions. And to our surprise, already with 3 hours after days of social isolation, within 3 hours, we could see a significant rescue of PTH2 levels starting to climb up. So we looked even earlier and we could see significant rescue at 1 hour and even at 30 minutes. So after days of isolation, a mere 30-minute exposure to conspecifics starts to elevate the PTH2 levels. So these experiments told us that the PTH2 system can be rapidly regulated and it's extremely sensitive. Now, what about the other way around? So these dynamics show that an individual fish can recover from isolation by being exposed to conspecifics, but can a lifetime of social experience also exhibit very rapid dynamics if a previously social fish is now isolated? And so we did this experiment by examining acute isolation after animals were socially reared. And what we could see in larvae is that after 3 hours of isolation following social rearing, PTH2 levels were slightly reduced, but not significantly reduced. But after just 6 hours of isolation, the PTH2 levels plummet and are statistically indistinguishable from animals that had been isolated their whole life. And we saw the same pattern when we looked at slightly older fish, at juveniles. Now, this interested us, and in our musings about the ability of these, the conspecifics to rescue the PTH2 levels, we were trying a variety of different experiments where sometimes we would add 10 fish and sometimes a few more. And we started to ask ourselves whether the rescue by conspecifics was absolute or graded. That is the fish actually sensitive to the number of conspecifics that are present in its environment? And so we did an experiment where we took larvae and we asked how high the PTH2 level could go depending on how many fish were present in the tank. So I'm showing you here the effect of PTH2 in an isolated fish. And here you can see that as we increase the number of fish per tank, we see an increase, a sublinear, but still a pretty dramatic increase in the PTH2 level. So that means that the PTH2 in an individual fish can be a good readout for how many conspecifics it's been raised in. This was really interesting to us. And we showed the same thing in a different experiment where instead of varying the number of fish, we changed the density by changing the volume in which the same number of fish were raised. And we saw the same pattern of results. That is that if you look at the density and you dilute the density of the fish by simply changing the volume in which they are raised, you see a size-dependent or dilution-dependent decrease in PTH2, indicating again that the PTH2 levels are sensitive to the number of fish. Now, we can also visualize this effect directly in the fish because we have probes where we can detect the mRNA using in situ hybridization, shown here in blue, and we also raised an antibody to the peptide, which is shown here in this immunofluorescence labeling. And I'll show you in a few slides better resolution of where these PTH2 containing cells reside. But here you can see the immunofluorescent labeling along with some of the processes that extend from the PTH2 cells. And you can also see the in situ hybridization labeling. And this is in fish that were reared socially. And if by contrast you examine in an isolated fish, you see a complete absence of PTH2 at the mRNA level. And this is accompanied by a complete loss of the protein. So the mRNA changes are also read out at the level of the protein. So where are the PTH2 cells located in the brain? So there is a bilateral cluster of around 22 cells split in half that are present in the ventral thalamus in the diencephalon of the animals. And this is a maximum intensity projection where you can see with immunostaining the presence of the cell bodies, but two sets of projections actually. And there's an anterior projection that goes to the telencephalon, and there's a posterior projection that sort of weaves its way through the middle of the diencephalon. So there are many potential sites that could see this peptide. And Lucas took advantage of a large single-cell sequencing data set that was published by Alex Shear's lab, where they took the brain of juvenile zebrafish and sorted all the cells and then sequenced all the cells. And this is a map of the different cell types that are clustered using a t-SNE plot to reduce the dimensionality. And what Lucas was able to do is to go and look through that data set and ask which one of these individual cells, which individual cells are actually expressing the receptor for PTH2. And what he saw was a very broad scale expression of the receptor throughout the zebrafish brain. Around 10% of the cells express the receptor for this neuropeptide. And by comparison, if we looked at the expression for the oxytocin receptor or for the arginine vasopressin, one of the arginine vasopressin receptors, there was a much more scant distribution. So these data, we think, support the idea that PTH2 may have a very broad effect on brain function. Now, I'd like to then turn to what really fascinated us for most of the project so far, which is how is the animal actually detecting the presence of others? Which sensory modality is driving the social environment induced rescue of PTH2? Is it because the animals, the fish, can see their conspecifics? Can they taste or smell their conspecifics? Or can they feel their conspecifics? So in the first experiments, Lucas set out to address whether vision played a role. And the way that he did this was to take an isolated fish and ask whether, if it could see neighbors, that was sufficient to drive the rescue. And the way he did that was to make a little chamber where this isolated fish can see the neighbors, but doesn't share swim water with the neighbors and therefore can't feel the neighbors or taste or smell the neighbors. So there's a transparent barrier here. So he asked whether if an isolated fish can see the neighbors, is that sufficient to drive PTH2? And he could compare that to the physical rescue, which we already showed that if you reintroduce an isolated, or you introduce an isolated fish to its conspecifics, this will rescue PTH2. And then we also asked if adding additional conspecifics added anything, because in our density experiments, we knew that the PTH2 levels were sensitive to the density. So if vision is playing a role, then we should double the PTH2 levels when the animals can both see and feel their conspecifics. So how did these data look? So here you can see the purple line indicates the PTH2 levels in the isolates. And this set of data points indicates the traditional rescue by introducing the animal to its conspecifics. And you can see here that the ability of the isolates to visualize their conspecifics did not induce a significant rescue. So although there was a little bit of a nudge up, this wasn't significantly different from the isolates. Moreover, we saw that adding extra animals just in the visual compartment didn't give the expected doubling of the PTH2 rescue. Another experiment that indirectly told us about the role of vision was a rescue that we used with the conspecifics, but in darkness. And what we could see is that preventing the ability of the animals to see their neighbors, but still allowing all the other senses to be intact, did not significantly reduce the rescue that we got if the lights were on. So these data together indicated that vision does not play a major role in the rescue. So what about chemosensation? To address this, what Lucas and Yvonne did was to take an isolated fish, and they reasoned that if taste and smell play a role, that by just changing the solution, basically taking the water from a dish where there were a bunch of conspecifics and adding it to the water of the isolate should be sufficient to induce a rescue. And so what he found is that that rescue of the medium, so taking the kin imbued water, did not rescue the PTH2 levels. So these data then indicate that neither vision nor chemosensation play a role. So what about mechanosensation? So in fish, mechanosensation is mediated by an organ system that's known as the lateral line. And the lateral line is a acousto-mechanosensory system that has a bunch of hair cells or neuromass that are deposited along the length of the animal and also in the head of the animal. And these neuromass respond to mechanical stimulation and transduce signals that result in excitation of downstream synapses. And fortunately for us, it had already been shown in zebrafish that there are chemical means by which one can ablate these neuromass using one of two chemicals, copper sulfate or neomycin. And there's also a dye that you can use to actually visualize the neuromass. And in these experiments, Lucas could see that copper sulfate or neomycin ablated the neuromass here in the head and also in the tail. So now he can ask, can the presence of conspecifics still rescue the effects, the PTH2, the isolation-induced decreases in PTH2 in fish that lack these neuromass? And what he found was that the absence of the neuromass or compromising the lateral line completely blocked the rescue. So these data suggested to us that actually the animals were really feeling by mechanosensation the presence of others, not seeing or smelling or tasting. So we thought about the other kind of experiment. So this experiment shows that blocking mechanosensation blocks the rescue. Can we actually show a sufficiency kind of experiment? Can we drive the rescue by mechanically stimulating? And the first experiment that we did was asked whether any kind of mechanical stimulation of the water might work. And we had on hand brine shrimp, which we actually feed to the zebrafish. And these brine shrimp, Anya Gemmer can actually do a very good in shrimpification of how the shrimp moves all of these appendages here, but I won't replicate it for you. Anyway, these brine shrimp are vigorous movers. And so we added brine shrimp to the isolates dishes. And we found that a small number of these brine shrimp, 14 of them, Artemia, could not rescue. And then just to really see what happens if we really shake things up, we added 1,000 brine shrimp and we still did not rescue. So comparing, this is the level of rescue that's induced by the presence of conspecifics. So then we decided we had to move away from a biologically produced form of mechanics to something more artificial. And we wanted in particular to try to mimic the effects that the zebrafish themselves give in mechanic-wise in the dish. And so Lucas set up a system to actually measure the movement of a zebrafish while it's swimming and measured the kinetics of this movement, in particular of the tail beat when the animals are moving, and quantified the frequency is at around 70 hertz from these kinds of experiments. So then we moved to an artificial way of imposing the mechanical stimulation. And the way that we did this was to drive a piezo device in a dish where the fish were sitting, the previously isolated fish. And we asked if the artificially driven movement of this piezo device could rescue the PTH2 levels, so could mimic the presence of the conspecifics. And so in the very first sets of experiments, so here is the level of PTH2 in the isolate. So we'd be looking for consistent data points up here to indicate rescue. In the very first experiments, we put the piezo in the dish and just delivered a continuous stimulation of 70 hertz. And we saw like a huge variability in the results here. Of course, I was immediately fixated on this data point, and Lucas was actually fixated on these data points. We went on to do an experiment where we said maybe it's not just the movement of the piezo. What if we also add kin water? That also did not induce any rescue. And then we basically systematically went through trying a bunch of different parameters. One idea was, well, we have one piezo, but the rescue is accomplished by multiple fish. So what if we put in 2 piezo devices? Will that rescue? No, that doesn't. What if we now vary the duration of how much stimulation? So for example, we stimulate at 70 hertz for 300 milliseconds on and then off, and then on and then off. That also didn't induce a rescue. And we marched through these different parameters and added also to the 2 piezo devices flexible extensions to create some kind of heterogeneity in the mechanical stimulation. And you can see that with these very regular on-off stimulation paradigms, none of them brought about a rescue. But then in sort of thinking about it and talking with colleagues, we reasoned that this regular stimulation might actually not really mimic the stimulation afforded by conspecifics in that it is so regular, and that these animals might actually be habituating to this stimulus. So we did an experiment then where we randomly delivered bouts of stimulation at 70 hertz, drawing from a distribution, so mimicking fish behavior, but not in a regular way, in a random way. And what we found is that that did the trick. So if we do an unstereotyped delivery of 70 hertz stimulation, trying to mimic the environment imposed by the presence of a bunch of different fish, we saw that this was sufficient to bring about a rescue. So based on these data then, we show that the mechanical stimulation is both necessary and sufficient for PTH2 to sense others. And we're really interested and surprised by these data and interested in the role that mechanosensation might play in zebrafish and in other systems in sensing others. And related to this, there are other systems, particularly in fish, where there are mechanosensitive neurons that are detected in many different brain areas, but one particular brain area is the torus semicircularis. And in electric fish, information about the conspecifics is actually signaled by electric fields that are detected in the torus semicircularis. And there's actually a laminar organization to this torus where different frequencies are represented. And in this regard, we're very interested in this idea since we see an exquisite frequency sensitivity in our rescue experiments, that the rescue seems to be tuned to the frequency that the fish actually swim at. And in other species, there's similarly an involvement of the torus semicircularis in other kinds of acoustic signaling between fish. Now, interestingly, the torus semicircularis is actually homologous to the inferior colliculus in mammals. And if you will remember from the very early slide where I showed the rodent PTH2 expressing cells, that one of these brain areas, the PIL, is actually reciprocally connected with the inferior colliculus. So we come up with then a kind of minimal circuit that may be operative in the zebrafish system, where lateral line afferents would signal to a nucleus that I didn't talk about, the medial octaval lateral nucleus. It then synapses with the torus semicircularis, which then would connect to PTH2 expressing cells, which then, as we described, have their afferents projecting both forward and throughout the diencephalon. So just to wrap up then, I told you that social isolation of zebrafish across developmental stages results in a strong downregulation of a single neuropeptide gene, parathyroid hormone 2. And we found that the PTH2 levels, transcript levels, are rapidly and dynamically regulated in both directions, up or down, by the presence or absence of conspecifics. And surprisingly, we found that the sensory modality responsible for this sensitivity is mechanosensation rather than vision or chemosensation. And the experiments that we're doing now are aimed at behavioral phenotyping of the isolated animals. We also have mutants of PTH2, and we're very much interested in cracking the circuit that's responsible for sending the signal from the lateral line into the PTH2 cells and downstream effects of PTH2 release. So just to circle back to Lucas, this is his baby, really. This is his PhD thesis that he's now writing up, and Yvonne Alcantara. And then to thank the whole Schumann Lab team who have supported Lucas throughout this endeavor with their masks on and their masks off. And thank you very much.
Thank you very much, Erin. That was a great talk. Very interesting results and surprising. Okay, now it's time for questions from the audience, and I already see 2 of them. And I want to invite Robert, if you want to ask live. Let me find you, Robert. Can you raise your hand, Robert, so that I can invite you on the screen? Or if you'd rather, I will ask the talk. Anyway, Robert, you can speak if you like. No, I don't know what's happening. So I will ask the question. So behaviorally, are the fish seeking out physical contact, or do they try to maintain a certain distance from other conspecifics? And when they come into contact, how long is it maintained, or do they act to move away? So whether this is a positive or a negative, let's say, detection.
So it's actually sort of both. So we know that they are attracted to one another. So if you, in that transparent barrier kind of experiment that I described, you will see an approach behavior that a single fish will approach its conspecifics behind that transparent barrier. And after an initial contact, you actually see that they maintain an optimal distance from each other, and they kind of do this mirroring of the polarity of their body axis, which is exploited, of course, in zebrafish and other fish when they school. So when they sort of line their body up in a sort of, so that their long body axis is kind of parallel to one another.
I see.
And we see, I mean, so the maintenance of their coming together, this has been documented that they can sort of habituate. So if you put them in a laboratory setting, they will coalesce and they will either shoal, like just loosely come together, or school. Schooling is when they're sort of polarized, but even those behaviors can habituate over time. But of course, that's in a laboratory setting where there's no other stimuli. So that would probably be interesting to look at in the wild.
Interesting, yes, indeed. Let me ask the next speaker whether they want to ask the question live. Alisa Moon, would you like to ask live? If so, raise your hand or tell me in the chat, otherwise I will read it. It's a little bit awkward with the Zoom. It's not very easy to just start talking. So Alisa wants to know, what kinds of behavioral phenotyping are you working on currently? I'd love to hear more about those plans and your expectations for behavioral differences in this model. Yeah, me too.
Okay, so we're doing a variety of things. I'm going to stop sharing so maybe I can at least see. We're doing a variety of things. One, some very basic experiments looking at how the animals swim. And we actually are looking at, their swim speed, their propensity to enter an open arena. We're looking at their propensity to interact with others. We're looking at their schooling and shoaling behavior. So whether the isolates and the PTH2 mutants show differences in their willingness to join a group, their orientation to the group, like in terms of polarity, their the duration with which they will stay in a group. But these kinds of experiments are computationally very challenging, especially looking, if you want to look at, for example, a single isolate or a mutant in a wild type background. This requires tracking a 5mm fish in a movie over time, which is rather challenging, but we're on it. Lucas is also doing some experiments looking at whether there are changes in anxiety regulated behavior by looking at the startle response. And then of course we could look at other things, learning. There's no real parental behavior in zebrafish, so I guess we won't be looking at that, but we have a whole bunch of assays that we're trying at the moment.
Is there any background information about this particular gene being involved in cognitive functions like learning and memory?
So far not. So the torch for PTH2 has really been carried primarily by Ted Usden, and he has looked with some collaborators at parental behavior. So PTH2 is regulated during lactation and in mother and pup relationships, and it also plays a role in nociception. But there's really, presumably a lot to be done on this.
Cool, exciting. And the next question is by Edmund Tully, whose hand I don't see raised, so I will ask it myself. Have you looked at the peptide receptor expression in mice and all the knockouts in response to social isolation?
Yeah. Yep. No, we haven't, but we are, we're actively thinking whether we should actually do those experiments or wait for someone to do them in mice. It's something that we're actually thinking about. Yeah.
Yeah, that would be really interesting.
Yeah.
And I was also wondering whether this has to do with the medium where the animal lives in. Right? And this is why they use mechanosensation, presumably because they're in water. And I'm wondering whether this is also the case for worms who live in sand. And what happens when in mammals who do not live in a medium? Do you lose the sense?
Yeah, so presumably, so it's definitely the case that mechanoacoustic sensitivity is prominently found in water-dwelling species, also some amphibians. But it's of interest, I think, that heavily touch-related behavior, like maternal behavior with pups and lactation, is also regulated by PTH2, which means that it doesn't necessarily need to be wave let's say, wave generated mechanics, but other mechanical stimulation that's not caused by waves, like in rats during lactation, also appears to tap into this system. So I think that's encouraging, even though I think it's super interesting, even if it's, just in fish or aquatic species, it's also interesting that we can also see it in mammalian species.
And there I would guess it would be grooming, right? I mean, the similar, let's say, sensing.
Potentially, yeah. Maternal induced grooming, or maternal grooming behavior, or even maybe self-grooming, I don't know. Yeah.
And we do know that if grooming is not, let's say, functioning properly, you do have deficits in learning, right? I think there is a link there. So I would expect that there must be an association between changes in this gene and some cognitive behavior. Would be fun to find out.
Yeah, we'll have to see. Early days.
Early days, yes.
Yeah.
Excellent. That's why it's so promising. Okay, is there any other question from the audience? I don't see another question. So if not, I would like to thank you, Erin, very much for joining us for this presentation and sharing with you the exciting news. And also, I forgot to tell in your introduction that you were recently elected in the US Academy of Science. So congratulations about that.
I think that we should also ask your question that you typed in the chat, because it's you said someone should measure the PTH2 levels.
Oh, yeah.
Before and after the pandemic. Before the pandemic, I told Lucas that he should study people in Beijing and people on a walkabout, by themselves in the wilderness. But I don't know why he didn't agree to do that. So
well, but the pandemic is a perfect excuse now for looking. Yes. I bet it will be down-regulated significantly.
Yep.
I'm pretty sure. Well, anyway, more exciting questions for someone to start working on.
Thank you, Jutta.
Thank you very much, Erin, for being with us. Have a nice day. Thank you, everyone, for attending.
So adapting these types of things, I guess, is probably easier for kind of on the go. But yeah, things are starting to get like a little bit movement. We're planning to do the first lab, like Gomez lab experiment next week. So.
Oh, fantastic. Okay, I think we are now live. So let's start the session. Okay, hi everybody, welcome. My name is Robert Fremke. I'm at NYU. I'll be moderating today's session with Andrea Gomez from UC Berkeley. So Andrea did her PhD at NYU in Steve Burden's lab, and then did a postdoc with Peter Schiffele in Switzerland. And now she's really just started her lab at Berkeley. And her work is exemplified by beautiful approaches in looking at the molecular mechanisms and signals that build synapses, which is the topic of her talk today. One fun thing is that Andrea did her PhD in the lab next door to mine when I got my lab started. And she came over to build a slice rig to do some physiology in brain slices, to look at function of LRP4 in the hippocampus. And so she took a rig that was basically in parts and put it all together. And years later, we're still using that rig. Anyway, take it away, Andrea.
Yeah, thanks, Rob. Good memories of that old rig. Yeah, I mean, just to kind of, reiterate, like, a lot of my work that I'm doing here at UC Berkeley is focused on the molecular mechanisms that pattern the flux of ionic current as it's transmitted across neural networks. And specifically, I'm interested in the designation of synaptic properties because these elements profoundly affect the propagation and transfer of network activity. Now, my group aims to discover the molecular coding principles of synaptic organization, and today I'll present a novel framework for how we think synaptic organization is achieved. And as the title of my talk suggests, our framework incorporates alternative RNA splicing, a powerful mechanism that can expand the coding power of the genome. And I'll also share how we test ideas within this framework in my lab using various strategies spanning molecular genetics, electrophysiology, and functional imaging. And toward the end of my talk, I'll take a step back and show some evidence of how synaptic activity interacts with and changes the intrinsic activity of a neuron. And here to demonstrate one of our approaches is an entire mouse brain imaged by 2-photon tomography. And as many of you may know, the structure that we genetically labeled here is indeed the hippocampus. Now, here is another view of the hippocampus, a brain structure that is essential for memory and spatial navigation. Now, when we think about network activity propagating through this circuit, and it does so in this general direction, we know the activity is not continuous. In fact, it is interrupted. As activity passes from neuron to neuron through the circuit, inputs summate, they trigger action potentials and repeat. And this is why I like synapses, or why I'm so interested in the properties of synapses, because they have a profound effect in shaping the way that neuronal activity is propagated across a network. But what are the cellular mechanisms for organizing these properties? Well, for one, we believe that a significant amount of the network organization is predetermined. And we think that unique transcriptional programs that are activated in distinct cell populations direct both the assembly as well as the maintenance of a network. Now, it is my main goal to decode what molecular programs are contained in these cell type transcriptional repertoires that contribute to synaptic organization and function, and specifically those that contribute to synaptic properties. Now, if we zoom in on a single CA1 pyramidal neuron in the hippocampus, we can appreciate that it is targeted by many different classes of neurons. And we can also appreciate that there are very different levels of highly stereotyped synaptic specificity. Now, cell type synaptic specification enables all of these inputs to appropriately identify and synapse onto its target with subcellular precision. Now, each of these connections also have different synaptic properties. For an example, one may have a specific neurotransmitter that mediates excitatory or inhibitory currents. Another may target special components to the presynaptic terminal that tunes the probability of synaptic vesicle fusion with the membrane, resulting in a facilitating or a depressing synapse. Another synapse may have special plasticity features such that when triggered by a strong or specific stimulus, can increase the number of neurotransmitters in the postsynaptic membrane, thus strengthening synaptic transmission. So how are these properties established? And we believe that this occurs through transsynaptic signaling molecules or transsynaptic linkers, each expressed by the presynaptic cell and the postsynaptic cell, that when bound, insist together the components of the presynaptic release machinery, as well as signaling to organize neurotransmitter receptors at the postsynapse. Now, these modules, which we envision as senders and receivers, act to drive overlapping elements of synaptic organizations, such as bidirectional adhesion, it could be presynaptic vesicle recruitment, active zone assembly, or postsynaptic stabilization of neurotransmitter receptors. And we also envision that these transsynaptic signaling modules, or sender and receivers, can be mixed in combinatorial ways to generate the diversity of synaptic properties that we observe in neural assemblies. Now, in our model, the limiting factor for how many different types of synaptic properties that can be achieved in the nervous system is simply the number of available senders and receivers. Which brings me to alternative splicing. So just as a quick refresher on the central dogma, before mRNA can be read into protein, it needs to mature. And introns need to be cut out and exons spliced together. And what can happen during this splicing process, diagrammed here, is that alternative splicing choices can be made. And this happens through RNA motifs and transacting factors which either promote or suppress the usage of specific exons, which then go on to make distinct mRNA isoforms. Now, relating this back to the synapse, alternative splicing has the potential to vastly increase the synaptic proteome, thus increase the modularity of transsynaptic signaling modules, or rather these sender and receivers. And as a final but critical aspect of our model is that we hypothesize that there are splicing regulators that are selectively expressed in different cell types that drive unique splicing events that shape synaptic signaling. Now, back in 2016, when I was a postdoc in Peter Scheifele's lab together with the fantastic student Lisa Tromuller, we found evidence to support this hypothesis. So let me introduce you to the RNA binding protein SLM2. And for the reasons stated before, we became extremely interested in SLM2, which is an RNA binding protein, because we found that SLM2 displays non-overlapping expression patterns in various cell types in the hippocampus. So we generated some SLM2 knockout animals, and we got motivated that perhaps SLM2 had some function in the CNS because they had a weird behavioral phenotype, in that they had an unusual response towards novelty. Normally, when a wild-type mouse is presented with a novel object, it will go and explore that object, here represented as an increase in time spent with the novel object. However, our SLM2 knockout mice displayed no interest in the novel object, and in fact, it actually preferred the familiar object, which is a perseverative behavior. And we didn't believe that this was caused by some type of like anxiety-like feature, like maybe the animal was like, didn't want to go to this novel object and cause stress. Because when we tested them in different kind of anxiety, like our assays, in this one example, the elevated plus maze, showed that SLM2 knockout mice did not avoid the open arms of the maze. Actually, in fact, they actually kind of non-significantly but trended toward just staying into the open arms of the maze. So the loss of this RNA binding protein caused novelty defects in behavior. Are these changes associated with changes in transcriptome? And so to determine what, if any, changes that this RNA binding protein made, we performed a comprehensive global analysis by whole genome RNA-seq. And using Illumina paired end sequencing of libraries prepared either from wild type or SLM2 knockout hippocampus, we observed highly correlated expression levels of genes from knockout and wild type, indicating that the RNA binding protein does not tune overall RNA levels. Now, what about splicing? And here in this volcano plot, where each dot is an exon, we can see statistical differences in excluded or included exons comparing wild type or knockout hippocampal tissues. And what we can, appreciate here is that only a handful of exons were deregulated. Now, at the time, this was an incredibly surprising result because most known RNA binding proteins regulate hundreds, if not thousands, of transcripts. And perhaps more impressively is that the deregulated exons encode for synaptic adhesion molecules, neurexins 1, 2, and 3, and also the syntaxin binding protein Thomasin 2, all of which localize to the presynaptic terminal. Now the question is, do these isoforms have any function at the synapse? And to show what region or what exons were changing in the synapse, here is a qPCR of wild type or SLM2 knockout tissue. And what you can see that in wild type neurexin 1, 2, and 3, if we take a look at this alternative splice site 4 diagrammed here, we can see that the isoforms are deregulated in SLM2 knockouts. I'm sorry, is it really pixelated on the screen?
It looks okay here.
Okay, sorry. It's really pixelated on my screen. I just want to make sure it's not on yours.
Just
So
maybe a little blurry at the top, but I can make out the characters.
okay, so if we take a look at which where these exons that are deregulated, where they map to the orexin protein, we can see that these alternative splice site 4 position maps to a known ligand binding interface on orexin. So then we looked at, how this changed the function of the synapse. So when we looked at the components of the postsynapse and we observed a significant increase in the AMPA receptor subunit GLU-A1 measured on the surface of hippocampal tissue. Now, this coincided with an increase in excitatory current mediated by AMPA receptors. And when I subjected a whole cell recorded CA1 neuron to a special plasticity paradigm, which normally strengthens wild type synapses, SLM2 knockouts failed to undergo long-term potentiation, a process which critically depends on AMPA receptor regulation. Now, I set out to ask if alternative splicing was important for the specification of synaptic properties. And when we disrupted a splicing regulator by knocking out an RNA binding protein, we found that SLM2 is critical for a selective regulation at the RNA level. Not upregulation or downregulation of specific genes, but specifically splicing was disrupted. Now, at the level of the synapse, we got a remarkably selective phenotype marked by a change in the level of AMPA receptors, which was coordinate with an increase in excitation. Now, since neurexins were targeted by SLM2, we hypothesized that neurexin splicing in the presynaptic neuron generates a transsynaptic signal that controls the magnitude of synaptic weight through AMPA receptors. Now, if this hypothesis is correct, then deleting SLM2 from CA3 neurons should produce the same phenotype. And taking this strategy is actually critical to rule out the possibility that the phenotype that we observe is coming indirectly from anywhere else in the circuit where SLM2 is expressed. And finally, to prove that the transsynaptic signal is indeed carried out by neurexin, we went on to restore the wild type repertoires of neurexin isoforms. And we would expect that this should restore the wild type synaptic function and behavior. So first, to probe presynaptic function, I made a conditional knockout by specifically deleting SLM2 from presynaptic CA3 neurons. And to achieve this, I utilized a cell type specific Cree driver that is selectively expressed in CA3 neurons in the hippocampus. So first, to test the selectivity of this Cree driver, cross this line into flocks TD tomato mice and assess the extent of Cree recombination using 2-photon tomography. And this is essentially the whole brain image that you saw at the top of the talk. And this you can appreciate just how selective this Cree driver is. Now, crossing this CA3 Cree driver into SLM2 flocks mice, we were able to generate a targeted deletion of SLM2 from CA3 neurons. And it is likely that this residual SLM2 positive neurons are interneurons that are spread out in CA3 region. Now, what happens to the splicing pattern in SLM2 deficient CA3 neurons? Now, before we did an RNA-seq and assess the RNA splicing pattern in whole hippocampal tissue. But we can't do that in this situation because we are only deleting SLM2 in CA3 neurons. And if we were to drop the whole hippocampus in sequence, we would get some contamination from other cell types where we're not manipulating the cell. So now to selectively look at the transcriptome only from CA3 neurons, we use the ribotag strategy, whereby ribosomes get selectively HA tagged in a Cree-dependent manner. We can then pull down ribosomes and assess the mRNAs. And if we look at wild type CA3 neurons here, this is a qPCR, and look at the neurexin splicing, we can see that the splicing is almost entirely excluded at this fourth position. On the other hand, selectively deleting SLM2 from CA3 neurons results in a loss of alternative splicing at the fourth position. And now all of these neurexin transcripts, neurexin 1, 2, and 3, contain splicing at the fourth position, or contain this exon. Now, because of this highly selective expression of the Cree driver, we can directly test the effect of neurexin splicing onto downstream targets because we think neurexin is localized to the presynaptic terminal. Now, what I've shown you is previously what changes to synaptic currents look like, but we also want to look at what other types of currents, like voltage-gated channels, things that like contribute to a spike, or the ones that determine the resting membrane potential, look like when we change properties of the presynaptic terminal. So now remember, I'm only deleting SLM2 from CA3 neurons. So the CA1 neurons that I'm recording from are wild type. And indeed, when I measure the magnitude of synaptic strength on wild type CA1 neurons that are contacted by mutant CA3 axons, indeed, I see an elevation in synaptic excitation, which confirms that splicing in the presynaptic neuron is instructing for postsynaptic strength. Now, as I mentioned before, what would we expect for the CA1 neuron to respond to this elevation of synaptic strength? And when recording from CA1 neurons, we can see that these wild type CA1 neurons that are contacted by SLM2 deficient axons, we see a decrease in the firing frequency in CA1 neurons. Moreover, when we look at other intrinsic properties like the HCN channel currents, or these IH currents, we see a significant elevation in these HCN channel mediated currents. However, intrinsic activity changes did not happen across the board. When we look at some of the active properties like the action potential, the dynamics did not seem to be changing. So it looks like even though we're seeing synaptic changes that lead to the decrease in firing frequency, the dynamics of these action potentials are not actually changing. And consistent with changes in IH currents and firing frequency, we also see a significant increase in the capacitance of these CA1 neurons that are contacted by mutant axons. We see a decrease in the resistance, and all this is consistent with what we would expect to see is a decrease in the time constant of the CA1 neurons. Now, to take kind of a broader look at what could potentially be changing kind of at a behavioral level, we put these animals through just the open field paradigm, and while most of the behavior didn't change as measured by the distance traveled in the open field maze, the number of crossings, we did see a significant decrease in the velocity of the animals navigating this arena, where only SLM2 was deleted in CA3 neurons. So to just kind of summarize what we saw with the intrinsic properties, which is that when we delete SLM2 from CA3 neurons, which we think targets neurexins, we see this elevation in synaptic excitatory synaptic strength. And coordinate with this increase in excitatory synaptic strength was increased IH currents, decreased firing frequency, faster time constants, and increased capacitance. Now, these results argue that specifically SLM2 in the presynaptic cell generates a transsynaptic signal that is responsible for a synaptic phenotype in the CA1 target pyramidal neurons, further, that this transsynaptic signaling is also controlling the intrinsic properties of the postsynaptic neuron. Now, we believe that this is mediated by neurexins. But to confirm this, we would need to restore the correct neurexin splicing in the presynaptic neuron. And to just remind you, the normal isoform contact in wild type CA1 neurons are neurexin isoforms that skip this alternative splice for position. And when we delete SLM2 from CA neurons, we see this shift. So to restore the neurexin isoforms, we would want to essentially try to make these isoforms that contain the exon and shift that back down to skipping. Now, to rescue this or to mimic this activity, we took a genetic approach. And here is just a diagram of the genetic approach. In using neurexin 1 as an example, we have flox sites surrounding this alternative splice position. And anytime this position sees a Cree, it will essentially mimic the splicing activity as we would expect what SLM2 is doing. Now, to generate a rescue mice of all three neurexins, I'm just kind of outlining the genetic strategy whereby we have a mouse that expressed the CA3 specific Cree with the SLM2 flox and that are wild type on this AS4 plus allele. And we cross them to SLM2 flox mice that are homozygous and flox allele at this AS4 position, with the goal of the rescue mice generating a mouse that has Cree in the CA3 neurons. We delete SLM2 entirely from these neurons, and we flox out, or we cut out one copy of the AS4 minus, essentially mimicking what we would expect in a wild type position. And indeed, whenever we restore the synaptic or mimic the isoforms that we would expect in a wild type neuron, whereas in the SLM2 knockout, we see an elevation in synaptic strength, as we saw before, restoring the neurexin isoforms actually significantly drop the synaptic strength, actually even further below wild type levels, and significantly diminish excitation on CA1 neurons, which confirms that splicing in the presynaptic neuron, again, is instructing for postsynaptic strength. Now, just to wrap up, as I mentioned in the beginning, I set out to identify molecular mechanisms that specify synaptic properties. And I share my view of how a synaptic adhesive code manifests in the hippocampus, and we kind of like to think about this as happening more broadly as manifesting in the central nervous system. But how robust is this code from mice to humans? How is the code manifested when you change the number of neurons by an order of magnitude from mice to humans? And this is why I'm interested in alternative splicing, because this mechanism can increase the coding power of the genome without disrupting the underlying evolutionarily conserved gene expression patterns and overlay another level of complexity. But for this to make sense for the brain, which is a highly heterogeneous system, increasing molecular complexity needs to happen in a selective way. And as I showed you, we discovered a dedicated alternative splicing program that devoted to the splicing of synaptic adhesion molecules. And when this was disrupted, not only did it alter behavior, but it also disrupted excitation and synaptic plasticity in the hippocampus. And importantly, by selectively disrupting splicing in a subset of neurons, we were able to demonstrate that alternative splicing can shape neural circuitry in cell type specific ways. And importantly, we had an RNA processing mechanism essentially control the postsynaptic properties of the connected cell. And this last point is is especially exciting because then we can now go and dissect and molecularly profile what molecular mechanisms are responding to these increases in synaptic strength and result in decreased firing properties and elevations in IH currents. And finally, considering that 95% of multi-exon genes are alternatively spliced, this also points to the possibility that, there are other cell type specific mechanisms out there. And really, can different combinations of RNA binding proteins encode other aspects about synaptic function? Now, our discovery that alternative splicing can shape neural circuitry by selectively modifying synaptic properties is just one study in a growing body of evidence shown by us and others that alternative splicing is a powerful mechanism for network organization and function. And this is especially exciting considering that despite having a comparable number of genes amongst vertebrates, the extent of alternative splicing has expanded in species with more complex nervous systems, such as mammals. And actually the highest complexity is of alternative splicing, or the highest degree of alternative splicing is observed in primates, which positions alternative splicing as a potential driver for the emergence of complex networks. Now, from the scope of synaptic integration, small changes to synaptic properties like we observed in SLM2 mutants would have a big effect on the information flow to a neuron engaged in ongoing network activity. And then knowing this, it may not be so difficult to then imagine how circuits may be changed in neurological disorders where we know that synaptic dysfunction is disrupted. And this last point is especially relevant as disruptions to alternative splicing has been implicated in disorders ranging from autism to spinal muscular atrophy. And finally, understanding how splice codes are distributed across different cell types, we can then start to derive some cellular rules for how these splice codes affect neural integration and computation. And with that, I would like to acknowledge the lab members in my group, Adrian, Maura, and Anjali, and especially Peter Scheifele and Lisa Tromuller, who a lot of this work was done in the University of Basel in Switzerland, and also for my funding. And of course, you for your attention.
Thanks, Andrea. It's fantastic. It's remarkable the level of control you have over specific connections. So we have some questions. For one, what are you thinking about in terms of potentially IPing SLM2? Are you going to follow up with sequencing or mass spec?
Mm-hmm. So I guess maybe is the question to, I mean, the benefit or the idea of IPing SLM2 or what we could learn from that is, what is SLM2 interacting with potentially? Right. Now we know that SLM2 already targets neurexins by direct binding assays, but we actually don't know, how is it that SLM2 biases the incorporation of, that AS4 region. And, potentially there are other factors that SLM2 is interacting with so that the RNA that exon is essentially being skipped. So yeah, I think that we could definitely learn a lot from understanding kind of the splice, or rather we would learn a lot by IPing SLM2 and understanding kind of what splicing factors are interacting in that engagement.
So you showed in the SLM2 knockout an impairment in LTP, which I think was induced by maybe data burst, some kind of technization, right? Mm-hmm. What about other forms of synaptic plasticity? In particular, the question asks about spike timing or input timing dependent plasticity. Is it because the strengths are already kind of maxed out, or do you think there are other ways of modifying the synapse?
Yeah, so we haven't done any type of other plasticity paradigms yet, except for short-term plasticity, which we didn't see changing in our mutants. And yeah, the kind of current hypothesis is that because we saw this elevation in AMPA receptor at the synapse, we think that the excitation has essentially, or rather the plasticity has kind of reached a ceiling, and we hypothesize that AMPA receptors are not localizing or increasing at the synaptic membrane. But another plasticity that we are really interested in, what are essentially the next experiments that Adrian is going to do, are looking at input dependent plasticities. So if we consider the CA1 neuron as where it sits in the hippocampus, it also receives input from the entorhinal cortex. So we would expect that, because we're biasing all the synaptic excitation from CA3, that now the CA1 neuron is unlikely to kind of receive the entorhinal cortical input in kind of the normal timing fashion. So we would expect there to be a deficiency in the input dependent plasticity.
Yeah, so on that note, I mean, is that how you're thinking about the changes in the intrinsic properties? That they're compensating for one set of inputs changing, which might be a driver input, but that could impact either positively or negatively the integration and arrival of other inputs.
Yeah, I mean, at this point, we don't know if it's a direct or indirect kind of compensatory mechanism, whether or not the neuron kind of senses, that there's this increase in excitation, therefore it maybe, increases or decreases specific genes to make sure that firing frequency goes down and IH goes up, which, should essentially balance this elevated excitation. Or, if it's direct, maybe that there are some kind of local signaling pathways that are engaged that elevate these specific intrinsic electrical properties. But that's actually a project that Maura McDonough, a student in the lab, is going to do, is essentially molecularly profile CA1 neurons, these wild type CA1 neurons, and see not just what gene expression changes are happening, but also look at more in-depth kind of alternative splicing.
Yeah, for sure. So how complex is alternative splicing? How much kind of variability does this provide? Specifically, is it enough to express, to give all the synaptic varieties that you might see in the hippocampus and CA1, CA3 in the brain?
Yeah, I mean, I think the what we have in terms of the role of alternative splicing, or the role of diversity and complexity, is at this point correlative. What I think is kind of evident, or maybe some evidence, is if we consider just the neurexin gene family, from 3 genes, 6 alternative splicing positions, and 3 promoters, we go from 3 genes to 12,000 different potential isoforms. And this would vastly expand the potential interactions that these neurexins can make, and we're just focused on neurexins. So I think it's certainly possible that if we look at other transsynaptic signaling systems, that we would see a similar trend, especially knowing that alternative splicing, or kind of evolutionarily conserved alternative splicing positions, target protein, our ligand binding domains, post-translational modification, and intrinsically disordered domains, which are posed in position to really affect signaling mechanisms drastically. And that's just one aspect of alternative splicing. So we're not even talking about the non-coding aspects about alternative splicing. So we can get alternative 5' and 3' UTRs, which can dramatically affect RNA stability. It can affect RNA localization. So there's a lot of ways that alternative splicing can dramatically expand essentially the processing and signaling of these RNA molecules.
In the RNA-seq data from the SLM2 knockouts, you highlighted SLM1. It looked like there might have been a 50% increase or so. Do you think that's real? Is there some compensation at that level, or?
Yeah, so Lisa Tromula, the student that I worked with on this project before, published a paper in 2014 that showed that SLM2 negatively regulates SLM1. we know that it actually SLM2 binds to SLM1 RNA and initiates a nonsense mediated decay pathway. So what that translates to is that anywhere there's SLM2, SLM1 is going to go down, and that's actually what we see in the splicing data. Is you see that that exon SLM1 go up because we have a diminished SLM2.
In the experiment you showed up towards the end, the rescue experiment, where the synaptic strength capped out lower than in wild types. I mean, the combinatorics are a little bit crazy, but have you tried to do subsets of the orexins, not rescuing 1, 2, and 3, but just one at a time? Do they kind of sum up linearly in terms of potential for rescue, or is there one that's more of a dominant isoform?
Yeah, so just by expression, as neurexin 1 is the most abundant isoform in CA3 neurons. And when I did single rescues, I saw a similar trend of it actually kind of overshooting and going all the way down. And now I haven't done kind of the combinatorial ones where with neurexin 2, neurexin 3, are kind of different combinations of those. I haven't tried yet, in part because these mouse genetic experiments are quite tedious. It took me just 3 years to generate this mouse, this rescue mouse. But I think that it's actually a very relevant question. We don't know how neurexins are being distributed to all the axon terminals. We don't know if some neurexins are targeting to CA1 neurons, or maybe some are targeting to PV neurons, or maybe some are not localized to terminals at all. Maybe there are some mechanisms that regulate whether or not an neurexin is trafficked to a terminal. We have absolutely no idea about these things. There has been some studies, super resolution studies from Sudov's lab looking at neurexin distribution and potentially showing that they're not at all synapses, which is actually really interesting. So for that particular question, be extremely relevant to know if they're affecting the function of the targets. And in that particular experiment, as you suggested, could potentially answer that.
Okay. Another question. Is there good pharmacology for neurexin? Are there agents you can use to kind of regulate it in the moment?
Unfortunately, no, not that I'm aware. I know that there have been multiple attempts to generate antibodies to target these, but at the moment, no. I think some potential avenues that I'm particularly interested is the use of these CRISPR epi editors, essentially to target the regulatory regions on the DNA and essentially try to shut down or change the potential expression of neurexin. But in terms of kind of acutely changing their patterns, it's been kind of difficult. And as you can imagine, there's so many isoforms. So which one do you choose?
Okay, thank you so much. With that, I think we'll close the session. Thanks, Andrea, for a fantastic talk. And thanks to all of you watching in Zoom and YouTube for the questions. And have a good rest of the day. Bye, everybody.
Bye.
Okay, so we have Claudio and myself have dressed up a little bit. If any of the audience have dressed up and want to go on screen to ask questions in a costume, that would also be possible potentially. Okay, so it's my great pleasure to have everyone here for this session that looks very interesting about basal ganglion motor control. So super cool, right up my alley. So I'm very much looking forward to that. Just a quick reminder, and first of all, thanks to Elena Tseva, who's our host in the background, for running everything smoothly. So thank you so much. And then a reminder that you should use the Q&A tools, please ask the question. Try to do that before the end of the talk so that we don't have to wait for the questions to come in, because that wastes some time. And of course, chat away as much as you want as well. Okay, so without further ado, it's my great pleasure to introduce Marie Labouesse, who is a postdoc at Columbia University in New York in Christophe Kellendong's lab. And I hear she'll be moving to Switzerland very soon, because apparently there's nice mountains and cheese there, and also some dopamine sensors in Tommaso Patriarchi's lab. So Marie will be talking about striatal D1 contralaterals to the global pallidus externa act as a second direct pathway to regulate motor coordination. Marie, all yours. Don't forget to unmute you.
Unmute. Can you hear me?
Yeah.
Okay. And you can see my screen as well.
Absolutely.
Okay, so thank you for the introduction, and I'd like to present a story about non-canonical projections from the striatum to the GPE arising from the direct pathway. So as many of you know, the basal ganglia, and specifically the striatum, is composed of two cell types, projection cell types. Direct pathway neurons projecting to the EP and SNR that are thought to promote movement, and indirect pathway neurons that project to the GPE and are thought to inhibit movements or inhibit opposing movements. However, there's been many studies, especially in the past decade, that have shown that this model is actually a little bit more complicated than initially thought. For example, these neurons are not as well anatomically segregated as we thought. For example, there's work showing that there's local collaterals between these neurons and that control circuit activity. There's also work showing that these neurons are not always functionally opposing. For example, they can both be activated concurrently during movement. So work from our lab also found that the direct pathway does not only, or D1 MSNs don't only project to the midbrain, but also have axonal collaterals, which we called bridging collaterals, emerging from the direct pathway axons and targeting the GPE, which classically is thought to be the target of the indirect pathway. So this was based on single cell tracing work that found that up to 60% of neurons projecting to the midbrain, the SNR, also had these collaterals in the GPE. And in this work, they found that, in fact, the density of these collaterals is actually extremely plastic. For example, overexpressing dopamine 2 receptors in the striatum led to an increase in terminal density of collaterals in the GPE. And this was remarkably plastic especially for adult animals, because turning on and then off and then on again, the D2E transgene led to dynamic changes in the density of collaterals. So what we wondered is whether this anatomical plasticity extended to behavioral experience. And so we looked at whether training animals on a rotarod for 1 month can modulate the density of these collaterals. And we found that indeed in D2E mice, there was a restoration to normal levels of bridging collateral density after this training. And so this indicated to us that most likely bridging collaterals contribute to motor control. It was just unclear in which direction. And so we had two hypotheses. On the one hand, bridging collaterals could act as a second indirect pathway, mimicking inputs from IMSNs in the GPE. On the other hand, they could act as another direct pathway using a different subcircuit. And so first, to answer this question, we decided to look at whether bridging collaterals are active during specific motor tasks. We used fiber photometry and in vivo calcium imaging to image terminal activity in the GPE during a rotarod task, and found that indeed the activity of bridging collaterals or of DMSN terminals in the GPE was markedly increased during the rotarod in a sustained manner. And this was illustrated by, for example, increased baseline and increased AUC. What was particularly interesting is that during the rotarod, we found that there was these regular small width and small height peaks at about 0.5 hertz And we wondered whether these specific transients could encode specific movements as the mice were running. And so in order to do that, we used deep learning based behavioral tracking, deep lab cut, to align high resolution behavioral tracking with calcium imaging. In this case, we actually decided to track terminal activity along the axonal tract in the GPE as well as in the SNR in order to determine whether terminal activity in these two regions was correlated, or rather whether they differed during movement. And so this is an example of the tracking as the animals are running. We had kind of two hypotheses. On the one hand, we thought that we could track steps as animals ran on the rotorod. This was actually based on previous work that correlated foot height with motor training. But then the other hypothesis, basically we observed that as animals perform this task, they didn't always actually walk, but actually rather jumped. They let their body go down on the rotorod and then jumped up. And so we wanted to track the lower body. And so we labeled as well the intersection with the tail. What we found is that all body parts had an elevated activity during the rotorod, but we also found that whereas the tail had a very high likelihood and accuracy of tracking across all animals, the other body parts were not necessarily tracked as well. And this is because since these animals are not fixed, they're freely moving, they cannot adopt different strategies, for example, hiding against the wall. And so we decided to focus on this lower body tracking to kind of identify these jumps. So we were looking for changes in tail position from the lower bound of the rotorod to the upper bound. And when looking closely at the tail position in the y-axis, we indeed found that there was these rapid shifts in a regular manner from the lower bound to the upper bound. And when we aligned data to these jump onsets, so the orange circles, we could see that there was like a low variability traces of tail position. And importantly, this position was regulated by speed. So the jump frequency, the inverse of the jump duration, increased as the speed of the rotorod increased, indicating that jump frequency was regulated by behavior. And this was about 0.5 to 1 hertz, which was actually pretty consistent with our calcium data. So we aligned tail position with calcium data and remarkably found that there was a highly similar pattern between calcium traces and tail position. There was a significant correlation both at the level of the GPE and the SNR with minimal lag, 100 to 150 milliseconds, which is consistent with the kinetics of the sensor. And so this shows to us that terminal activity in both these regions is time locked to the onset of jumps during running. What we also looked at is whether there was a correlation between the GPE and the SNR calcium data. And indeed, we found that expectedly, there was a high correlation between a GPE and SNR terminal activity, specifically during the pre and the post phase, so before and after running. And there was a reduction in this correlation coefficient during the running phase. So this shows us that although GPE and SNR terminal activity is highly correlated, there may be a differential regulation or different encoding of specific movements other than the jumps in these brain regions during running. So in a next and final step in this investigations, we also wanted to confirm that we're definitely recording bridging collateral activity. The question was basically, since there's axonal tracts in this region, there could be contamination of calcium terminal activity by axonal calcium. And so we used a GCAMP sensor that was tagged to synaptophysin and enriched in synaptic terminals and co-expressed with a presynaptic marker VGAT. And what we found is that indeed the activity of bridging collaterals definitely increased during rotor-rod running. And so this, in the next step, we wanted to determine whether if we inhibit this endogenous activity, does it modulate motor behavior? So this was based on previous work from our lab that showed that stimulating both the canonical neurons, D2 neurons, and the non-canonical neurons, bridging collaterals, D1 neurons, leads to an inhibition of GPE activity. And if you look at D2E mice that have excess collaterals, there was actually elevated inhibition, basically consistent with the existence of excess bridging collaterals. So what we needed to, in order to inhibit activity of bridging collaterals, is a tool that would specifically inhibit synaptic release, but not affect action potential propagation, because this would affect downstream activity in the SNR and EP. And so we basically decided to use HM4D, which has been shown to inhibit synaptic release without affecting action potential propagation. We injected it in the striatum and locally infused CNO in the GPE. We used autoradiography in collaboration with Mike Michaelides lab to confirm that the the CNO was restricted to the GPE after local infusion. And when we infused CNO in HM4D animals, we saw a marked reduction in total locomotion, which was not the case in controls. And we found similar effects when looking at motor performance. So HM4D animals infused with CNO had a marked increase in the number of falls. And then we looked at D2E animals, which have excess bridging collaterals. And we were looking at whether these animals were more sensitive to this manipulation. So at a high dose of CNO, we did not detect any difference. But at a low dose of CNO, only D2E mice were sensitive to this manipulation. And so this indicates that bridging collaterals, in fact, have a role in supporting motor control and motor coordination. We also confirmed these effects using optogenetic in a small cohort. We found that E-arch inhibition of bridging collaterals led to a reduction in locomotion. And so to conclude, here I showed that bridging collaterals, in fact, act as a direct pathway using a different subcircuit, likely using a different subcircuit. I don't have time to go into the details of this, but there's been a number of studies showing that there's a rich cellular diversity in the GPE. There are canonical neurons projecting downstream, but also these back projecting neurons going to the striatum. And there's recent work in preprints that show that actually bridging collaterals have a bias in their targeting towards NPAS1 neurons. And this has been shown both in slice and in vivo. And so we're currently looking into the behavioral relevance of this. So I'd like to thank you for your attention and thank my mentor, as well as everybody who's helped on this project, our collaborators, and our funding. Thank you.
Great. Thank you so much, Marie. Are there any questions? Please ask your questions in the Q&A. We have time for a couple questions. While we're waiting for questions to trickle in, so how do the different basal ganglia pathways help in controlling movements according to your new findings? How do you think about it?
Yeah, good question. So, I mean, we don't, basically, like one possibility is that if you have these different direct pathways, of course, I didn't mention the EP, but there's the direct pathway to the SNR, to the EP and the GPE now. It could be that they have different functions in the regulation of circuit activity. SNR neurons, I think we'll talk about it later, project to the thalamus, to many other regions, more like downstream, and have a more direct effect on motor output. In this case, we don't know the details yet, but basically the slice work shows that bridging collaterals target NPAS neurons, and in turn, NPAS neurons go back to the striatum, and they affect striatal activity, specifically many cell types, but specifically D1 and D2 MSNs. So there could be, there's like a direct synapse back to the striatum that could act somehow as a gain control, as a local feedback. And so this could help to fine tune the movement rather than affecting direct motor output at the level of the thalamus.
So that's with respect to the fine tuning, as you call it, which I actually like in terms of hypothesis. But you also talked about motor coordination, and I find it in general a much more open question in the field, how different movements are coordinated. Do you have any suggestions there on what the basal ganglia in general would be doing?
That's a big question. I don't know. This is like purely speculative, but there's been some hypothesis that there's like these kind of functional units in the GPE that basically, like, especially at the level of these local collaterals. So not my collateral, not the axonal collaterals, but the local collaterals, that neurons that inhibit each other are actually part of specific functional units, and that one unit might affect one motor output and another unit might affect another motor output. And so it would be really interesting to know whether these bridging collaterals actually inhibit neurons that are end-pass neurons that go back to neurons in the same functional unit, the same neurons that they receive input from in a really tight closed loop, or whether they actually impact the opposite functional unit. And even this functional unit concept is still under debate. So yeah, that would be really interesting to follow up on.
Cool, thank you so much. There is a question in the Q&A, but we're unfortunately out of time. So Marie, I encourage you to just type out the answer in the Q&A directly. You can answer there directly and get in touch with the person asking the question. And then we're moving on to our next speaker, Claudia Villalobos Dintras, who told me that he was originally from Chile and got interested in neuroscience through work from the Montemar laboratory in the 60s and 70s. And some of you might know that they also recorded in the local version, as Claudia called it, of the giant squid, and did some cool electrophysiology there. And today, Claudia will talk about optogenetic activation of the inhibitory nigrocollicular pathway and how it evokes paradoxical contralateral orienting movements in mice. So please, Claudia, take it away. You have to unmute yourself.
Okay. Is that okay?
Perfect.
All right, cool. Thanks for the invitation and running flawless and NeuroMatch 3.0. Yeah, so the work that we've been carrying out involves the regulation from the basic ganglia and the inputs into the superior colliculus and how this controls especially orienting movement. So in the superior colliculus, and I'm going to show a picture of volleyball because we are in South California. I know this seems warm, the rest of the country is getting cold, but we need to, the superior colliculus integrate a lot of sensory information in order to try to decide what's the best to transform those sensory inputs into motor actions for reaching different goals. In the case of volleyball, you need to see the speed of the wind, you need to look at the visual inputs of the ball to try to get the ball and get the point. When these motor actions, they get somehow intervene or you don't get the right motor action, you get, undecided effects that they can be painful under certain circumstances. So the normal, the current model of how the superior colliculus and the basal ganglia interact to produce oriented movements is derived from the work from Hikosaku works from the 80s, where they see that during the eye movement, there's a reduction in the activity of the substantia nigra right when at the time that the eye start moving. This led to create a hypothesis that to be able to move, to control eye movement, there's this inhibition of the substantia nigra into the superior colliculus that release the concept inhibition, allowing the cell to the superior colliculus to burst and produce and activate it to produce the contralateral movement that generates the eye movement. All this work has been done mostly in monkeys, and it generates this model where the substantia nigra plays a passive role, where the lower activity of this inhibitory inputs into the superior colliculus allow cortical excitatory inputs to produce these oriented movements. And this has been the textbook model of how the oriented movements have been developed. And it hasn't been challenged in the past 20, 30 years. However, a recent studies have suggested that the model might not be completely correct or not as simple as the original idea suggests. For example, in birds, which has an homologue of the superior colliculus in mammals, you see the connection between the area 8 and the BLM that is similar to the connections from basic ganglia and thalamus. When the activation of the area X, that is an inhibitory synapse through the BLM, you can see that there's an inhibitory, this is an inhibitory response, but after the cessation of the inhibitory response, there was some spike on the cells from the BLM that it started to create this idea that the role of these inhibitory inputs can actually not just be passive, but it could be actually active. And in results from our lab in monkeys where they stimulate in the suspension nigra in the record cells in the superior colliculus, they see that in these panels, you see when there's an activation of the suspension nigra, you can see the inhibition of the activity of the cells in the superior colliculus, but there's a ramping of the activity. So it's not on associated with a more concentrated eye movement. So this association between the inhibitory inputs of the superior colliculus, the basal ganglia into the superior colliculus might produce a different model than the ones that we know from the original cells in monkeys. So my goal was to actually test this idea, and we did a simple experiments where we inject an opsin into this suspension nigra and activated the terminals of the nigrocollicular pathway into the superior colliculus in freely moving animals to see what happened, what kind of movement we will get, and whether we will move contralateral or ipsilateral to the side of the stimulation. So the possible outcomes is that if we activated the cells from the nigrocollicular terminals, so not only stopping the inhibition, but increasing the inhibition, one of the outcomes is to include the superior colliculus gets inhibited, as the model suggests, and we'll have no movement. An alternative hypothesis is that the activation of the nigrocollicular pathway will inhibit the ipsilateral superior colliculus, allowing the increased activity of the contralateral superior colliculus. And because they project contralateral to the brainstem, they will generate ipsilateral movement. So these are all hypotheses that agree with the current model, and this is kind of what we expected. But what is what we see? So, and this is just videos of the mice, and you can see when we activate, actually the optoprobe is on the right side, so you can see when we put the light pulses, the mouse actually not only doesn't move or ipsilateral, but it has a strongly contralateral oriented movement that is evoked by the activation of the nigrocollicular terminal. And you can see in a mouse that it is being transfected into suspension nigra only with GFP. Activation of the light pulses doesn't produce these oriented movements. And this is just a quantification. We did like 4 or 5 mice, and all the mice, they show increased contralateral movements compared to ipsilateral movements, and there is significant movement compared to the control mice. So this is definitely an unexpected result that doesn't agree much with the previous model of how the basal ganglia and the superior colliculus interact to produce oriented movements. So we next wanted to see, so what's the mechanism that underlies this oriented movement that is evoked by inhibitory inputs. So we decided to test the outputs, the activity of the output cells of the superior colliculus, that they're the ones that they go contralateral into the brainstem. And we saw that the cells actually, with the inhibitory inputs, they do spike after the presence of inhibitory input, but the big majority of these cells. So these cells can spike with inhibitory inputs. So then we want to see, so what is the mechanism that it produces spiking through inhibition in these cells that they can trigger these oriented movements? So we decide to label these cells using a retrograde virus and using the same type of injections that we use for the in vivo experiment, and see using patch clamp on these specific cells whether the neuromuscular activation can actually reduce this inhibition and spiking. And we can see exactly on what happened is that's what we thought, that these cells from the alpha cells from the superior colliculus, when activated through the neuromuscular pathway using optogenetics, they do present inhibition, and upon the cessation of this activation, the cells start spiking. So these type of cells, and you can see that there's no spiking, and when you do the optogenic activation, the cells start spiking. So these specific cells, the alpha cells of superior colliculus, they do spike with increase of activity of the neuromuscular pathway. And then we further want to see just a pharmacological corroboration that how they do it, and they do it through activation of the T-type calcium channels, which is the ones that are producing the rebound depolarization that you see at the end of the inhibition, and it produces the spiking of these cells. So the conclusions is that so we have the optogenic activation of the neuromuscular pathway evolves instead of just preventing movement. It goes contralateral movement in oriented movement to mice, and this oriented movement is produced by a rebound depolarization and spiking in these cells in the alpha cells of the superior colliculus. And this rebound depolarization and spiking is produced by a T-type calcium channel that is producing these rebound depolarizations and spiking in these cells. And at the end, I just wanted to acknowledge Michelle, and all the personnel in the laboratory, my collaborators, and our funding institutions. Thank you.
Awesome. Thank you so much, Claudio. We have a question in the Q&A by Marcus. Marcus asks, if excitation of the SNR causes superior colliculus excitation, what is the difference with the canonical model of disinhibition, as inhibition or excitation of the SNR would appear to have the same effect?
Right. So the canonical model says that the cessation of the activity of the substantia nigra, what it produces, it produces this inhibition of the cells from the superior colliculus, allowing excitatory cortical inputs to excite the cell. What we're suggesting is that the cortical cells, the activation of the neurocollicular pathway by itself can produce this oriented movement. So the role of the basal ganglia is not just passive, like a gate, but it's actually inducing these oriented movements by activating the output cells of the collicular through rebound depolarization.
So can I follow up on that? So is that just because of the way you manipulate the cell's activity essentially throughout the genetics in your experiment? Or do you think that that would also hold in like natural behavior?
Right. So this is another hole that there is in the knowledge of these systems, where it's commonly known that the cells from the substantia nigra spike between 20 and 40 hertz. But recent studies have suggested that there's an increase on the activity of the cells up to 100 to 200 hertz, which is physiologically correlates with the type of simulations that we have done. And that increase in activity in the substantia nigra might be correlated with behavioral. So we think that the substantia nigra also modulates its activity, and it not only decrease, but also can increase its activity, and this increase in activity could produce these oriented movements in the superior colliculus.
I have another question. So when you showed us the video of the mice moving with the stimulation, it seemed like that the movement of the mouse was continuous, despite the fact that the stimulation went on and off. And I would expect, have expected that the movement was like, there was a movement, then it would stop. There was a movement again, it would stop. Can you comment on that?
Yes, that's exactly what happens. I'm sorry to mention the video is speed up for time's sake, but that's exactly what happens. If you see a slow down video, you can see that not only the movement is not continuous, but the movement happens right at the end of the stimulation. So it's not like we're stimulating something, but you stimulate, and at the end of stimulation, the mouse moves. You stimulate again, and at the end of stimulation, the mouse moves again. In the preprint, or whoever wants to know more, I can send you a slow down video when you can really see this effect, and that's exactly how it happens. It's not a continuous movement, but it's a movement that happens right at the end of the simulation.
Yeah, so that was just the way that the video was a little too fast to notice that. Okay, cool. Thank you so much. Are there any other questions? Maybe someone from the panels? Any of the panelists have any questions? If not, then we can move on. Thank you so much again, Claudia. That was a great presentation. So next up, we have Priscilla Ambrosi, who is a PhD candidate in Thalia Learner's Lab at Northwestern University, and will tell us about dopamine circuits driving the transition to habits. All yours. You have to unmute yourself.
Thank you. Okay. Hooray, Zoom. Hi, everyone. My name is Priscilla Ambrosi. Thank you for the introduction, Gunnar. So yeah, it's a pleasure to be here today. So, okay, habits often develop from goal-directed behaviors. Right now we're all washing our hands to protect from coronavirus. But with repetition, you might develop a habit or a behavior that is driven by a stimulus response association. So every time you touch a doorknob, you wash your hands. And this is a benign habit, but habits can become maladaptive and turn into compulsions or obsessions. So if you constantly feel that your hands are covered in germs and you wash them to the point of damaging your skin, that is no longer beneficial. It is far removed from the goal of protecting you from infection. And I am interested in the circuits underlying this transition in the brain. Thankfully, we can model this transition in rodents by teaching them to nose poke or lever press for a reward. And with that, previous studies have shown that the basal ganglia seems to play an important role in habit formation. Specifically, the dorsal striatum, which can be subdivided into dorsal medial striatum, or DMS, and dorsal lateral striatum, or DLS. So if, as animals learn a new motor task and transition from being goal-directed to habitual, there's a shift in synaptic activity from DMS to DLS. And if you lesion or inhibit DLS, you hinder habit formation. More specifically, if you lesion the dopamine cells that project to DLS, you also hinder habit formation. So together, these findings point to this putative circuit connecting DMS to DLS through substantia nigra as important for habit formation. And this circuit is part of a larger model called the ascending spiral. And the strongest evidence supporting the existence of this circuit comes from a tracing study in non-human primates. So here I'm showing you the striatum subdivided into caudate and putamen. And just for the sake of clarity, I'll be referring to these as the rodent homologue, DMS and DLS. And here we have substantia nigra. So what the study has shown is that there's this topographical organization of the projections from substantia nigra to striatum, such that medial goes to medial and lateral to lateral. And this organization is mostly conserved on the opposite direction, with the exception that DMS also projects to lateral substantia nigra. So if you put these two things together, there's this overlap between terminals from DMS and cell bodies from DLS projecting cells. And with this, it has been hypothesized that DMS activation can somehow excite DLS projecting dopamine neurons, leading to dopamine release in DLS. And the repeated activation of the circuit would then strengthen incoming synapses from sensory motor cortex, building those stimulus response associations required for habit formation. Now, there's there's a growing literature relying on this model as the mechanism behind habit formation, but there's a few important missing pieces here. So first, neuroterminals from DMS, axon terminals from DMS are GABAergic. So a direct connection between DMS and SNC would actually lead to the opposite effect on dopamine release. So the current ascending spiral model assumes that this connection between DMS and SNC is indirect, such that DMS activation inhibits tonically firing GABAergic neurons in SNR, thus disinhibiting DLS projecting dopamine neurons, allowing them to fire more and release more dopamine. Okay, but then if you are an electrophysiologist like me, you might be a bit skeptical about the anatomical data supporting this circuit, given that the overlap between axon terminals and cell bodies is not necessary nor sufficient for the existence of a functional circuit, especially not a polysynaptic circuit. And that's where I come in. So I am testing the existence of the circuit in mice. And here I just have a cartoon showing the striatum of mice, substantia nigra pars compacta, SNC, where dopamine cells are, and pars reticulata, SNR, where GABAergic cells are. And today I'll discuss two questions I'm trying to answer. One, is there a polysynaptic circuit of disinhibition from DMS to DLS projecting neurons? And two, does DMS activation actually lead to dopamine release in DLS? And to answer these questions, I'm using ex vivo electrophysiology and in vivo fiber photometry. All right, so for the first question, I'm also using projection labeling and optogenetics to study this long-range circuit in brain slices. And these techniques allow me to record specifically from these DLS projecting dopamine neurons while stimulating DMS-targeted GABAergic neurons in SNR. So I'm not interested in the general connection between SNR and SNC, but really the connection between these two subpopulations based on their inputs and outputs. Okay, so how do I test this circuit? First, I need to insert chondrodopsin in this particular subpopulation. How do I do that? I take an adult mouse, I inject a virus encoding Cre into DMS, and this particular virus has been shown to move transsynaptically in the anterograde direction, such that cells that are postsynaptic to DMS will also be infected and carry Cre. And that allows, so cells, importantly for this experiment, cells that are DMS targeted in SNR will carry Cre. Unfortunately, some dopamine cells will also carry Cre, given the direct connection between DMS and SNC that was previously shown by my advisor. So to prevent chondrodopsin expression in dopamine cells, I do two things. One, I use a Th2A FLIPO mouse in which FLIP is expressed in Th positive or dopaminergic cells. And two, I inject a virus encoding Creon flip-off chondrodopsin in substantia nigra. So this virus ensures that chondrodopsin is only expressed in cells that have Cre, but not FLIP. That is how I get chondrodopsin expression only in this subpopulation, cells that are Cre positive and therefore DMS targeted, but FLIP negative and therefore non-dopaminergic. To complete the setup, I also inject retrobeads in DLS, and these are fluorescently labeled latex beads that travel retrogradely from axon terminals to cell bodies. And this allows me to find DLS projecting cells in substantia nigra and patch from them while I'm stimulating this subpopulation of SNR cells. All right, and when I record from these DLS projecting dopamine neurons, I use a high chloride internal and a cocktail of drugs to isolate monosynaptic inhibitory synapses. And here is an example cell. You see the current in response to a light pulse, a 5 milliseconds light pulse. The thin traces are 10 individual sweeps and the thick trace is the average. And this cell is in fact inhibited by these subpopulation of SNR cells. If it wasn't, this would be a flat line. And here are the average responses from all of the cells I recorded. As you can see, there are some flat lines, but in fact, around 80% of the cells I recorded were inhibited. So that's really exciting because this is the first functional evidence of the existence of this synapse, which supports the existence of the first, of the whole polysynaptic circuit. All right, cool. So it looks like the circuit exists, but does DMS activation leads to dopamine release in DLS? To answer that, I injected a virus encoding D-light into DLS, implanted an optic fiber over DLS to record dopamine transients in the area. And then I injected another virus encoding crimson in DMS and implanted an optic fiber over substantia nigra so I can activate terminals from DMS in substantia nigra while the animal goes about his business in an open field box. All right, brace yourselves, here comes the data. It's a whole lot of nothing. So surprisingly, in the 4 animals I recorded, the activation of these DMS terminals in SNR does not seem to cause a dopamine transient in DLS. So, my first thought was great, my setup doesn't work. So to check that I could detect dopamine transients in DLS, I did a positive control in which I injected Cre-dependent crimson in the substantia nigra of that Cre mice. So now I have crimson expression in dopamine cells in SNC. So the key difference between these two is that here I am stimulating terminals from DMS, and here I'm stimulating cell bodies in SNC of dopamine cells. But in both, I am recording dopamine transients in DLS. And this was a good sanity check because I did find a nice dopamine transient in DLS after activating dopamine cells in SNC. So, this is actually a bit confusing because the circuit seems to exist, but doesn't work the way we predicted. Why not? So one possibility is that the effect of this polysynaptic circuit is masked by the simultaneous activation of this direct connection between DMS and SNC, and these two pathways cancel each other out. Another possibility is that this polysynaptic circuit increases tonic firing of the dopamine cells, but doesn't cause them to burst. And it's unclear whether increased tonic firing translates to dopamine release in striatum. So maybe you need coincident excitation or maybe even coincident modulation of release of the axon terminals in DLS to unmask the effects of this polysynaptic circuit. And if you do need any extra inputs encoding saliency or value, those would not be present in the open field experiment I performed. So I hope I have convinced you that there's a lot we don't know about this circuit. And before we fully rely on it as a mechanism behind habit formation, more experiments needs to be done. In fact, my ex vivo data is the first functional evidence supporting the existence of this circuit, but my in vivo data suggests that it doesn't work the way we predicted. So, I hope we don't go into a second lockdown so I can continue to do experiments and figure out what we're missing. And with that, I want to thank you all for watching. Thank you, NeuroMatch organizers and all these beautiful people and funding sources.
Thank you so much, Priscilla. That was a very nice talk. Are there any questions by either the panelists or the audience? Please submit them. So when you showed the depiction of the pathways, it seemed like that there might also be a pathway going the other way. Is that just an optical illusion, or can you comment on that?
Oh yeah, you're absolutely right. So according to the anatomical data shown by Haber, the anatomical data suggests that this ascending spiral exists, but not a descending spiral going from DLS to substantia nigra to DMS. However, I have collected some preliminary data that, let me see if I can jump slides here. Yes, so I have collected some preliminary data showing that this descending spiral also seems to exist. So if you just invert the injections of the viruses here in the retrobeads and you do the same experiment, I also found, again, this is preliminary results, but it seems to be lower probability of connection, but it's there.
So is it just an impression, or does it target a different portion of the substantia nigra?
Oh no, that's not an impression. That is real. So this was previously shown by my advisor that these DMS projecting dopamine cells are more medially located, and yeah, the DLS projecting dopamine cells are more laterally located. And you can also see that these DMS targeted versus DLS targeted SNR cells also have some topography in substantia nigra.
Right. So then if you have both circuits working together, how does that affect the interpretation of your in vivo results?
Good question, right? And if you consider the bridging collaterals, I mean, I don't know. It's complicated. I don't know. I just need to do more experiments to somehow, dissect all of these different pathways. The problem with basal ganglia is that it's one loop inside of other. It's just it's loopy. So that's actually a very hard question to answer. I don't have the answer.
Marie has another question. Marie, do you want to unmute yourself and ask the question?
Yes, great talk, Priscilla. So since there's all this topographical organization between DS and SN and DS, I was wondering, do you think there's like not only one ascending spiral, but actually many that might, like be specific to specific movements like turning, licking, and so on?
Yeah, it's possible. Again, these one thing that I have not looked at, but has been suggested to me, is to look who are these SNR cells that are DMS targeted, and it's probably not a homogeneous population. So it's very likely that you have all kinds of SNR cells here projecting to different places. So one experiment that I may or may not do, depending on time, is to check who are these. I don't have the final answer because everything's so loopy. But you're absolutely right. I do think you can have these different loops for different things that are part of this bigger ascending spiral.
Great. Thank you so much. For the sake of time, we have to stop our session here. If there is remaining questions, I encourage the audience to get into contact directly with the speakers. I'd like to thank Marie, Claudio, and Priscilla again for their very nice talks. I really enjoyed them. Thank you so much. And also for the audience for participating and listening in. And I hope to see you guys soon again. Take care.
Thank you.
And this is the focus on Alzheimer's disease. So we're going to talk about the syndromes, other syndromes associated with the disease, different gene expression in Alzheimer's-like disease, and some neuroinformatics approaches to study interaction between genes and this pathology. So I want to introduce our first speaker of the session, Pedro Clavel. He's an undergraduate student in Brian Phillips' lab at the National Institute of Pediatrics, and he's doing very fascinating work studying metabolic syndrome effects on olfactory function, memory, and neurodegenerative process in rat. And they recently published a related paper about metabolic syndrome that you'll be able to find the link to it in the chat. And finally, very exciting that Pedro, he's doing science communication. He's a part of a Spanish spoken science communication podcast. And Justo Medio, I apologize for my Spanish. And you'll in the chat, and you can also follow him on Spotify. So with that, Pedro, welcome, and please go ahead.
Thank you very much, Kathy, for the presentation. So let's begin. This is the title of my project, Effect of the Metabolic Syndrome on Olfactory Function, Memory, and Neurodegenerative Process in Rats. So Alzheimer's disease is a neurodegenerative condition characterized by a cognitive decline, mainly in memory function. There is evidence that shows that olfactory impairment, such as anosmia or hyposmia, is present in Alzheimer's disease patients, even years before the onset of classical symptoms as memory loss. Neuritic plaques and neurofibrillary tangles are the hallmarks of the amyloid precursor protein drives the neuritic plaques formation in the This is favored by the metabolic syndrome, which is a cluster of metabolic disturbances such as obesity, and specifically its core dysfunction and oxidative stress. So let's review more of that. The evidence shows that it promotes the amyloid pathway processing of the APP in neuronal cell cultures. In Alzheimer's disease transgenic models, a high-fat diet feeding, or a streptozotocin injection promotes the amyloid beta peptides accumulation, as well as overexpression of base-1 beta amyloid cleaving enzyme and full-length APP. Thus, this evidence shows that insulin impairment is present in the development of neuritic plaques. The evidence shows that amyloid beta peptides in the olfactory epithelium deposition is present at early stages of Alzheimer's disease patients. There is also evidence that shows that oxidative stress is present even months before the deposition of neuritic plaques in an Alzheimer's disease transgenic model. So part of this previous evidence comes from diabetic models with impaired insulin and glucose metabolism. But there is insufficient data whether if these alterations occurs in a metabolic syndrome model induced by a high carbohydrate diet, which exhibits insulin resistance, but glucose metabolism seems to be unaffected. So, I have some problems. So our hypothesis was that metabolic syndrome will generate behavioral alteration similar to those observed in Alzheimer's disease in olfactory and memory function, as well in its neuronal and molecular substrates. Our main objectives was to evaluate if there are alterations in olfactory and memory function in a model of metabolic syndrome induced by a high carbohydrate diet, and to assess whether insulin resistance and or oxidative stress alter the proteins involved in the formation of neuritic plaques in the olfactory bulb and the hippocampus. So for this investigation, we use male Wistar rats divided into two groups, a control group that received standard chow plus water ad libitum, and an experimental group which received 30% sucrose in their drinking water. This treatment lasted for 23 weeks. Olfactory and memory function were evaluated on week 21 and week 22. At week 23, somathometric and biochemical parameters were measured, and sample recollection was performed after euthanasia. Olfactory bulb and hippocampus were dissected to perform the following techniques: Western blot analysis, MTA determination, and histological analysis. So rats that received sucrose in their drinking water developed metabolic syndrome, since final weight, triglycerides, and adipose tissue were significantly increased. So this model is ideal to the early metabolic disturbances, since insulin resistance is present, as seen as in a hyperinsulinemia state, but glucose remains unaffected. Olfactory function was evaluated using the habituation and dishabituation task and the varied food test. In the former, rats are presented with consecutive olfactory stimuli. Habituation is defined by a diminished olfactory exploration for the same odor, and dishabituation is defined by an increase in olfactory exploration in the presence of a new odor. So both groups exhibit habituation and dishabituation. Both metabolic syndrome rats had a trend to shorter exploration times. In the varied food test, rats with metabolic syndrome took longer to discover the hidden piece of food. Thus, this evidence shows that metabolic syndrome rats have olfactory impairment. Memory function was evaluated using the novel object recognition test. In this task, rats are familiarized with two identical objects for several trials, A day later, one of the objects is replaced by a new one with different characteristics. So both groups prefer to explore the novel object more than the familiar object. Both rats with metabolic syndrome had shorter exploration times for both groups. Despite of that, rats with metabolic syndrome prefer to explore the novel object. So these results seems to indicate that rats with metabolic syndrome had a memory improvement. Next, we evaluated the APP and base-1 expression and insulin pathway activation in the hippocampus. Insulin pathway is hyperactivated, as seen as in higher phosphorylation levels of insulin receptor and AKT protein, together with lower expression of the APP protein and a trend to lower base-1 expression. So insulin pathway hyperactivation might account for the apparent memory improvement seen in the rats with metabolic syndrome. Nevertheless, none of these changes in protein expression and protein phosphorylation were observed in the olfactory bulb. So we propose that olfactory bulb might be one of the first brain areas to exhibit insulin resistance, since insulin receptor remain unchanged despite systemic hyperinsulinemia, in contrast to what we found in the hippocampus. Oxidative stress was evaluated using by lipoperoxidation, measured using malondialdehyde levels. MDA levels were lower in rats with metabolic syndrome in their hippocampus and tended to be lower as well in the olfactory bulb. Thus, likely, the metabolic syndrome likely produced an antioxidant response. Finally, we evaluated neurodegeneration was determined using the fluorogate-C staining in hippocampus and olfactory bulb. No apparent signs of neurodegeneration were observed in the hippocampus, and despite a trend to higher signal of this staining, the data dispersion was too high, so we couldn't find any differences in neurodegeneration markers in the olfactory bulb. So as a conclusion, we provide novel evidence that shows that olfactory impairment is present in rats with metabolic syndrome induced by a high carbohydrate diet. Memory was appeared to be improved in these rats, likely due to an hyperactivation of the insulin receptor, insulin pathway. And this together with lower APP expression and MDA levels. None of these changes were observed in the olfactory bulb. So we propose that the olfactory bulb shows early insulin resistance, and this could account for the olfactory impairment seen in these rats. So on insulin pathway impairment and amyloid beta deposition accumulation in the olfactory bulb merit further research. So finally, I want to thank everyone involved in this investigation, to my lab, the Laboratorio of Nutrición Experimental, as well as Laboratorio de Neurociencias and Laboratorio de Biología de la Reproducción from the Instituto Nacional de Pediatría, and I want to thank my friends. So that's all for me. I'll do my best to respond all of your questions.
All right, thank you very much, Pedro, for a wonderful talk. And it's a very interesting, comprehensive study that gives us some insights into the link between metabolic syndrome and olfactory dysfunction and Alzheimer's disease. So thank you again. And I encourage everyone to please ask questions.
So we have a few questions on YouTube.
Okay.
So I'm going to read it. Cynthia has asked, can you speculate why the imbalance on diet have an effect on olfactory function?
Yes, because the hyperinsulin, the insulin pathway is involved in many of the neuronal process as synaptic strain, protein expression that forms the synapses to form the synapses between the neurons. So we, as well as clinical evidence shows that metabolic syndrome and obesity have this olfactory impairment. We wanted to study the correlation between this alteration and the Alzheimer alteration that have also as well olfactory impairment. So I don't know the causal link, but we established that maybe insulin activation, insulin pathway might account for this olfactory impairment, but we didn't evaluate other options like free fatty acids or the influence of immune cells. But yeah, we found an apparently insulin resistance in this area in the olfactory bulb, and so we propose that this is implied in the olfactory impairment.
Thank you. She continues, sometimes it is speculated that olfactory malfunction predicts neurodegenerative diseases, but you show maybe before something at the metabolic, so you show something even before at the metabolic level is happening.
Yes,
So
yes,
prior.
yes. Olfactory impairment is being proposed as an early biomarker of neurodegenerative diseases, and because the interrelation between insulin pathway and the development of the Alzheimer's disease hallmarks as neuritic plaque formation. So we established as well as that this interrelation could be important to study these alterations.
Thank you. I hope she writes back if the answer was enough or she needs more.
So, yeah.
Okay, thank you.
Can you hear me?
The questions from the chat. So looks like we have one cancellation, which is the last talk, and the next panelist also didn't seem to show up. So we can have more time for the discussion of Pedro's topic. And Carla is saying nice talk. And I personally actually have a question myself. So I found it very interesting that you showed that rats with metabolic syndrome, they have improved memory in comparison to control rats. So can you talk a little bit about it? Why do you think that could be the case? Did we just lose Pedro?
I hope not, but he's not reacting.
Yeah, he's frozen. His video is frozen.
Oh, okay, he's sharing his also.
Yeah, looks like we lost him, right?
I believe so.
That's very unfortunate. I think a lot of people are experiencing troubles because of the different issues. Some people because of the hurricane, and that's probably partially why we don't have some of the attendees today, unfortunately. So yeah, if Pedro is not back, I think we will have to end our session, probably, unfortunately, which was very interesting to learn about the link between metabolic syndrome and Alzheimer's disease.
That's really unfortunate.
Yes.
Okay, we can. I thought we can go ahead. Yeah.
Okay, well, thanks for joining. Bye.
Thank you, everyone. Thank you for your questions and listening. Thank you.
Thank you.
Bye-bye.
Bye.
And just so you know, this is being recorded on cloud as well. If you would like me to send it to you afterwards, I can do that for you as well.
Thank you.
Okay, we're live on YouTube as well.
I don't know.
Yes, great. Okay.
Okay.
Hi, everybody. Welcome to the last day of NeuroMatch. I hope you've been enjoying the meeting. I'm Robert Fremke at NYU, and it's my pleasure to introduce Ioana Karsia from Rutgers here to give a talk today. So Ioana is an MD PhD, and she did her PhD work at Mount Sinai in Deanna Benz's lab. And then she did a postdoc in my lab. And I sort of lost count. I just looked this up on PubMed. Ioana and I wrote a total of 10 papers together and counting. And what she's gonna be talking about today is paper number 11. She's had her own lab for about a year now or so. So take it away, Ioana.
Okay, sounds great. Thank you, Rob. Let me share my slides. Okay, this is good, right? You can see them? Okay. So hello, everybody. Thanks for being here. Today, as well, Rob didn't mention the title, but I will be talking to you about our work on how maternal behavior can be socially transmitted in mice. We all know that maternal behavior is essential for survival of the majority of mammalian and avian species. And it is such an important behavior, in fact, that in many cases, unrelated conspecifics contribute to caring for the young. These individuals are called alloparents or non-biological parents. And they have been described in more than 130 different species, including primates, whales, dogs, and rodents. Rodents, and in particular mice, are a great model to study how alloparenting behavior develops. Mice in the wild live in colonies, generally composed of one male, a couple of reproductive females, and several non-reproductive females, who are, in general, daughters of the colony that stay around to help caring for new generations of pups. So how can we best measure mouse maternal skills in the lab? One robust test for this is the pup retrieval behavior. So here, what we do, we isolate a pup from the nest. And when isolated, this pup will start emitting distress calls in the ultrasonic range, like the example call I'm showing you here. When hearing these calls, the mother immediately, within seconds, locates the pup and picks it up, retrieves it back to the nest. It has been shown previously that this behavioral response depends on the activity of the left auditory cortex, where these pup vocalizations are primarily processed. Importantly, virgin females that have experience with pups can also retrieve pups. And when oxytocin, a hormone which we know to be heavily released in mothers during parturition and lactation, when this hormone is given to virgins, either systemically or topically in the auditory cortex, the left auditory cortex, these animals can also start retrieving pups. On the contrary, inexperienced virgins or animals where the oxytocin activity has been decreased do not retrieve pups. So what we see here is that experience and oxytocin go hand in hand when it comes to pup retrieval behavior and then more generally to maternal behavior. So we wondered then if experience could use the endogenous oxytocin system to induce alloparenting behavior in virgin female mice. Before I go into how we address this question, I want to tell you a little bit more about what I mean by experience. Traditionally, virgins are allowed to gain experience with pups by co-housing them with a number of pups for hours or days. More recently, we started co-housing the virgins not just with pups, but also with the mother of those pups. So one of the first things we did is to test how these two different types of experience can impact alloparenting skills in the virgin, and in particular, pup retrieval skills. So here I'm showing you a graph where we plotted the probability of virgin retrieving on the y-axis, and on the x-axis is the time in days of co-housing. What you can see here is the probability that virgins retrieve after being co-housed just with pups. And you can see that they do start retrieving pups. However, when we co-house virgins with the dam and pups, we see that the acquisition of this pup retrieval behavior is significantly accelerated. So virgins start retrieving much sooner. And the question then is, how is this socially mediated acquisition of pup retrieval behavior induced? What exactly happens during this co-housing experience? So to answer that, we took three approaches. One was to look at behavior, particularly or specifically interactions between the virgin and the dam. Secondly, we wanted to understand how these interactions might modulate the activity of the oxytocin system, which is the main neuromodulator that we know to be involved in maternal behavior. And lastly, we wanted to understand if this co-housing interactions might change the representation of pup calls in the auditory cortex. So to answer this, we built a system for continuously monitoring the interaction between animals throughout the co-housing procedure. So the system has a camera that acquires continuous video recordings, has an ultrasonic microphone for picking up pup calls, and a neural activity acquisition system for recording the activity of oxytocin neurons in the virgin hypothalamus. So with this system, we acquired days of video recordings, and we first analyzed those to look for interactions between them and virgins. And we indeed identified two behaviors that I will describe, which we believe to be important in the acquisition of alloparenting skills by the virgin. So the first one of these two behaviors is the fact that the dams direct the virgins towards the nest. So I'm going to show you an example in this video, and what you'll see here is the nest with pups. The dam, you can see, is already emerging out of the nest, and she's headed towards the virgin, who is in a different location of the cage. And then you'll see the mother directing the virgin towards the nest. So the mom comes out, and then she pushes the virgin towards the nest. She will join the virgin in the nest subsequently. We see this kind of spontaneous interaction occurring many, many times during the co-housing procedure. And we wanted to look at the spatial organization of this interaction. So here I'm showing you a representation of the co-housing enclosure. The white circle represents the location of the nest, and the yellow pixels represent the probability that the mother will start chasing the virgin at that location. As you can see, the start points are, the vast majority of them are outside of the nest. However, when we look at where this chasing ends, we see that almost exclusively the chasing ends within the nest. So we find that this chasing that's directional from outside of the nest towards the nest, which we call shepherding behavior, occurs about 85% or more of all chasing done by mother. So what are the behavioral effects of shepherding? What might they be? Well, obviously one of them would be that the virgin ends up spending more time in the nest. And that's indeed what we see when we quantify the percent of time spent in the nest by the virgin over the course of cohousing. But importantly, we also see something quite unexpected, and that is the fact that the frequency of shepherding during the first day of cohousing correlates with the probability that the virgin will start retrieving pups at the end of the first day of cohousing. And this is a very strong correlation. So this suggests to us that the more the virgin is shepherded, the more likely she is to retrieve pups consequently. The other behavior that, before I go into the other behavior, just to summarize the fact that we see the shepherding occurring many times throughout the cohousing procedure. I didn't show you, but it depends on the presence of pups in the nest. And I showed you data indicating that shepherding contributes to the acquisition of alloparenting behavior in virgin. So the other behavior that we found is that the dams self-generate maternal retrievals during cohousing. You will see in this example video that I'll show that sometimes when the mothers come out of the nest, they drag accidentally one or more pups with them, and then they return to retrieve those pups. So you saw the pup coming out, being dragged out by the mother, and then the mother returns to retrieve it back to the nest. And the virgin is present in the nest and has the ability or the capacity to observe what the mother is doing. So we wondered, given that the virgin has this possibility repeatedly during cohousing, is it possible that virgins can learn pup retrieval by observing the mothers doing this behavior? So to answer that, we first wondered if there is a correlation between the frequency of dam retrieval during the first day of cohousing and the probability that the virgins retrieve at the end of that day. And again, we find the correlation here is not as robust as the correlation with shepherding, but it exists. Furthermore, we then isolated this interaction from the context of the cohousing behavior. And so now we repeatedly exposed virgins to maternal retrievals in series of 10 for a total of 4 days. So each day the virgins observe 10 maternal retrievals, then they get a break, and then they are tested themselves for pup retrieval. So what I'm showing you here are 15 virgins that have been exposed to this paradigm. And in green, you see the probability that they will start retrieving pups. As you can see here, the vast majority of virgins start retrieving pups sooner or later during this procedure. Now we wondered what might be the sensory modalities that contribute to the acquisition of pup retrieval by observation, by being exposed to dam retrievals. So to start answering that, we first wanted to block the touch interactions between the virgin and pups and also between virgin and dam. So to do that, we placed a transparent barrier in the cage that allowed visual, auditory, and olfactory information to still cross across this barrier. So when we do this, we see again that many of the virgins, again, the majority of them, start retrieving pups after being exposed to maternal retrievals via the transparent barrier. However, if we make this barrier opaque, blocking visual information, now we no longer see robust acquisition of pup retrieval behavior by virgins. In fact, only 2 out of 18 animals showed pup retrieval behavior after the opaque barrier condition. Yes. So this indicates that there is a visually guided observational learning of pup retrieval behavior that might occur during co-housing. So I showed you that both shepherding and observational learning might be behavior interactions, spontaneous interactions between adult animals that allowed the virgin to behave as an allo parent. And now what we wanted to address next is whether these behaviors can use neuromodulators, in particular the oxytocin neurons, in order to contribute to the acquisition of pup retrieval behavior. So to start testing that, we first wanted to know whether the activity of oxytocin neurons in the paraventricular nucleus of the hypothalamus is required for acquisition of pup retrieval during co-housing. We focused on the PVN, the paraventricular nucleus, because this is where oxytocin neurons that project centrally to other brain regions are located. So what we did here, we induced the transduction of an inhibitory dread, specifically in oxytocin neurons. And then we administered CNO to these animals, to the injected animals, over the course of co-housing. And we found that in this CNO condition, there is a decrease in the acquisition of pup retrieval behavior compared to control conditions. So this indicates that the firing of oxytocin neurons in PVN is required for acquisition of pup retrieval behavior during co-housing. Now, if shepherding and observation of dam retrievals are indeed important for acquisition of pup retrieval behavior, then we would expect that they are able to activate oxytocin neurons based on this result. To test this, we decided to do single unit recordings in the PVN. We implanted an electrode bundle in this very small structure, and we used co-registered MRI and computer tomograph scanning to verify the proper location, the correct location of these electrodes. Moreover, to specifically record the activity of oxytocin neurons, we induced the transduction of channelrhodopsin in these cells, and then we used blue light to determine which neurons respond robustly and with short latency to this blue light laser. So those neurons that respond in this way are going to be bona fide oxytocin neurons, and the rest of the neurons we're going to name unidentified PVN neurons. So let's look at how these neurons respond to the behaviors I described earlier. Here's a cell recorded in PVN where the spike trains have been aligned to the onset of shepherding. You can see the different shepherding episodes highlighted here in red. And as you would expect from a spontaneous behavior, they have different lengths, and they sometimes occur with different frequencies, sometimes closer to each other, sometimes further apart. We looked at the average firing rate during the shepherding events and compared that to the firing rate during the preceding baseline. And what we see is that this particular neuron is strongly activated during shepherding behavior. Overall, when we look at all neurons that we recorded, we find that 19% of oxytocin, bona fide oxytocin cells, respond to shepherding by being activated by this. And by comparison, only 12% of unidentified PVN neurons are activated by shepherding. Interestingly, neurons that do respond to shepherding only respond to this type of event and not to another event that is similar in terms of motor characteristics, but not dissimilar in terms of the social motivation. So here we see that neurons that respond to shepherding do not respond to events when the virgin is pursuing or chasing the mother. As I mentioned earlier, shepherding is followed by the virgin entering the nest. So we also wanted to see how oxytocin and other PVN neurons respond to nest entry. Here I'm showing you 9 units recorded simultaneously in PVN. One of them is a bona fide oxytocin cell in blue here. And what we can see is that several of these neurons, including the oxytocin neuron, has increased firing after the virgin enters the nest. And that is more clearly shown in this peristimulus time histogram here, histograms for example cells. So overall, we find that a similar number of oxytocin and unidentified PVN neurons respond to nest entry. About 33% of them are activated by nest entry. Importantly, this happens when the nest entry follows shepherding, but also when the nest entry is voluntary. So this indicates that there might be cues related to the nest, either visual, tactile, olfactory, that generate a response of oxytocin and other PVN cells. How about observation of dam retrievals? Here I'm showing you 4 units that again were recorded simultaneously in virgin PVN, while the mother retrieved a pup during cohousing. The spike trains are aligned to the onset of the retrieval event, and you see that preceding that we have an interval where the pup vocalizes. It emits distress vocalizations. And what we can see here is that, again, the oxytocin neuron in blue here and some other PVN neurons respond to the onset of dam retrieval. So here I'm showing you that almost 30% of oxytocin cells are activated by observation of dam retrieval, and by comparison, only 17% of unidentified PVN neurons respond to this behavior. So I described these 2 spontaneous interactions, shepherding and observation of dam retrievals, and then we showed that both of these are capable of increasing the activity of oxytocin neurons in PVN. I should mention here, although I didn't show the data, that we have control behaviors that occur during cohousing, which we don't find to activate oxytocin or other PVN neurons. For example, entry into an empty nest is not capable of activating the cells to the same extent as entry into a nest with pups. All right, so then let's see what are the effects of oxytocin neuron activity and PVN neuron activity on the processing of pup calls in the auditory cortex. Previous work in Rob Fromke's lab showed that there is a really striking difference in how the auditory cortex of mothers and virgins, inexperienced virgins, process pup calls. So here we see a very robust response in the auditory cortex of mothers in response to playback pup calls. By comparison, the virgin auditory cortex does not robustly respond to playback pup calls. This increased responsiveness of the auditory cortex is important for pup retrieval. When the auditory cortex is silenced using mucimol, we see that there is a decrease in the success of pup retrieval behavior compared to pre- and post-activation conditions. More recently, Jennifer Schiavo, also in Rob's lab, showed that there is a transition from low responsiveness to high responsiveness in the auditory cortex of virgins during the cohousing behavior. So we wondered if this transition is mediated, at least in part, by activity of oxytocin and other PVN neurons, then we should expect to see a correlation between neuronal activity in the 2 structures. And indeed, this is what we observe here. I'm showing you dual recordings in the auditory cortex, where we did fiber photometry recordings, and in PVN, where we did single unit recordings. And we did these dual recordings during the observational learning across a transparent barrier. And what we can see here is that there is a correlation between the activity in PVN and the activity in the auditory cortex, which is robust and significant, as you can see here. Now, given this, we then wanted to know if blocking the activity of oxytocin in the auditory cortex can impair pop retrieval behavior or the acquisition of pop retrieval behavior by observation. So again, here we did the observational learning with a transparent barrier, either with animals where we injected saline in the auditory cortex, or animals where we injected an antagonist for the oxytocin receptor in the auditory cortex. So again, importantly, compared to control conditions, we see that blocking the oxytocin receptor impairs the acquisition of pop retrieval behavior by observation. So overall, I showed you that the spontaneous behaviors that we identified during co-housing can engage the endogenous oxytocin neurons in the PVN in virgin females, and that this activity might be important for controlling the representation of pop calls in the left auditory cortex of virgins. So with this, I would like to thank my lab and also many people in Rob's lab. I'm just mentioning some of them here, but there's many more that contributed to this project. Also collaborators at Rutgers, where I am now, and at other institutions. I would like to highlight John Cunningham, with whom we're collaborating now on automating the analysis of video recordings, and Justin Reisberg from Mount Sinai, who's done a lot of work on the neural analysis and continues to help us with that. Okay, thank you. And I will take questions now.
Okay, great, thank you. So we have several questions. Have you tested if virgins that didn't retrieve pups with the opaque barrier testing then would retrieve pups with the transparent barrier?
So basically use the same virgins in the two conditions. We haven't done that. One thing that might speak to this experiment, should I stop sharing? No, I'll keep going. So one experiment that we do that could speak to this is we are now currently doing an experiment where we activate oxytocin fibers that project to the auditory cortex during the opaque barrier condition experiment. And so basically what we're trying to see, if we artificially induce the activity of oxytocin neurons, particular oxytocin fibers in the auditory cortex, during this paradigm that generally is not effective, can we then increase the possibility that these virgins learn to acquire pop retrieval? And that's what our preliminary data shows. In fact, out of all virgins that we tested so far, all of them learned to retrieve pups with the opaque barrier as long as we activate oxytocin fibers in the auditory cortex.
And what about blind virgins? You think blind virgins, what mechanisms do you think they have to learn to retrieve?
Right, we haven't done that. I mean, it's a little bit tricky, and some decisions would have to be made as to when we want to blind these animals, right? Do we want to blind them immediately after birth so that they never learn to rely on visual cues, or do we blind them as adults, in which case they might behave very, very unpredictable, given that one of their sensory modalities is all of a sudden gone. We don't even know what to expect from their, motor activity, exploratory behavior in the cage very much. So we haven't done those experiments. I think it would be good to do them as long as we can figure out the exact timing for when we should blind these animals.
So about the oxytocin neuron recordings, what are your criteria to define if a neuron is being activated or suppressed?
Activated or suppressed. Yes. So here we, calculate the average firing, or for each trial, we calculated the firing rate during the event and compared that to the baseline. And we do permutation tests to see, if we were to move these events in time, would we be would this difference in firing rate be above, 95% of what we would get with all permutations? So that would define activation, and inactivation or suppression would be lower than 5%.
How homogeneous or heterogeneous is the oxytocin neuron population? Is it the same population that's activated during shepherding and nest entry, or are they different populations?
We do have the data. There is some overlap, but not much. So it's interesting, and it seems to be the case that it's mostly different populations that respond to probably different cues, right? We would expect that shepherding, nest entry, observation would all have different or rely on different modalities to reach the oxytocin neuron population. And we do see that they're largely non-overlapping.
So you showed data on the dread-based suppression. But what about after animals have already learned? If you inhibit or block oxytocin neuron firing, does that impair execution of pup retrieval?
We did that experiment as well in virgins that already acquired the behavior, and we don't see the same effect with chemogenetics. Now, there might be a trend, but it's mild. Now, I don't know, perhaps in mothers that would be different, perhaps with more animals this would be different, but we didn't find a significant difference in virgins that already acquired the behavior.
Follow-up question to the question about blind animals and the need for visual input. Basically, can you clarify how you're thinking about it, what the hypothesis is? Is visual input required for oxytocin release, even in auditory cortex? And oxytocin plus the sounds of pup calls that required for successful retrieval, either behaviorally or retrieval learning initially?
Yeah, so the way I think of this in the context of the observational learning paradigm, I do think that the visual input is required for increasing the activity of oxytocin neurons that will result presumably in the release of oxytocin in the auditory cortex. And that in turn will induce a stronger representation for pup calls in this brain region, which is necessary for pup retrieval. So yes, I think this would be the mechanism pretty much in the case of the observational learning. And, again, I would say that the fact that we can replace the visual input just with activating oxytocin neurons, oxytocin fibers, I think it's further proof that this would be, if not the mechanism, at least one of the mechanisms. Now, of course, the mechanism, as I said earlier, would be different when we think of oxytocin cells being activated by shepherding or by nest entry. But yeah, in the case of observational learning, I do think the visual input is key.
Maybe on that note, you could talk a little bit more about the specific aspect of the visual input. Oxytocin clearly enhances sensory responses to pop cues, like pop calls. But what about potential imitative parts of learning? If, for example, do you think there are changes or activity elsewhere, for example, in M2, higher motor cortex areas that are also important?
I think this is a fascinating question, and we haven't looked at that. I think we should, in fact, look at that aspect. But in terms of how much this is imitation versus, object enhancement or location enhancement, or, some people might go as far as emulation. We don't know that. I mean, imitation is probably harder to argue, right? Because each pop retrieval is by design going to involve a different motor sequence, right? Like the location of the pipe is not going to be precisely the same for every trial. The position of the adult animal is not going to be the same for each trial. That's up to them where they want to be in the cage. So saying that this could be imitation is probably not, in the stricter sense, is probably not likely to be imitation. Yeah, just because we do not see the same exact motor sequence in the mother and the virgin. Now, having said that, I don't think we should, exclude the possibility that the motor cortex is involved in this. In fact, I do believe that plasticity in the motor cortex is probably a big component of this behavior as well, right? I mentioned, I think in the beginning, that mothers very quickly detect the isolated pup and move swiftly towards it, pick it up without a problem. Now, like, the virgin doesn't do that. The virgin takes a winding path to get to the pup, if she goes there at all. Then we have some studies on errors, and we find that they initially grab the pups in weird, different ways. They grab it a little bit and then drop it, and they're very awkward in their motor skills, and that refines over time. They become experts over a relatively short period of time. Okay, so I do think there is plasticity in the motor cortex, and I do believe that we should look at that as well. But I would not necessarily expect to see, the classical motor mirror neurons or anything like that, just because the paradigm is not designed to show those neurons or to highlight those neurons. I didn't mention this talk, but we do see sort of mirror neurons in PVNs. So in PVN, so neurons that are activated by both observing a dam retrieving and by the virgin executing that retrieval. It's not the exact sequence of activity, but the same neurons are activated by both of these. So in that sense, there is sort of like a social maternal cell population in PVN, or if you want, mirror maternal neurons in PVN.
Okay, have you looked at markers of plasticity in oxytocin neurons over time? Maybe CFOS or other things might indicate a change in the system.
Over time, like during co-housing or during the, we are now using the TREP2 animals to look at FOS activity in different behavior settings. We don't have yet an answer on that. But bottom line, no, we didn't do much of that. I believe actually Silvana in your lab, Rob, is working on something related to plasticity markers. So she would have a better answer to that, but I haven't looked.
Okay, thanks.
Okay.
That was great. A lot of questions. Thank you, Ioana.
Thank you, everybody.
Thank you to everybody who showed up, those of you who asked questions. And with that, we'll close the session.
Okay, thanks. Bye.
Bye, everybody.
And, yeah, I will ask them to send their questions to Q&A and I will read them to you. I think we are going live now.
Okay, I guess we can see the Q&A ourselves.
But I will read them so everyone knows, and then
Wonderful.
You can also read them too. Thank you.
Okay, we are live on YouTube now.
Ooh. It may be my first time or second time on YouTube.
Okay, great. And we need to wait for the attendees to join. Like, and the session starts at 5, so it will take a couple of minutes for them. I think we are all in the same time zone, right? Or no?
No, definitely no.
Because so you're in Central Time?
Yeah, I'm in the Central Time, but Trey is not.
Yeah, I'm on the Eastern side. Yeah.
Oh, okay. And Pennsylvania is also in Eastern Time, right? I think so. Okay, we're getting attendees. I think. Okay, we wait 1 minute or 2. Okay, that's a start. Hello, everyone. Welcome to this session. I'm the MC of this session, Yalda Mohsenzadeh, and I'm happy to introduce our speakers today. So we have 3 interesting talks on working memory and also its interaction with visual and processing and perception. I'm like you, I'm excited to hear these interesting talks. Let's start with our first speaker, Jiangang Shan from University of Wisconsin Medicine. He's a graduate student, and you can start your talk, Jiangang.
Yeah, thank you. Let me share my screen. Can you see it?
Yes.
Great, wonderful. So hi everyone, I'm Jiangang Shan from University of Wisconsin Medicine. I'm happy to share the findings of our recent work, the influence of active removal from working memory on serial dependence to you. So in a simple one-item recall task, the item in each trail is randomly selected. So at the end of each trail, the item in that trail will be irrelevant to the task and is discarded by the subject. But most of studies have shown that the loss of information from working memory at the end of each trail is not always very perfect. So the recall report on the trail end here is often found to be attracted to the value of the memory item from trail n-1. So this effect is called serial dependence. Serial dependence indicate that there are residual neural traces of the irrelevant memory item, or IMI. Indeed, researchers found the orientation of sample in trail n-1 could be decoded from EEG data after the onset of the trail n sample. And with monkeys, there's an activity silent representation of the n-1 sample location in the intertrial interval, and a reactivated representation near the end of the intertrial interval. And there is similar reactivation in EEG of humans too. So the passive loss of working memory may be implemented by the withdrawal of attention, leaving the trace of IMI there. On the other hand, in tasks with more than one item holding working memory simultaneously, competition may occur in stimulus representations being held simultaneously rather than items from trail n-1 and trail n, especially according to the interference model of working memory. When multiple memory items are bound to and retrieved by similar contexts, interference between them reduces the working memory of it. For example, like here, in a trail with only one orientation to memorize, the orientation is the content of working memory, and it is associated with its spatial context. But in this trail here, there are two orientations shown at two locations. So these two, so they should be related to their respectively spatial context, like this one. But if the two orientations are shown at the same location, there should be some strong interference. Sorry. Yeah, some studies have found that people could actively remove IMM from working memory when multiple items are maintained. For example, in an ideal serial retroqueuing task, a single pulse of TMS cannot reactivate the representation of IMI or increase the false alarm rate of it, but it can reactivate and increase the false alarm rate of an item that is not removed yet unattended by the subject. There are several works on the possible mechanisms of the active removal. The interference model says that the active removal is implemented by unbinding the to be removed item from its spatial context, like this one. Another more intuitive mechanism is the suppression of the neural representation of the IMI, like this one. So in this study, we want to see the mechanism whereby an item in working memory can be actively removed and how it may be different from the passive loss of working memory or withdrawal of attention. So to do this, we tested the serial dependence of recall in a one-item recall trial on the actively removed or passively lost IMI from the N-1 trial. So a passively lost IMI should attractively influence the report in the next trial, but an actively removed IMI should cause no severe influence if the removal leaves no residual trace of it, and it should has an attractive effect if the removal, for example, the unbinding operation is not relevant to the representation itself. So like in this study, in order to evoke the active removal and the withdrawal of attention, we made a novel task that involves a retro queue and 3 memory items. We call it the ABC retro queuing task. So in this task, first, there are 2 orientations shown on the screen, and the subject should memorize their orientations. After this period, there's a retro queue indicating that this item was going to be this item is could be asked to be sorry, I'm going to be nervous right now. So this retro queue here just indicating that this item is relevant to the task, and the other one is not going to be tested at all. So in this case, the item B here is the irrelevant memory item or IMI, and the other item, item A here, is the retro queue item. So after this queue, the third item, item C, was shown here. So there's a 50-50 chance that either item C or the retro queue item would be tested at the end. At the end of this trial, there's a recall period. A circle was shown on the screen, and the subject should recall the orientation shown at this location. Importantly, there are 2 conditions. In the no overlap condition, item C's location was randomly selected from the 4 locations that had not been occupied by A or B. In the overlap condition here, item C always appear at the same location as the IMI. Before the start of each, the subject were told the subject were told which condition this is going to be. So like so in the no overlap condition here, the subject had no expectation of interference for IMI and item C. IMI and item C, and IMI could be passively lost through the withdrawal of attention in this period. But in the overlap condition here, the expectation of interference is high, and subject should actively remove IMI after the retro queue. So this removal is the unbinding of the context of IMI. So in this experiment here, there are interleaved ABC retro queuing trials and 1 item recall trials. And to test the serial effect, we tested the influence of IMI on the recall of the following 1 item trial for 2 conditions separately. We also see the serial effect of the retro queued item as So we used a DOG function to model the serial dependence effect. X here is the orientation of IMI or the retro queued items, minus the orientation of the sample in the following 1 item trial. And Y here is the error of recall in the 1 item trial here. The recall in the 1 item, for example, here, when, for example, IMI is 25, I'm so sorry. I'm really nervous right now. So for example, when IMI is 25 relative to the sample in the one item trial, the recall is attractive to somewhat 1.5. But if IMI is minus 25 relative to the sample in the one item trial, the recall is somewhat minus 1.5. And there's also the repulsive influence. The function will look at something like this one. So we use the peak to peak to measure the magnitude of the effect. It is this thing here. We let it be positive if the bias is attractive and negative for repulsive bias. And here is the result. As you can see, for the retrocute item, we find attractive effect on the no overlap and overlap, similar to the end of trial loss of working memory. For the IMI, we found attractive bias in the no overlap condition, consistent with the withdrawal of attention. But in the overlap, we found that the pattern inverted and IMI has a repulsive influence on the next trial. So how are we to understand this repulsive effect? If the active removal is implemented by the unbinding, there are two possible outcomes. If the serial dependence effect is influenced by the representation of IMI itself, and unbinding did not influence the representation, there should be an attractive effect here. But if the effect is about the relationship between the content and the context, there should be no effect here because the unbinding already removed it. But on the contrary, we found residual trace that is like this one, a repulsive effect. This indicates that there is some residual trace of the actively removed working memory, but it's very different from the memory item that is the result of the withdrawal of attention. Okay, to summarize, we found an attractive effect for the serial dependence of IMI in a no overlap. But we found an repulsive bias in the overlap. This result is inconsistent with the unbinding hypothesis. It indicates that the active removal leaves a residual trace of IMI, and the residual trace is very different, maybe inverse. Maybe subject just remove working memory through the suppression of IMI. That's all I got for this talk. Thank you, and I'm happy to take any question.
Thank you so much, Zhenggang, for the great talk. We have one question in the Q&A, and I don't think we have more than that to handle. This is a question from Suri Charan, and the question is, first, nice talk. And then the active removal condition assumes that item A was coded in working memory, even though it was irrelevant, not retrocute. Did you test if this was the case?
I see. Let me go back to the task. So in this case, the IMI here is not irrelevant. We know it is in the working memory. After the retrocute here, it was removed out of working memory because the subject are not sure which item are going to be retrocute here. So it must it must be encoded in working memory first. And then we assume it is removed by the subject.
Okay, thank you. So there's a question about, like, is there a paper out for this work?
We are doing a replication of this work. So hopefully it will be out by next year.
Okay, great. Okay, thank you so much.
Thank you.
Thank Yang for the great talk. Our next speaker is Shekufe Hedayati from Penn State University. Shekufe is a graduate student, and we're excited to hear her talk.
Yep, thank you so much. Really glad to be here. Let me share my screen. Let me just. Okay. Hello, everyone. Again, my name is Chico. I'm going to talk about a project that we've been working on for about a year. Its title is Encoding Complex Shapes in Memory, the VAE-BP model of working memory. However, as we've been working on this, we came up with a new title. So I guess my animation is not working. Sorry about that. Is it because it's in the presentation mode? Oh, it's working now.
Please share your screen. We don't have your screen now.
Okay. Okay, let's do it this way. Okay, great. Now it's working. Do you have my screen? Okay, so as we've been working on this, we came up with a new title that is TLDR, Transforming Latent Descriptors in Memory Representations. So whenever I refer to TLDR, I mean the model that I'm proposing here. So current models of working memory, the questions that they're concerned with is about what is the capacity of working memory, how many items can be represented in working memory, how this precision varies, if the set size increases, how are features bound. However, what is missing is that most of these models do not use real-world stimuli. So they use things like colored square or oriented bar rather than a more ecologically valid stimuli that we're going to use in our model. And also the importance of visual knowledge has not been paid attention to. So these models do not really allow for encoding of familiar versus unfamiliar items, the things that we draw from our visual knowledge and the things that we haven't seen before. The importance of visual knowledge has already been considered in more descriptive non-computational models. For example, a multi-component theory, Battle and Hitch, they had episodic long-term memory and visual semantics as subsystems of the working memory, and also the activated long-term memory theory says that working memory is nothing but an activated part of the long-term memory. And also the importance and the impact of visual knowledge in working memory has already been shown in neural and behavioral experiments. For example, Chinese reader can remember more Chinese characters than non-Chinese reader or German readers, for example, as you can see in Zimmer and Fisher data. And also there are brain evidence showing that regions responsible for working memory, they show reduced bold activity when items are familiar versus when they are unfamiliar. So here we are going to propose a computational model, TLDR, that accounts for how visual knowledge impact working memory encoding. What are the behavioral characteristics of TLDR? It is reconstructive. So if we show people this image, they're able to draw that image from their memory, as Carmichael did in 1929. the model can focus on relevant attributes. For example, there was a study showing that if people are shown with an oriented arrow with a color, and they were asked about its color for 25 trials, because they haven't been focusing on its orientation. In a surprise trial, when they're asked to report the orientation, they don't have a good memory for that. So their memory for orientation is poor. And also, the working memory can remember novel stimuli. So as Lake and colleague did, they showed people different stimuli from different characters from different language that people were not familiar with and haven't seen that specific configuration before, but people were able to sort of draw that characters from their memory with some variations. What are the neural plausibility of the model? It has rapid encoding and rapid forgetting, which is one of the important characteristics of all working memories, because it's been shown that working memory is, the information working memory is storing activations rather than synaptic weights, and that allows for rapid encoding and removing information. Also, it's consistent with the structure of the visual system. As we go higher in the ventral stream from V1 to B2 to IT cortex, the representation becomes more complex and abstract. And also visual knowledge part of the model is formed through synaptic weight adjustments. So just how human visual system is being trained through adjusting of synaptic weights by seeing a lot of visual examples throughout their lives. There are two important elements of the TLDR. One is the visual semantic one, or the variational visual knowledge one, or the variational autoencoder, or I call it VAE, that has an encoder and a latent space and decoder. So in the encoder, when image comes in, it's encoded into the model, and then there is its compressed form. It's represented in the latent space, and then the decoder learns to decode that information and reconstruct it in the output. And that was proposed by Kim and Welling in 2013. That's going to be the visual knowledge part of our model. However, we did some changes to this original model. And also a binding pool, which was a memory model first done by Swan and Weibel in 2014. So we need to have a binding pool that allows for encoding information, because the VAE itself cannot hold multiple objects. This is the memory part of the model that can encode multiple objects, and then they are connected together in the way that I'm going to talk about more details. The binding pool component, it has a type layer that represents high-level features. For example, for this oriented bar over here, it has a color and location and orientation that are represented in the type layer. There is a token that indexes and individuates objects and can keep track of multiple items. Without tokens, we cannot store more than one item or retrieve more than one item in the binding pool. And the binding pool layer is the shared resource pool that encodes the information. And the variational autoencoder, as I said, we made some changes to it. So we trained this model on shape and color separately. We divided the latent space to shape and color maps so that we can represent shape in the shape map and color in the color map. And the reason to do this was to sort of being able to focus on certain attributes and store them into the memory, to be able to implement a top-down control and decide which attribute should go to working memory, because working memory is kind of selective to that. So this is the architecture of the whole model and the way that interacts with the binding pool. So the upside is the visual knowledge, or the VAE, modified VAE, and the bottom is the binding pool. Imagine that this image, handwritten for, comes into the model. It generates some activations in the layer one, and this activation is projected through fixed random weights to the binding pool. And then again, with an inverse multiplication, it goes back to the L1 and is reconstructed through the model. And this is how the reconstruction is. And again, we tested this with different layers with L2. So L2 projects to the binding pool and goes back and reconstructed, also color and shape maps. And we wanted to see which one has a better, efficient reconstruction. Just talking about more about the details, the stimuli that we used were MNIST digits. We colorized them with 10 different colors. So we ended up having 10 categories of digits from 0 to 9 and 10 categories of colors, red, yellow, blue, etc. And to train the model, we trained the shape map with a shape objective function that minimizes the loss on gray images, and for the color map, we trained the model on using color objective function that minimizes loss on uniform color patches. And at each batch training, only one of these objective functions were active. And then later they were combined, these shape and color information are combined in the later levels. So for example, this is an example of the training procedure. So these are the MNIST images that are going to the model. And then these are the reconstructions from both maps. As you can see, this is the reconstruction from only the color map. So if I set the shape map to 0, then this is what I get from the color map. So it's going to be color of the original images with no specific shape information. You get just a bunch of 5s. And then this is how the reconstruction from shape maps looks like. So you have the shape information with no really color information. So we kind of separated these representations in the latent space. But how the memory retrievals look like. So these were just reconstructions. So imagine that I have only WS active over here, meaning that I project only shape map activations to the binding pool, encode actually shape activations to the binding pool, and I reconstruct them. And this is how the reconstruction looks like. This is the memory retrieval from shape map. And this is the memory retrieval from color map. And this is going to be the memory retrieval from both maps at the same time. Also layer 1 and layer 2. So for L2, this is the memory retrieval from L2, and this is the memory retrieval from L1. They do not have good resolution as the latent space. And we also tested this quantitatively. We had 4 radial basis function kernel SVMs that were trained on the latent space. SVM-SS were shape map activations trained on shape labels. And the accuracy for the memory retrieval after it was projected to encoded into the binding pool and then retrieved was almost 76%. We had SVM-SC shape map activations trained on color labels. So this was to ensure that there's no color information in the shape map. And the accuracy was like 12%. SVM-CC color map activations trained on color labels. That was pretty good again, 84%. And then SVM-CS, that was 17%, meaning that there was no shape information in the color map. And also L2 and L1. So with L2 and L1, you saw the reconstructions weren't as good. Also, the classifier accuracy is much lower, as you can see in these columns. And for L1, it's even worse than L2. So working memory performance is better when information is received in its most compressed or abstract form as it is represented in the latent space. We also tested the model with encoding categorical labels. For example, for a given image of a blue or teal we extracted the label, and using the softmax function and one-hot coding, we encoded both color and shape labels into the binding pool, and we realized that the model can store categorical labels with 100% accuracy along with the visual information in just one memory trace. Now we're part of encoding novel shapes. So I talked about the memory can't store novel information. So this part is interesting. We showed the model Bengali characters. This model was trained only in MNIST data set. It was like handwritten digits. And these were the original images. So if I reconstruct these images from the latent space, meaning that I encode that information from the latent space to the binding pool and then retrieve it, this is how the reconstruction would look like. So the VAE would give me only digits because that's what it's been trained on. However, if I create a skip connection from layer 1 to layer 5, after the input layer and before the output layer, and train it, and then when I reconstruct it directly from the model, this is how the reconstruction would look like, which is more similar to the original images. And if I encode that layer 1 activations to the binding pool and then reconstruct it, this is how the reconstruction would look like. So the L1 is the best layer to reconstruct the novel information, but for familiar information, it was better to encode that from the latent space to the binding pool. In the end, we proposed here a model of memory with visual knowledge. The model was reconstructive. So for a given image, it could reconstruct the image. It could focus on a relevant attribute, whether shape or its color, and also remember a novel stimuli like a Bengali character over here. Thank you for your attention. I'd be happy to answer questions.
Thank you, Shikufe, for the great talk. We don't have much time for questions, but we can take this one. So Brad asked that Ela and collaborators have adapted noisy channel theorem for working memory. Does that make contact with how you're thinking about binding pool or latent space?
Noisy channel theorem. Yeah, well, I am not quite familiar with that specific work, but I'm really interested to see what that is about. But I think the binding pool idea is that, yes, so there is like there is some noise during the retrieval that makes the sort of memory not as good as the actual visual image. Yeah, but I mean, I write it down to like look at that work specifically because I think that would be related to what I've talked about.
So we don't have time for more questions, but Kon has a good question. Maybe you can type right there and chat with.
Yeah, sure.
To answer these questions. Okay, so let's move on to our next speaker, Chun-Yu Tang from University of Wisconsin-Madison. Chun-Yu is a postdoc, and she will talk about her research on investigating specificity in the interaction between working memory and visual perception.
Thank you so much. Hi, everyone. Thank you so much for coming to this talk. I'll jump right into it. Wait. Oh, okay. Sorry about that. So in our daily life, what we hold in our working memory constantly interacts with our ongoing perception of the environment. And studies have demonstrated that working memory can influence visual processing in a bunch of different ways. For example, if you hold a color in your working memory, it can influence attentional selection in the visual search task. And working memory content can influence perceptual sensitivity for low-level visual features such as color and orientation. And in addition, it can even influence the conscious access for new incoming information. And so one potential mechanism that accounts for such a rich interaction between memory and perception is that both memory maintenance and visual processing might recruit the shared neuron substrates in the early visual processing areas. And so one question that remains unclear is how spatially specific this memory perception interaction is. And there are a lot of evidence suggesting that the memory representation can be represented in a global fashion. So if we maintain a blue color, for example, in mind, it will activate all the blue selective channels regardless of the spatial location of where the memory representation was firstly presented. And so if that's the case, we will expect the interaction between memory and perception to be more in the global pattern. Alternatively, there are also evidence showing that the memory representation can have a degree of spatial sensitivity into it. And if that's the case, then maintaining a blue color will actually influence processing at this specific location to a greater extent and will predict a more spatially specific interaction between memory and perception. So in the current study, I want to show you a set of 4 stack physics experiments that looks into the spatial specificity of this interaction and trying to come up with an explanation for the observations of both global and spatially specific representations here. So in the first experiment, we looked at the influence of maintaining an orientation on a secondary discrimination task. So subjects are first presented with 2 gabors on each side of the screen, together with a central arrow pointing either to the left or right. So subjects memorized the orientation that's pointed by this arrow. And then during the delay period, they perform a secondary discrimination task based on the orientation of this new gabor. So this new gabor could appear either on the left or the right side and superimposed by some noise patterns. And so the task here is to make a judgment about its orientation, whether clockwise or counterclockwise compared with the vertical direction. And after they make the response, the memory recall will start, so they are going to press keys to rotate this orientation dial to match with the memory orientation. So the key manipulation here is the congruency in orientation and location between the memory sample and the discrimination stimuli. So they could internally match in their orientation or location, or they could be different. And so we also manipulated the contrast of this discrimination stimulus ranging from low to high contrast, so that we can derive their accuracy in this test as a function as the contrast of it, and use this to derive their contrast threshold that they could reliably detect its orientation. So if we first focus on the feature-based facilitation, right? So how does orientation congruency might have effect on the perception of this discrimination stimulus? We predict that if the orientation of the 2 are entirely the same, the discrimination threshold should be lower than the incongruent condition. This is because the congruency between the 2 in orientation should facilitate the processing of the discrimination stimulus. And then we are more interested in is, how does location congruency might have a modulation effect on this effect, right? So if, according to the global account, we should see this effect regardless of location congruency, but if it's more spatially specific, then we will see a bigger effect when the location of the 2 also matched. So now I'm going to move on to show you the results in this experiment. First, I'm going to show you when the location of the memory sample and the discrimination stimulus are both matched. And as you can see, people's performance were more accurate when the orientation of the 2 were congruent compared with the orientation incongruent condition. And the derived threshold in the orientation congruent condition was significantly lower. So which means that there is indeed this feature-based facilitation between memory and perception at this congruent location. And interestingly, when we turn to the location incongruent condition, we did not find much of a difference between orientation congruent versus incongruent condition. So and if we plot the full derived threshold here, right, full location and orientation congruency, we can see that there is a very obvious location modulation effect here. So there is an interaction between the 2 factors, and suggesting that there is this degree of spatial specificity in this interaction. And we further analyzed, besides from the threshold, we also analyzed the memory precision. So how does perceiving this discrimination stimulus has an impact on the memory representation? And we this time we actually find main effects for both location and orientation congruency, but didn't find the interaction. So it could be that when people are shifting their attention away from the memory encoding location and process and additional information, this has a detrimental effect on their memory precision. So overall in this experiment, we find that there is this certain degree of spatial specificity in this interaction. And so in experiment 2, we carry on and had made a very small tweak on the paradigm. And the goal here is to see does memory still enhances perception when the orientation is now task irrelevant in the discrimination task. And the purpose of this task is to make sure that what we are observing in experiment 1 is driven by stimulus-specific interactions between these memory and perception, and not by some response mode or the shared task demands. And so the memory task is exactly the same. What's different is in the discrimination task. So they first see this discrimination gabor followed quickly by a mask. And the task here is to directly report what was the contrast they perceived during the discrimination task so as to get a direct measurement of what was the perceived contrast in this secondary task. And we predict that if what we observe in experiment 1 is actually due to low-level feature-based interaction, we should see the same level of facilitation here, even if orientation is now task irrelevant. And that's in fact what we observed in our results. So I plotted their reported contrast, so the perceived contrast as a function of the presented contrast. And we can see when the location of the two were congruent, the congruency between the memory sample and the discrimination stimulus indeed facilitated the perceived contrast for the discrimination stimulus. And this effect was not evident when the location was incongruent between the two. And so then what I did was simply just average over the reported contrast for all these presented contrast levels and looked at for the difference for the 4 conditions here. And we see that there is a significant interaction between the 2 factors. So a big difference when the location was congruent and less of a feature-based facilitation when the location was incongruent. And similarly, this time when we analyzed memory precision, we find the same spatial specific effect in the memory precision as well, consistent with the perceived contrast. And so across these 2 experiments, I demonstrated that the interaction between working memory and visual processing can be spatially specific, right? But if we think about the literature, there are also a lot of evidence showing that memory can influence perception in a more global way. And what could that be? So we specifically tested a hypothesis in the next 2 experiments that the context binding demand might influence the spatial specificity of this effect. So in the context when location is more relevant for the task, people might represent the information in a more spatially specific way. And so in the next 2 experiments, we explicitly manipulated the context binding demand. So in experiment 3, we created a task that's very similar to experiment 1 and 2, but with a low context binding demand. So now the memory sample was only presented alone by itself, right? So this is to reduce any competition and binding demand during encoding. And then they perform the same orientation judgment task during discrimination. And then during the recall, this recall dial now is presented centrally on the screen instead of at the memory encoding location. And so we hypothesized that with this design, it should reduce people's context binding demand and even discourage them to use location information. Versus in the high context binding demand experiment, we make the location of this memory sample actually task relevant here. So they need to hold on to the location of this information as well in order to succeed in this task. So what it means is that during the recall, right, now the recall dial could appear on at the same location as the memory encoding location or on the opposite side of the screen. And so if it appear at the same location, subjects just report the exact same orientation that they memorized. Versus when they appear on the opposite side, then they need to report the orientation that's orthogonal to it. So 90 degrees away from it and requires extra mental manipulation, right? And so we hypothesized that when the context binding demand should be higher here, right? Should be pretty high here because the location is relevant. So in this case, we think that we should see a spatially more specific effect. Versus in the low context binding demand, we should be more likely to see a global effect. So now I'm going to show you the results for the low binding demand condition here. So in experiment 3, and this time we find that for both location congruent and incongruent conditions, we see the same exact feature-based facilitation. And it was not modulated, the interaction was not significant. And the same pattern was confirmed in the memory precision as well. So consistent with each other, showing that when the binding demand is low, people tend to represent this feature in a more global pattern. And in comparison, when the binding demand is high, we again see that the memory orientation had an impact on perception. And the difference, the feature-based facilitation was obvious and significant for both congruent and incongruent condition. But more importantly, the magnitude of this enhancement was modulated by the location congruency. So the effect was bigger when the location was congruent and smaller when the location was incongruent. And when we look at the memory precision, when the memory probe, right, appeared at the same location, we see the same spatial-based modulation, but we didn't see it when the probe was different. So anyway, that's all the results I have for the 4 experiments. And so to conclude, in all 4 experiments, we demonstrated that there is this bidirectional interaction between working memory and perception that is stimulus specific. And moreover, how spatially specific this interaction is depends on the relevancy of the location. So if in the context that requires low context binding, people tend to represent it in a more global way. But when location is not as relevant, then there is more of a spatially specific representation. So I think this basically highlights the flexibility of memory encoding, right? We are able to optimize the memory presentation depending on the exact text demand or the context, and to optimize our behavior. So I want to thank my lab, and thank you all for your attention, and thank the organizers of NeuroMatch. And I'm now ready to take questions. Thank you.
Okay, thank you, Chunyu, for the great talk. We have a question in Q&A. Jason says, excellent talk. And he asked, do you think that some level of, some low-level interaction between the working memory stimulus and the discrimination stimulus is playing a role as opposed to working memory storage per se? What if subjects had to drop the item? Would the effects go away?
I think that's a great question. So if I understand it correctly, he's asking, if this effect that we're seeing here can be accounted for other factors like perceptual priming or adaptation. So I think although my current study didn't really have a condition that the memory item is dropped or not relevant, but in my previous work I found that if you're holding on to memory maintenance, it had a different long-lasting effect compared with just perceive the information. So if the feature is not maintaining working memory, it has a brief effect on perception, but this effect goes away with a longer interval. So I think maybe I would say perceptual priming and working memory driven effect might have a very similar neural basis, right? But the temporal dynamics of the 2 processes would be different.
Okay, thank you. Okay, good. It's in Jason is happy to answer, so you're good. So we are run over time, but I would like to thank everyone, the attendees, and also our great speakers today. They did a great job with their interesting talk. So thank you, everyone, and I hope you have a great weekend.
Thank you.
Thank you so much. You too.
Here we go. Okay, fantastic. And we are now live. And so as the attendees come into the room here, I will just do a very brief introduction of this session. So my name is Megan Peters. I'm one of the co-organizers, the many co-organizers of NeuroMatch Conference. And with me here on the technical back end doing all of the tech wizardry is Dimitra Mautsa. And Dimitra has been really instrumental in organizing all of the show running and MC schedules. So I want to give her a hand as well, especially because we are now in the last parallel session. So thank you, Dimitra. And so today, for our last parallel session here at NeuroMatch, we have a fantastic session for you. We have 3 excellent speakers. We have Mengsen Zhang, Stefan Milash, and Alice Schwartz. And so without any further ado here, I think we will just go ahead and get started on the first talk. So the first talk is Mengsen Zhang from Stanford University. And Mengsen is going to tell us about intrinsic dynamic landscape of the brain shaped by multi-scale structural constraints. So go ahead and share your screen and take it away whenever you are ready.
All right. Thank you so much for the introduction. And so yeah, as Megan mentioned, I will talk about how the intrinsic dynamic landscape of the brain is shaped by structure. So when I think about the structure of the brain, I think a little bit more abstractly. I think about a bunch of nodes connected by some arrows that are the structure constraints. And these nodes could be neurons or brain regions or even functional networks. So you can be represented in a structure of multiple scales. And so are the connections between them. So a fundamental question for the brain is, we want to know how these structures are connected to what's going on in the brain, the dynamics. And today I will primarily be talking about how the structure of the brain constrain its function. So first I will give you a little conceptual overview of what do I mean by constrain. And the structure not only constrain a single function, but a repertoire of multiple functions. So I will focus on dynamics as a repertoire. And then I will talk about using a model to show you how the dynamic repertoire of a single brain region can be controlled or constrained by its local connectivity. And then how a large scale network can connect these local regions and synergize their dynamic repertoire. And eventually, I will connect this to the data. In particular, I want to know what is the feature of the dynamic repertoire of the model will reflect something such as the functional connectivity patterns observed in human fMRI. So I will begin with the dynamics of a single brain region. First, why do I say constrain rather than determine? So I want to bring in the concept of multi-stability. Multi-stability means that for the same system, multiple viable patterns are achievable, but which pattern is selected depends on the external input or intrinsic noise and other factors that are not structural. So in a sort of cartoonish way, I think of structure really determines the dynamic landscape of the brain. And what is happening on the landscape are the dynamics that are actually observed. For example, we can say that these 2 points on the landscape represent 2 different patterns of this single brain region. And if you perturb it a little bit, then you will relax back to these patterns of activation. So these patterns are stable, and we call them attractors. And complementarily, if the brain is poised at this point, if you perturb the pattern a little bit, then it will fall into either of these basins, and this is called a repeller. So what is really going to happen in the brain does not only depend on the shape of the landscape, also where are the perturbations going and where the system is starting in this landscape. So it's constrained, not determined. And when we try to visualize or understand, represent how the landscape is going to change as we vary the structure or other parameters, we usually keep track of the position of these attractors and represent them in a lower dimensional space. And that's what we call a bifurcation diagram. So in the bifurcation diagram, the original landscape is sort of compressed into a single slice in this diagram. And the point where an attractor would disappear as the landscape changes is called a bifurcation. So you can sort of estimate the functional complexity of this system by looking at how many colored stripes there is in this bifurcation diagram, which are the number of attractors. The more stripes, there are more attractors, and there are more functional complexity. And now if we use a non-cartoonish model, which actually have biophysical constraints, and it describes 2 interacting populations of neurons, one excitatory, the other is inhibitory, and with some excitatory and inhibitory connections, and some external input to the system. And when we look at their bifurcation diagram, we will notice that if there's an insufficient amount of excitatory recurrent connectivity in this region, then no matter how much input you put into the system, it will always only have 1 attractors. It does not have multi-stability. So we call this a monostable system. If there is more excitatory recurrent connectivity in the system or in this brain region, then for certain levels of input, we will have at least 2 attractors, and this gives the multi-stability of this brain region. So what I've told you is that the local complexity of the multi-stability of the region is depending on its recurrent connectivity, but it also depends on what levels of input other region may give it. So the complexity of the whole brain, you can imagine, will be depending on how you plug these different regions to each other. And that gives us the global network model, where we connect all these local models into a large-scale network with these dashed lines that are representing long-range connections. And as we have more regions and the model becoming a little bit more complex, and so is the landscape. And so here, just to show you some intuition, and this is a toy example, I show you a 2-dimensional landscape that represent the activation patterns of 2 regions, that is the left and right hemisphere. And to be able to do a bifurcation diagram for this landscape, so in a relatively low dimensional space, I just simply calculate the average of the activation pattern for each of the attractors from A1 to A4, fictionally here. But in the real model, the bifurcation diagram will look something like this. And here I'm showing you what happens if the long-range connection between the regions are uniform. So every brain region is connected to other region in the same way. And at best, for certain level of overall strength of long-range coupling, you will have maximally 3 attractors. And they represent really boring uniform activation patterns over the whole brain. However, if you use the human connectome from the Human Connectome project to define the long-range connectivity, the dashed lines here, you will have a very densely populated bifurcation diagram with a lot of attractor for each level of global coupling. So, and these attractors correspond to much subtler patterns of activation than what you've seen on the left. So I've showed you that if you have in realistic long-range connections in this global network, they can greatly amplify the local complexity of the regions. What is also interesting here is that as your activation patterns becoming more subtle, you will notice that the brain regions are not necessarily being activated altogether, as on the left-hand side. So I want to bring your attention back to this toy model, that what I mean is if your brain is starting in this state, this landscape determines not only the repertoire of attractors, it also determines what kind of transitions are possible. If you are here, you can either activate your left hemisphere or right hemisphere, or both together. But if there is some reason that the landscape is undergoing a certain change, if it take the B route, then the left and right hemisphere can only be activated together. If it took the C route, then the left and right hemisphere can only be activated independently. So to quantify how this landscape will constrain how the brain regions coordinate across these attractors during the transition, we can list all the attractors abstractly in a matrix and calculate a rank correlation. And that will give us a correlation coordination matrix across different attractors between regions. And here you have a 0 entry in this spot, means that the left and right hemisphere are pretty much acting independently as they traverse this landscape. In contrast, the left and right hemisphere will coordinate positively if it's taking the landscape is in this shape. But their coordination will be negatively if the left and right hemisphere can only be activated independently across the attractors. So if I take a slice of this bifurcation diagram that's constrained by the human connectome and show the coordination of brain regions across all these attractors, I will get something look like this. So here, the region were indexed. These are all the right hemisphere regions and symmetrically left hemisphere regions. And you can see some large-scale symmetry in this cross-attractor coordination matrix. And this symmetry between the upper left and lower left reflects the similarity between the functional connectivity within the hemisphere and across hemisphere with the homologous regions. And when I look at this, I thought, oh, that looks really similar to the human functional connectivity. But it was very different from some earlier theoretical exploration that I did when I was simply simulating the brain near a single attractor. And this is a best case scenario. And what is fundamentally lacking is a large-scale symmetry between the functional similar connectivity within hemisphere and across hemisphere. And in fact, within attractor coordination, look very much like the structural connectivity. And what is really happening here is that the cross-attractor coordination better captures the nonlinear dependency of function on structure. And finally, I want to say that the transitions does not come for free. So they are associated with certain biophysically interpretable energy gaps. And if we quantify this energy gap in different functional networks of the brain, then we will observe that the ratio of the energy consumption between the default mode network and other networks will be greater than 1 for most of the conditions. It means that the default mode network dominates the energy consumption of the model brain. And this levels of energy consumption, the dominance will be modulated by the local recurrent connectivity. And with that, I want to show some take-home messages. I hope you will remember after this talk that the functional complexity, the repertoire of all possible function stable patterns are shaped by structural connectivity across multiple scales. And that the functional connectivity that we observe in, such as observed in human fMRI data, it might reflect the transitions between stable patterns of brain activities even more than the stable activity patterns themselves. So with that, I want to conclude and take any questions.
Thank you. That was great. And in lieu of applause, because we can't do that in Zoom, if you enjoyed the talk, please drop some praise in the chat. So we have time for a couple quick questions maybe. So if you have a question, please drop it in the Q&A box. But while those are rolling in, I just have a very quick, maybe kind of elementary question, which is this constraint on the connectivity patterns due to the structural connectivity. Mm-hmm. How much flexibility is there? This is all resting state functional connectivity, right? And so like we know that also functional connectivity can change due to task, constraints and so on. So do you have any plans, or how would you build that into your model to actually like push around on variations in this landscape so that you could actually look at functional connectivity as a result of task or task condition?
Yeah, I think that's a very good question. And there are multiple ways to look at it. There are a lot of possibilities of how this could be further studied. And I'm only showing you I would say an elementary quantification of what is going on with this landscape. So what the functional connectivity is really depends on how locally and globally you're looking at the landscape. So right now I'm quantifying the coordination across the entire landscape. So we imagine that will converge to a resting state functional connectivity and would be better for long time recording of functional connectivity. So, but you can also look at this more locally. You can have a part of the landscape and you see where that locally, what type of transitions are possible. But there are a lot of possibilities of what kind of analysis could be done with this kind of framework. But yeah, I'm going to try to do some of it. But there are a lot of possibilities.
Sounds great, thanks. So unfortunately, the other questions came in a little bit late, so we don't have time unless we can get to the end of the session and do them then. but please feel free to answer them in the Q&A box or in the chat as they go through. And hopefully we can get to those. So I apologize for that. But we'll now move on to our next speaker. Thank you, Ming-Sen, for the fantastic talk. And our next speaker of the session is Stefan Mihalash from the Allen Institute for Brain Science. And he's going to tell us about principles of cortical circuit organization. Looks good.
Right. So thank you very much for participating in here. And I want to say that I'm going to present work which has been done by a huge number of people. I'm going to try to mention multiple people along the way, but at the same time, we are part of a large organization which is focused on team science, and we have a lot of data, which I'm happy to be able to just synthesize very, very briefly. I would want to say that as a theoretician in computational neuroscience, I'm very much interested in a model of cortical processing. And I view that as the capacity of linking data relating structure, activity, and computation. And I would want to make a distinction between activity and a computation or a task in order to be able to build models which reflect this. However, it is the brain is incredibly complex, especially the mammalian brain. It's difficult to imagine how much information can be present, even if we were to just simplify it at the level of numbers of neurons and synapses. There are about 10 to the 12 synapses in a mouse brain, maybe on the order of 12 to the 15 synapses in a human brain. Just as a comparison, if we were to just assume a few bits per synapse, a human brain, which is many orders of magnitude larger than mouse brain, it's going to be on the order of information compared to a Library of Congress in terms of the amount of information, such that we already know that there needs to be simplifying principles which are going to be able to link that type of information, because the brain structure does not appear to be encoded with 10 to the 15 elements of information being coded. It's coming from a DNA which is much, much lower in terms of amount of information which is present. So one of the approaches which I think would be very important to try to consider is to first extract what are most important principles, or we can call them knowledge, first at the level of structure, activity, and computation before they are necessarily integrated into a model. Simply trying to imagine that we are going to measure all of these parameters, put them into a data-driven model, turn the light on, and computations would just appear out of that complexity, I think is naive. On that, I will try to think about how we did some small steps on trying to extract some of these organizational principles and to begin at the level of structure. Cerebral cortex can vary hugely in size across mammals, and it's very interesting that evolutionarily, you can sometimes see large brains in branches very close to nearly relatively small brains. And in such a way, there is an expectation that there are going to be relatively simple principles which would allow scaling of part of this structure to allow dynamically building these types of modules which are needed. Some of those are described at the level of cortical columns sometimes, or areas, and the global organization in the past has been described as hierarchically in macaque in a fantastic paper from Fellerman and Van Essen, in which they characterize the patterns of terminations of connections and say that feed-forward patterns have the tendency to terminate into middle layers of the cortex. Lateral patterns have the tendency to be columnar, while descending or feedback pathway have the tendency to terminate into deep or superficial layers. But that assumes that we know what are ascending, lateral, or descending patterns. Now, if we assume that these patterns are of this sort, then it is possible to build a hierarchy of the macaque cortical areas. At the same time, when hearing this, it sounds a little bit of a chicken and egg problem, that if you define the patterns of the connection, you get the hierarchy. If you take the hierarchy, you define the patterns of connections. Question, can we do better about it? Can we try to define more mathematically a hierarchy and to try to understand this more in a data-driven process to be able to extract this principle? In order to do that, we have analyzed the Allen Mouse Brain Connectivity Atlas, which is a fantastic resource. There were many people who contributed to this. Just some of the major contributors are Julie Harris, Jennifer Whitesell, Carla Hirokawa, and under the leadership of Honghui Zhang, who over many years have built an anterograde track tracing method based on two-photon tomography. All the data is integrated. All the data is available on our website. Please come see it. We did analyze these in the past, looking at strengths of connections. However, hierarchy did not appear during the analysis regarding strengths. What was very interesting from our perspective was that we needed to move away from a theory which is based in a type of strengths of connections to one which is centered on types of connections. We need to move away from graph theory and to begin to think about principles of multi-graph theory. What does it mean? It means that instead of simply having one type of connection which you measure in weight, you do have multiple types of connections which can link different cell types or different positions for these columns in the hierarchy. And we characterize these types of connections in the brain. And I was very happy to be joined in this endeavor of analyzing the graph theory by a fantastic mathematician, Hannah Choi, who is starting a faculty position at Georgia Tech next year, and I presume that will look for young scientists to join her lab. Again, the main part for that, what we did initially was to characterize the types of connections which are present, and for that we did an unsupervised clustering of the layer patterns. And we did that for both corticocortical and thalamocortical connections. We did end up characterizing 9 connectivity patterns generally, and one of the most important next steps was trying to build a mathematical definition of a hierarchy. Let's try to go a little bit through that. We want to map a connection to a value which is either of +1 or -1. Let's start with the simplest principle. You can either go up or go down in the hierarchy. And then we begin to look at and mapping the types of connections with the value, which is going to be, does it go up or does it go down? We end up defining a set of patterns at the end, but the way we did it was to try to assign a global hierarchy score. You can think of a global hierarchy score as how self-consistent these patterns are. Are you going to go up, 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 and then down, 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 back? Or are you going to see patterns in which you go up, 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 and you end up in the same position? So in the end, you can think of a global hierarchy score as counting or quantifying how many inconsistencies are present in a purely hierarchical organization. More precise mathematically, we define an average over the number of areas for these values and try to compute a hierarchy for every single area, and then to try to see the self-consistency of the difference between the hierarchy for the areas and the hierarchy for the connections which are present. We did these analyses, data-driven analyses on the very large-scale data set we have, and we have observed that we have statistically a very significant, while it is very shallow, it is nowhere near as what was the expectations based on the drawing from Felleman and Van Essen. The hierarchy is relatively shallow, but it is statistically highly significant. But hierarchy is not the only organizational principle which we found. There is a modularity analysis, there is an intermodule hierarchy, there is an intramodule hierarchy, and we can put all of these principles together. So that was an extraction of principles at the level of the anatomy. Now, very briefly, I'm going to compare it with what we see at the level of activity and computation. In order to correlate it with anatomical principles, we used a large-scale data set based on neuropixels recordings. And we looked at trying to define a functional hierarchy. For example, in this case, it is defined as a function of the median functional delays as to what happens when you present a flashed image. And what we see is that even that the hierarchy is very shallow, it correlates incredibly well with what we see in terms of function. That was only one measure. There are many other measures which one can consider, which is a time to first spike, receptive field areas, modulation index, and multiple other measures, which all relate very well to this measure. Again, remember, this measure comes not from characterizing connection strengths in the brain, only by the characterization of the connection types which are present between areas. And lastly, I'm going to try to briefly mention how we can think about linking this to computation. We did a relatively interesting side project almost as a result of that, when trying to think what would happen if we were to take a hierarchical network, a deep neuronal network, but add two types of plasticity in it. One, we are going to keep the standard backpropagation on a supervised level of plasticity, and add an additional layer on unsupervised plasticity within the lateral connections, which is almost Hebbian. What we have observed is that the networks end up not being quite as good as the original networks, like on CNN on the original images. However, they end up being way more robust to noise. Currently, we have a project which I'm collaborating with Michael Bice on, trying to understand much more generally what are the constraints of anatomy on what are the levels of tasks, and looking for scientists to join us in this endeavor. In summary, I would want to think that it is very important to first introduce principles at the level of structure, activity, and computation rather than data, that we want to think in terms of data-driven, but with an intermediary step of including principles. And to add the fact that one of the principles which we've seen come up all the time is the ideas of cell types and connection types. And for us, when we were to link to some of the in vivo activity and to tasks, not the strengths, but rather the types of connections ended up being more relevant. In the end, I would like to thank the founder, Paul G. Allen, for his vision, encouragement, and support. And again, this is the result of a large team. Thank you.
Thank you. Really fantastic talk. We have time for one quick question. And if you enjoyed the talk, please drop some praise in the chat. So Madine Sarvestani is asking, can you speculate why the avoidance of feedback projections to middle layers seen in primates is preserved in mice, whereas the distinct laminar distribution of feed-forward projections is not?
It's a very good question, and one can speculate quite a bit. I would say that it is a little bit more present than the similarities are a little bit stronger than what is necessarily observed in the data, and one would need to quantify better the data in monkeys because we don't really know that very well. Hannah at the moment is looking at particular models which are going to try to think from the perspective of hierarchical predictive coding, if these types of laminar distributions are going to be important for that. And we generally go along principles of conservation across this, but I do not know exactly why the feed forward can be broader. It might have to do with longer timescales for integration needed for a monkey, but that's pure speculation.
Great, thanks. And unfortunately, we are going to have to move on to the other, the final talk of the session here, but there are two more questions in the Q&A box, and so please do feel free to answer those there as well. And so thanks again, Stefan, for the beautiful talk. Got some nice reactions in the chat as well. And we will go on to our final speaker of the session. So next and finally, we will hear from Alice Schwartz from the University of Washington. And the title of her talk is Motifs for Processes on Networks. And so Alice, whenever you are ready, please take it away. Looks good. Here we go. Oh.
Okay. Do you see the title screen?
Yep, we can see it.
Awesome.
Here we go.
Thank you. So I would like to talk about motifs for processes on networks. This is more of a theoretical work, but it is helpful, I hope, to uncovering the principles that Stefan mentioned in the end that connect dynamics and structure in networks. So I'll start with a one-slide summary. And like Stefan, I'm interested in network structure in the brain and physical processes that can happen on that structure and how that relates to questions of neural coding. So I'm taking a motif-based approach, which means that I focus on how small parts of a network structure influence microprocesses on networks and how these microprocesses in turn are associated with small changes in information theoretic measures. And this is, of course, work that builds on previous work by lots of great people. There's quite a few excellent papers on connecting mathematical models of brain dynamics to information theoretic measures that are used in neural coding. Connecting these mathematical models to structural connectivity is essentially a combinatorial problem, and that's part of the aspects that Mason and I hope to bring to the discussion with our paper. So before I move on, I want to talk briefly about what I think this is good for. Connecting motifs in models of physical processes to information theoretic measures can give us a physical interpretation of the abstract measures that we use, for example, in IIT. And secondly, by making the connection between structure and information, we can get another way to go about understanding the relationship between structural and functional connectivity. So, and here's the pipeline for the rest of the talk. Well, I'll start with the pipeline for the framework that Mason and I are proposing, and then I'll give a brief example using just covariance and correlation for the Ornstein-Uhlenbeck process. And so here comes the pipeline. As I said before, our starting point is that we choose a mathematical model of a dynamical system for neurodynamics, and this could in principle be something like an integrated fire model. In practice, it makes sense to choose a model that is as simple as possible, and that's why we're going with the Ornstein-Uhlenbeck process in a minute. Then we choose a system property, which I call Y, and again, Y in principle can be anything. In practice, we need to be able to obtain a matrix power series expression for Y, and that's not always easy, but it definitely works when I choose Y to be correlation, covariance, mutual information, and entropy for the Ornstein-Uhlenbeck process. Next, we interpret the matrix power series as contributions of microprocesses to Y. And this has something to do with the fact that one can interpret elements of powers of adjacency matrices or connectivity matrices as weighted sums of all walks that connect one node in a network to another. And counting the microprocesses that can occur on a small piece of network structure, we can infer the contribution of a structure motif to Y and from the contributions of microprocesses that happen on it. And finally, using these contributions of structure motifs, we can learn how network structure can enhance or reduce information theoretic measures in a network system and get something like a ranking of motifs and how much they contribute. So let's start with the example. We have the Ornstein-Uhlenbeck process, and we're looking at covariance and correlation in that simple system. Many of you probably already know the Ornstein-Uhlenbeck process and have worked with it yourself. It's a noisy mean reverting process, meaning that when you poke the system, like I've done here and here in 2 time points, it noisily returns to its mean value, which in this case is 0. A nice feature of the Ornstein-Uhlenbeck process is that one can derive its steady-state covariance matrix, which I call sigma, in a very nice form. You see here that sigma is a matrix power series of the adjacency matrix. And this equation tells us that you can get the covariance matrix just by summing over two indices. So we have here uppercase L and lowercase l. And each of these terms corresponds to one summation of some product of matrices. So we can get a similar expression also for the correlation matrix. That one comes out to be a little bit longer, and you have to kind of be aware of more summation indices, but essentially the idea is still the same. So in this, now we're at step 2, right? So we have the matrix power series that we wanted. And before I move on, I want to talk about why this is important. So a graph theory fun fact is that powers of adjacency matrices tell us something about the number of ways that a signal can walk from one point in a network to another. So for example, when we have, when all connections have the same strength, then the ijth element of the, say, fifth power of the adjacency matrix tells us something about the ways that a signal can travel from the node J to the node I. And the fifth power of A transpose tells us something about how a signal can travel from the node I to the node J. And if we have a term like this that consists of A and A transpose, then what that refers to is that somewhere we have a source node, and from that source node, in some specified number of steps, we can walk to node I and to node J. And for the rest of the talk, I draw these out as these kind of walk diagrams with numbers on blue edges indicating walks. And this brings us to the third stage of the pipeline, where we look at the matrix power series that we derived in stage 2, and we find the process motifs that are associated with the summons in the matrix power series. And for the covariance power series, we have seen on the previous slide the associated microprocesses, and they look like this. So here we have the process motifs for covariance, variance, and correlation. And what I like about these results is how intuitive they are. So we see that the process motif for covariance between the nodes I and J, those are microprocesses where a signal can move from a source to both I and J, or just from I to J, which we get when we set this parameter to 0, or from J to I, when uppercase L minus lowercase l is 0. So what this tells us is something that we already know. When we have covariance, statistical dependence between the nodes I and J, it either indicates that there's a causal relationship from I to J, from J to I, or that there's a third variable that causally influences both I and J. So similarly, we get the process motifs for variance from the diagonal elements of the covariance matrix. And in that case, the nodes I and J are the same nodes, so the process motifs look like this. And for correlation, they look a bit more complicated, but they are actually just made up of one process motif for covariance that connects the nodes I and J, and then several process motifs for variance, both at the nodes I and J. And this also makes sense because correlation is essentially covariance normalized for the variance at each node. So now we get to the part where we connect process motifs to network structure. And the main idea is to enumerate all process motifs that can happen on a small structure. And when we do that, we get a ranking of the small subgraphs and how much they contribute to covariance and correlation. And the results, they can look like this. But actually, the slide is really just me flexing that I can compute these small motif rankings. The ones that are actually insightful are the small ones, so where we have just 1 or 2 edges in the system. And so these are just all possible structure motifs with 1 or 2 edges that can contribute to covariance and correlations. The blue ones have a positive contributions, the red ones have a negative, and the white ones have a 0 contribution. Now, unsurprisingly, a single directed edge from the node J to the node I, or vice versa, as we see here in this panel A, has a positive contribution to correlation. But there are structure motifs that have a negative contribution to correlation, and these tell some interesting stories. So we're taking panel J over here. It tells us something. It tells us if we have a network structure in which J sends a signal to I, which we already know from panel A that it has a positive contribution. But if we then add more in edges to I, that decreases the correlation between I and J. And this is again intuitive because from J's point of view, it sends a signal to I, but what arrives at I gets perturbed by kind of noise from other sources. Another maybe less intuitive and more surprising example is when we look at panel K. So we already know that sending a signal from J to I has a positive contribution to correlation. But then adding another edge in the other direction, so we're going both from I to J and from J to I, has an added negative contribution. And this has something to do that this kind of echo chamber effect actually increases the variance in both nodes, and therefore net has a negative effect on the correlation between the two. So to conclude, I'll go back to the slide from the beginning. We use the Ornstein-Uhlenbeck process as a simple approximation of physical processes that might occur on a network of neurons, and we derive process motifs and structure motifs that contribute to covariance and correlation in this model. And in doing so, we found a lot of results that either confirm or extend intuition on the relationship between covariance, correlation, and network structure. I hope to have convinced you that it's possible to obtain physical interpretations for information theoretic measures from a combination of dynamical systems and graph theory ideas. And this approach gives a clear and quantitative link between network structure and information theoretic measures. In particular, we can get a mechanistic understanding of how network structure can enhance and reduce information theoretic measures. And in terms of future work, I'm currently working on getting the process motifs for entropy, which kind of look like this, but there's like a few more details to hack out, and mutual information. And in principle, we can look at other information theoretic measures too. Here I rely on people who are more versed in neuroscience than I to tell me what is interesting here. And I also expect that the process motifs can be helpful for improving methods for network inference. And so if any of this sparks your interest, feel free to look at our preprint or get in touch with me. And that's it for me. Thank you.
Fabulous. Thank you so much, Alice. This was really interesting. And so if you enjoyed the talk, please drop some praise in the chat. So we have time for just a few questions here. We have first some questions from the panelists, actually. So the first question is from Stefan, and I'm not sure whether you just want to ask this yourself or whether I should read it. Please go ahead.
I can easily ask it. I just think that this is absolutely fantastic work, Alice, and linking structure to activity is important. But personally, I think that the main task of a brain is to compute. Do you think, can you speculate on how these types of tasks can be, these types of models can be applied to computations?
I think it helps indirectly with that. So my main idea was to look at things that people use to characterize activity in the brain. And there's a lot of that going on using information theoretic measures. And so my ambition was to get a better understanding of what these measures measure in terms of structure, and thereby understanding how, as Mengsen put it earlier, how structure can constrain what we're actually going to see in terms of dynamics. So these small graphs that I had on my slides, they don't tell you anything directly about what kind of computations are going to happen. But if you believe that information theoretic measures can tell you that, then this is a way to link those back to structure and thereby make a link between computation and structure.
Great. So it looks like we have time for just one more quick question. So in 2 words, actually, let's do 2 questions. Mengsen from the panel is also asking, how difficult to compute higher order motifs? Very, not very?
Not very. So the results that I showed today, the stuff that I looked at in the end, those were for 1 or 2 edges, right? And so that corresponds to kind of first and second order in the matrix power series. But going to higher order is really just keeping more of the terms, and that's what I had on the previous slide. So it's possible. It becomes harder to interpret what you see, because you saw already that there's like a rich kind of thinking of, oh, if I have 2 edges and it's negative, how am I supposed to interpret that? And as you have more and more in there, it gets a bit nitty gritty. But like the computation is not the bottleneck. The making sense of it then becomes the challenge. So if you're into doing something like that, I can always use help in interpreting what the computer spits out.
Fantastic.
Thank you.
And very, very briefly, because we are just about out of time here, Anatoly Bushin is asking in the Q&A, is it possible to compare your theory with the graphs observed in neural systems, C. elegans, connectome, EM data, etc.?
I think it should be. I haven't done that, but looking at how this relates to actually measured network structure is something that I'm really interested in. Stefan knows that I am, so I've been looking to work with actual data sets and do some comparison on the mathematical models and what we find in the data. So yes, definitely.
Fantastic. And I think we're just about out of time, but very quickly, Rainier Engelkind is asking, great talk. Can you comment on how you think these results would change in nonlinear networks in 30 seconds?
30 seconds, nonlinear networks. Okay, so this was for a linear system, as you've correctly pointed out. If you move to a nonlinear system, you can't really interpret things that you see as the sum of small parts, because that's what nonlinearity does to it, right? So what you can do in a nonlinear system is you can linearize around a point that you find interesting, like a steady state or something, and then do the same stuff that I did. If you want to be really in a nonlinear regime and want answers that apply to all of your state space, this is not going to help you. But I think generally a motif-based approach won't be applicable to that, because again, you can't, in a truly nonlinear case, you cannot take something and divide it into parts and say, I know what each of those parts contributed.
Well, that seems like a good note to end on. So thank you very much, Alice, for the fantastic talk. And thank you also to all of our speakers, to Mengsen and also to Stefan for their great contributions. And I also want to thank again, Dimitra, who is our technical backend from NeuroMatch here to make sure everything went well. This was a great session to end on, guys. So thank you for being part of NeuroMatch in general, and thank you for sharing your work with us. And I encourage everybody to skip over to the closing session that's happening in just a few minutes now. So thanks again, everybody. And this was really fun, and I will see you next time.